Hello, yeah, good morning, everyone. For this section, we will going to talk about the food safety, nutrition, quality. I think this is a very important topic, as we know the end user of the peanut is human being. So we will deliver to the end user most important. So for today talk, we probably have five topics here. The first one, we will invite Dr. Camila Nicodoji, from the exercise Hawaii. So Dr. Najura, now is your.
Good morning. So my name is Sam Joroge. I'm a plant pathologist. I'm based in Malawi. So I will give a talk on the current status of aflatoxin mitigation strategies, mostly in Africa. So this is the outline of my presentation. I will start by highlighting the impact of aflatoxin contamination, mostly around trade and also on the health the consumers. And then I will talk about the current mitigation efforts, mostly around good agricultural practices. And then what are the future prospects on reducing aflatoxin contamination in the food chain? And lastly, a few conclusions. So in this slide, what I'm showing is the impact on trade. So starting from 1961 to 2019. The line represented by the bold graph is Malawi. So basically, when aflatoxin contamination standards started to be enforced in Europe, trade really dropped. And countries like Malawi were not able to export groundnuts to Europe. And that really affected the foreign currency that was now really being dependent on in Malawi. But that has changed a little bit. However, what you see is the value of money that is earned per ton is still significantly less. So basically, trade has shifted from Europe to other African countries. So Malawi is no longer exporting to Europe, but it exports to Kenya, it exports to DR Congo, where the value is a little bit lower. And this is also captured in this particular graph, where the blue line shows the proportion of groundnuts that money earned from high-income countries has significantly reduced over the years. And the green bars represent destinations where standards are not highly enforced. On the impacts of health, we know that aflatoxin is the most pervasive challenge in Africa today, especially in the groundnut value chain. And a third of the global liver cancer cases, 40% of these occur in Africa. Aflatoxicosis occurs when you ingest a lot of acute levels of aflatoxin and your liver shuts down. So we've had more than 20 aflatoxicosis cases in Africa. And the worst happened in 2003, 2004 in Kenya, where over 120 people died from ingesting aflatoxin contaminated, but it was mostly maize, it was not in groundnut. And we do know that stunting is one of the health effects that is associated with aflatoxin contamination. And in Malawi, the stunting rates are very, very high. 37% of children under 5 are stunted. Last year, associated with drought, we started seeing a lot of animals also dying in southern Malawi when they ate maize that was contaminated with aflatoxin contamination. So it also affects groundnut. So we are really encouraging people to really go and just buy whole grain where you can be able to see the quality, and you can do the milling yourself at home. When you buy powder, and even sometimes peanut butter, you will not know the original quality that went into making that product. So contamination was very, very high. What we are also saying is that there are a lot of studies that have been conducted over the years, and it is well documented how severe the aflatoxin contamination problem is in Malawi, in Kenya, in South Africa, in Tanzania, and also in Zambia. So to try and mitigate against this problem, we really have to understand that you really have to have a host that is susceptible. You have to have a pathogen that is virulent, and it's usually Aspergillus flavus or Aspergillus parasiticus, and an environment that is favorable for infection and for the production of the toxin. So we've conducted experiments, and most of the contamination usually occurs pre-harvest. When the groundnut crop is stressed by drought, phytoalexins in the plant usually go down. And during post-harvest, how do you handle the groundnut crop after you've harvested it? It's very, very important. In Zambia, what we did was we tested time of planting. So we had time 1, time 2, time 3. Time 1 was at the onset of the rainy season. And then you plant again 1 week later and 2 weeks later. Mulching is just using grass at the base of the plant to conserve moisture, tide ridges, and a control where we didn't do anything. And we also had treatments where we applied manure at different rates, 2.5 tons per hectare, 3.5 tons per hectare, and a control where we do not do anything. You can significantly reduce aflatoxin contamination at the farmers' fields. So these experiments were done on 400 farmers' fields in Zambia. And recently we've had a product for biological control. It's called AflaSafe, that was released from research done by IITA. We've tested AflaSafe. This experiment was done on station where we combined calcium application, fertilizer application, water harvesting together with AflaSafe, and a control. AflaSafe, yes, it reduces aflatoxin contamination, but you still get aflatoxin contamination even with the application of AflaSafe. You have to apply AflaSafe when the plant is just about to flower, and a rainfall event has to occur within a week or two for the fungus to be able to sporulate and to be able to displace the toxigenic fungus that is also in the soil. However, that timing is very critical. And we do know with climate change, it can be a hit or miss. So in as much as it's a good technology, it is not a standalone technology on its own. Sorting is also equally as effective in reducing aflatoxin contamination. And what we are showing here is those two bars were the contamination levels before we sorted. And after sorting, you can reduce contamination by over 90% by just removing groundnuts that are shriveled, that are moldy, that are insect infested. You can reduce contamination by 90%. We also did sorting experiments in eastern Zambia, where we collected about 148 samples in Chipata, 13 samples in Katete, 95 samples in Mambwe. And the farmers were shown how to sort, and they did sort themselves. And what you see is a distribution of contamination between 4 and over 1,000 parts per billion of aflatoxin B1. So when you sort, the proportion of contamination is much lower. And basically, you can eliminate anything that is more than 1,000 parts per billion. However, what you select as bad grains, you still get good grains that have, levels between 0 to 4, 4 to 20. Or however, you increase the proportion of um, um, groundnuts that have more than 20 parts per billion. So you do remove significantly groundnuts that are significantly contaminated. And as you can see, you can eliminate things that are over 1,000 parts per billion. Unfortunately, there are several key challenges in the African setup where we still have to grapple with. For example, we are mostly dealing with underregulated informal market. And what we are seeing is that in those unregulated markets, it's very difficult to reduce contamination, right? Nobody goes out to do surveillance and enforcement. And even in the formal markets, where peanut butter really is a target, you're still getting high contamination level. And there's a lot of home consumption. People grow and consume what they grow. And in that case, it's very, very difficult to reduce aflatoxin contamination from the food chain. So our submission is that aflatoxin continues to be a problem in the peanut trade. And what we see is that peanut trade has shifted from good markets, if you can call them that way, in Europe, to regional markets where enforcement is not really being done. So we do need interventions, both in formal and informal markets. And I do continue insisting that even when you tell farmers to sort out, they can sort it out. But when you sort out, you're removing close to 20% of what you've harvested. And that could be your profit. So we do need to find good alternative uses for contaminated groundnut. You press for oil, and if you refine that oil, you remove proteins, and you can also significantly reduce aflatoxin contamination in the oil. The oil is a very valuable product, especially in certain markets. So we need to put value on sort out. And we also need to move away from, or even move beyond standard. Over 100 countries have standards on aflatoxin contamination, but aflatoxin contamination is still a problem. In Malawi, for example, the standard for peanut butter is 4 parts per billion. 4 parts per billion is very stringent. But in most cases, when you go and sample from the markets, you will find aflatoxin contamination. So there's no enforcement. We can say that we have standards, but that will only take you so far. And we need to create awareness where people demand for better quality. The consumers need to raise their voice and demand for better quality, and that might probably lead to change. We've had projects that have come, and during the span of the project, you get positive effects of reducing aflatoxin contamination. But a lot of this they only last during the duration of the project. After the project closes, people go back to old habits. And so we need to also find ways of really streamlining or mainstreaming aflatoxin contamination beyond project to really help farmers. So that's my talk today, and I thank you for your attention.
Thank you. So we have time for questions from the audience. Please.
Hello. Association of the drought on aflatoxin contamination. Once upon a time, it was theory and myth that aflatoxin is associated with the drought. But in current era, what is your understanding on association of drought on aflatoxin contamination. I mean, in some 2 to 3 years back, it was proven that they are independent. There is no association between these two. But I would like to know your views on that.
Okay. I am not aware of those findings that it's not associated with drought. When drought happens, the phytoalexins in the plant have been shown to reduce. So the plant is more susceptible to infection. And then you also get these cracks that form during drought. And those cracks create avenues for the fungus to be able to penetrate and infect the groundnut. But I would maybe have discussions with you afterwards for that particular information that maybe it's not associated with the drought. Janila. Yeah.
Thank you, Sam. So, how about the use of avirulent strains for managing the contamination?
So that's, for example, AflaSafe, right? So AflaSafe is it's a good technology. What we've seen is that it reduces contamination, but it's not a silver bullet. The way it was being promoted earlier was that when you use AflaSafe, you more or less can be able to control the problem. But from our experience, yes, you get a reduction. It displaces the toxigenic fungus from the soil. But if those conditions are still, you have drought and whatever, you still get contamination. So you need to apply it together with good agricultural practices. You still need to sort. And sometimes when you even, what I was talking about, when you broadcast AflaSafe, you really need to get a rainfall event soon after. Otherwise, you will not get the activation of the fungus. So it is a good technology. In the US, they mostly use it in combination with irrigation. But under our conditions, it's mostly smallholder farmers conditions where rainfall events are not a guarantee. So what do you really do?
Oh, all right. That we move on. Thank you, Dr. Najarka. Yeah.
So now we are inviting our second speaker, Sejal Asija from USDA USA, and she will present on construction and the analysis of the pangenome graphs for Aspergillus flavus and Aspergillus parasiticus to elucidate the genetic diversity and aflatoxin production.
Thank you. Hi, good morning, everyone. I'm Sejal. I'm working under the supervision of Dr. Baozu Guo and Dr. Bob Kemmerer at the University of Georgia. And I'll go ahead and share my presentation. Can you please confirm if you can see my slides? I hope my slides are visible.
Yes, visible.
Okay, thank you. So, okay. So my research title is Construction and Analysis of Pangenome Graphs for Aspergillus flavus and Aspergillus parasiticus to elucidate genetic diversity and aflatoxin production. I'll introduce the topic, I'll go over my hypothesis and research objective, my material and methods, the results that I have so far, and conclusions. So aflatoxins are carcinogenic secondary metabolites which are produced by fungi, Aspergillus flavus and Aspergillus parasiticus. There are 20 different types of aflatoxins known. However, the important ones that we talk about are aflatoxin B1, B2, G1, and G2. Like Dr. Sam talked about it, it's a huge global food threat. There are stringent laws in countries like USA which regulate aflatoxin contamination. For example, for food and feed, we have a limitation to 20 parts per billion here, but the problem still persists and we are still working towards it. So, aflatoxin biosynthesis is the genes which are responsible for regulating it and for the biosynthesis are located on chromosome 3. It includes transcriptional regulators like AFLR and AFLS, which are present here, and then there's genes like PSKA, which is polyketide synthase gene. So it is a multi-step process which requires a lot of oxidation reactions facilitated by cytochrome P450. However, this toxin is not produced by all the species present in this genus. The species that we talk about, Aspergillus flavus and Aspergillus parasiticus, are present in this section called flavi. As you can see on the right hand side in this phylogenetic tree, Aspergillus oryzae and Aspergillus flavus are located very close together. But Aspergillus oryzae is used for making soya sauce. On the other hand, Aspergillus flavus produces this carcinogenic metabolite that we are so concerned about. So even in the section, there is a lot of diversity, and there is very high genomic diversity at species level. So to capture this high genomic diversity at the species level, we can now use a tool called pangenome. Basically, a pangenome represents the entire set of genes present within a species, and it consists of a core genome and a dispensable genome. So when I put it into simple words, if I collect the genome of everyone present in the room today, I'll make a pangenome from it, and the genes that are common between us will be represented in the core genome. For example, the genes that are used for making our organs will be common. And then there'll be certain genes which will be accessory, which reflect the differences between us. So that will form the dispensable genome. And with a decrease in cost of sequencing over the years, it's easy for us to now sequence multiple genomes. So hence, making a pangenome is not that big of a deal. So our lab worked on it, and we made a pangenome for Aspergillus flavus. So basically, our lab had collected isolates from 11 different states in USA. There were 346 Aspergillus flavus isolates, which showed quite interesting results, which showed 59% accessory genes and 41% core genes. And these genes were a part of the accessory genome, and a pan-GWAS study was also conducted, which showed that these genes might be linked to aflatoxin production. Also, this pan genome identified over 1.1 million SNPs, which are single nucleotide variants. But the only limitation with this kind of pan genome was that it was a linear pan genome, and it could not show variations which were more than 50 base pairs. So why do we care about these large variations, large structural variations? Basically, in one of the highly toxigenic strain, which is AF13, there was a 310 kb insertion which was reported, and it was absent in the other reference genome which is being used, which is NRRL3357. Hence, this structural variation was studied, and this 310 kb insertion had genes which are responsible for aflatoxin biosynthesis. For example, this BZIP transcription factor over here, this is involved in aflatoxin biosynthesis. Hence, they concluded that maybe some large structural variations are responsible for for aflatoxin biosynthesis or are linked to it. Hence, we need to look at these structural variants. So before I go further into detail, a pan genome can be divided into two kind of formats. The first format is a consensus FASTA file format, which basically shows up the single nucleotide variants better. For example, you can see here that if there's change from A to T, you can easily see. So single nucleotide variants can be basically represented better using a FASTA file format. Whereas when you need to look at like large structural variants, you need a graph-based format for that, which I am working on basically. So my hypothesis is that constructing these pan-genome graphs will help me visualize large structural variants along with the single nucleotide variants that we were seeing earlier in the first version of aflapan. And my research objectives are to construct the pangenome graph for aspergillus flavus and conduct a pangenome wide study. So I'll be making a pangenome graph subset using the 225 isolates that were earlier used in the first pangenome. And then once I have that done, I'll be adding 332 isolates of Aspergillus flavus and 51 isolates of Aspergillus parasiticus to the pangenome subset. The purpose of doing this is to see if the number of genes are increasing in the pangenome or is it closed. So that'll help us to understand that. So for my material and methods, we already had sequenced reads for 225 isolates of Aspergillus flavus, which were done using short read sequencing using Illumina, and I assembled them using a tool called Spades, and then I did scaffolding with a tool called DragTag. So basically, my pangenome graph subset dataset looks like this. So this is the basic first subset that I've constructed using 225 isolates, and I'll be doing my GWA study with these. And then I'll be adding 332 isolates of Aspergillus flavus and 51 of Parasiticus. So for the construction of pangenome graph, basically we first remove the adapters that are present in the reads using a tool called Skewer. And then once we have those, I do my assembly using Spades and then scaffold the context that I get using a tool called DragTag. And then I check my assembly quality. And then once we have our scaffolds ready, I put it into a pipeline called Cactus, which basically maps the scaffolds to the reference genome. So for example, this black line represents the reference genome, which for me is AF13, which has the large 310 kb insertion that I was talking about. And then I keep adding my scaffolds onto this reference genome. Once I have my pangenome made, so it's very large in size. So what we do is we basically chunk it into smaller pieces using a tool called VG, and those pieces are then visualized on using different tools. So I'm using a tool called Bandage to visualize it, but there are multiple tools that I'll be trying out in future. So I was able to make the pangenome graph for 225 isolates, which is my pangenome subset. And I could see around 700,000 variants which were identified in the pangenome graph, which included both single nucleotide variants and then non-SNPs, which basically are indels and structural variants. Something interesting that I saw on this was that chromosome 3 had maximum number of variants, which is also the chromosome which has the aflatoxin biosynthesis gene cluster. So I'll be looking at the variants, especially the structural variants, more in depth in this chromosome. And then even the non-SNPs or the indels were higher on chromosome 3. So that was quite interesting for me. So I had my tipping pool from the pangenome graph that I made for 225 isolates. So I just went ahead and made a phylogenetic tree out of that. So these are all my 225 isolates. And I could see that 46 isolates were clustering together on the phylogenetic tree. So they had very similar variant pool, these 46 isolates, although they were from different plant parts and they were from two different states also, Georgia and Mississippi. So when I checked the genotyping file, I could see that there's a 2 kb insertion on chromosome 1, and then I could also see an 8.7 kb insertion on Georgia isolate separately on chromosome 8. So I'll be going ahead and doing my gene annotation for these structural variants that I'm seeing, and I'll be looking at more structural variants in relevance to aflatoxin production. So for summary, the pan-genome graphs were able to show me a significant number of SNPs and non-SNPs and chromosome 3. And then I'll be adding Aspergillus parasiticus isolates to the pan-genome graph subset to see if the number of genes are increasing or if it's closed. And I'll be conducting my GWAS study for the pan-genome subset that I have constructed. Okay, these are my references. And I would like to thank my advisors, Dr. Bauzugo and Dr. Bob Cameron, my committee members, Dr. Fountain and Dr. Datta, my lab, and I'm really grateful to Dr. Sunil and Dr. Manish Pandey for giving me this opportunity to present here. I'm really happy to be a part of AAGB. Thank you.
Thank you, Sajal, for your nice presentation. Any
Okay.
questions from the audience?
Hi, Sajal, Sunil here.
Hi, Dr.
So,
Sunil.
very good presentation. So, how many isolates you are using for this pan-genome graph?
In total, I'll be using 608 isolates. 225 is for my pangenome subset that I'm also using for GWAS, and then I'll be adding onto it by like some of the isolates I'm taking from NCBI, which are like publicly available. So I'll be adding all those together.
Okay. So, this
Mhm.
phenotyping is completed for all the isolates you are using?
No, I'll be conducting the phenotyping. Also, I'll be using the phenotyping that was conducted for version 1, and I'll be using the genotyping pool from my pangenome graph instead of just the linear pangenome. And we'll be
So,
looking
the
at,
pan-genome will be used as a reference genome for SNP calling again?
yes, yes, pangenome graph will be used.
Oh,
Yes,
okay.
yes.
Thank you, Sajal.
Thank you, Dr. Sunil.
Thank you.
Right. So our next speaker is Dr. Nagagala, and the presentation is talk about the optimum part size, key, reducing variability, association with aflatoxin contamination while designing experiment.
Good morning, everyone. I am Mangala from Groundnut Pathology, ICRSAT. Today, I want to give you a brief glimpse on the topic of optimal plot size and key to reducing variability associated with aflatoxin contamination while designing field screening experiments. And here, as many of you might know, the peanuts face a major unresolved issue in several countries due to the notorious pathogen Aspergillus flavus. Before we develop and standardize any effective screening methods or techniques to control the aflatoxin contamination, it is crucial to control the aflatoxin contamination. It is crucial to understand the nature and behavior of this pathogen as it describes impacts the plants itself. So let us begin with the basics of this pathogen. Aspergillus flavus is a soil-borne and saprophytic ubiquitous pathogen that causes significant damages in peanut production, and it has a remarkable ability to adapt a wide range of wide range of environments, make it even more challenging to manage the aflatoxin contamination as well. So the major crops, aflatoxin contaminated crops is the corn and peanut, and other crops are chilies and maize, which are severely infected by this aflatoxin contamination. Here, aflatoxin types of aflatoxins are B1, B2, and G1, G2. When it transforms into milk, M1 and M2 in milk production, and B1 is the highly potential toxic and hepatocarcinogenic compound, which is aflatoxin, and that is aflatoxin contamination is highly heterogeneous in nature. It does not spread evenly in a crop or a batch of food. It is due to localized mold growth and the inconsistent contamination levels and environmental factors, harvesting and handling and sampling challenges. Proper sampling here for this, we have to minimize the heterogeneous nature. We have to take proper sampling is essential to ensure the accurate results and to avoid the false and positive or false positives and negatives of aflatoxin contamination. The aflatoxin contamination majorly favored by different factors that occurs in two stages, that is pre-harvest aflatoxin contamination as well as the post-harvest aflatoxin contamination. Here, the factors responsible for the pre-harvest contamination means that pre-harvest contamination occurs in the crop at the time of the crop stage or before the crop harvest. That is the factors imposing responsible for the pre-harvest contamination is the environmental factors, mainly that is the high temperature, moisture content, and drought, and as well as insect damage and susceptibility of the genotypes and some of the soil and agronomic practices like soil type and poor soil fertility and the improper or farm practices. And also the crop growth stages, that is during the crop growth, if the crop is imposed to drought stage, it causes the aflatoxin contamination, and the presence of highly toxic strains in the soil and in the environmental and genotype interactions also will play the role in the pre-harvest aflatoxin contamination. Whereas in post-harvest aflatoxin contamination, it occurs generally in storage conditions, and it is due to the high temperatures and improper drying and presence of using the contaminated bags that leads to the post-harvest infection of this aflatoxin contamination of flowers and leads to the aflatoxin contamination. And to minimize the pre-harvest aflatoxin contamination, we have to follow our good agricultural practices like crop rotation and timely planting, proper fertilization, especially application of the calcium is important for the good agricultural practices, and water management like means avoid the excess irrigation and minimize the drought effect during the crop season period, and insect and avoid that insect and nematode means damage, and so improve the soil health management and improve harvest and pre-harvest handling as well as the storage conditions. Here the main important factor is for a pre-harvest aflatoxin is the host plant resistance. In host plant resistance, generally we will use this, in vitro as well as the field screening. Here the field screening to parameters we take, that is say, kernel infection, that is a percent means flavus infection as well the pre-harvest aflatoxin contamination through LS, and IVS means in vitro seed colonization, and it is used in artificial inoculation methods, so used in the laboratory conditions. And then for here it is the main constraint for this for host plant resistance is screening of resistance is not only because it is highly influenced by environmental and pathogen in interactions. Reliable phenotyping standardized sampling processes are also critical for success. And here at ICRSAT we screened here at ICRSAT we screen for field screening under the sick plot conditions where the soils are artificially inoculated with the Aspergillus flavus inoculum, and the inoculation is done inoculation is done multiple times during the crop season, that is after 35 days or 30 days after inoculation, and then with the different 3 to 4 split doses, and imposing the drought stress 3 to 4 weeks. Here and after this we will go for we will go for the quantification of the aflatoxin as well as the Aspergillus flavus infection. And for a screen, but we have to face major challenges in effective screening requires is uniform levels of fungal infection. If the environment, major challenges here is the uniform level of the fungal infection. That is another one is the environmental factors. If during environmental days favors the aflatoxin development, even susceptible cultivars also sometimes, if the environmental factors are not suitable for that aflatoxin means A flavus infection, and the susceptible cultivars also not means show the aflatoxin contamination. So this result making it is hard to detect the differences. To overcome this overcome this, we have to select the light and sandy soils as well as post-rainy season. We have to select the season like post-rainy season. Here, one of the here the constraints in analysis of pre-harvest aflatoxin contamination data is one of the biggest bottleneck is the high variability in the aflatoxin contamination levels. We cannot practically measure contamination across the entire field. So we have to go for the sample small plots, and however the sampling errors accounts for 90% of the variability from the sampling size, and the 10% is coming from the sampling processing. So that here even the aflatoxin contamination shows the less means a low level of aflatoxin contamination, but the range of the standard error and the standard deviation is the more. So it shows the huge range, and it makes it difficult to draw a conclusion about the treatment's effects or genotypic resistance. And this is where the plot size becomes critical, and larger plots may reduce variability by capturing more representative samples. For this, here our goal was to determine how plot size influences aflatoxin variability. If we can identify an optimal plot size that minimizes the sampling error, and we can conduct more efficient and reliable screening trials. And ultimately, this will help in selecting resistant lines with the greater confidence and improve our understanding of aflatoxin dynamics in the field. Here, earlier, field screening was conducted in generally is randomized block design. Next, we shifted to alpha lattice to screen the large number of genotypes for aflatoxin contamination at the same, single season or same time. And then, even though we have used this latest alpha lattice design, we could not able to analyze the data because the variability in the replication to replication. So we didn't we are not able to draw any results from this. So recently we used the randomized block design by using the large plot sizes. That is, we used a large plot size of 70 meter for one single genotype, and with splitted with subdivided into subplots, that is 1.5 meter subplots. And then we have here each subplot was his subplot parts means subplot parts were collected, and the kernels were used for the estimation of the aflatoxin content. Here for our preliminary, we have carried out one preliminary experiment, that is using the small set of resistant cultivars, as well as the some moderate to high susceptible varieties. Planning for this, how the means variability differs in the field aflatoxin contamination. So here we have taken 6 magic populations and 1 released variety, and 2 checks for this study. We used with RCB, means randomized complete block design. Here we have taken the at the time of harvesting, we have collected all subplots of the individual subplots of pots were collected, and the kernels were used for the analysis for aflatoxin estimation. So here, based on this individual plots, means that a total 9 genotypes with the 3 replications gave that 1080 subplots. So all the 1080 subsamples kernels were used for used for analysis, and here is a simple statistical analysis of variance was performed to understand the standard deviation and assess the variability in aflatoxin contamination levels. Here, these are we have shown that individual lines in a 70 meter length, and we have split it into 1.5 meters like this. Here, the analysis of variance it shows that shows the means aflatoxin levels varies ranges from the 0 to 99.78 is the means ppb is the aflatoxin levels, and the standard deviation is at 6.490. And the means this the linear ANOVA model was analyzed based on this formula, that is y equal to means of all the aflatoxin content plus genotype plus and replication and error. Here the analysis shows that there was no significant differences between the replications, and but we found that there was a difference, significant difference between genotypes. Here the genotypes used as the genetic trait, and wild as whereas replication as the design trait or field trait. So the replication trait will show the means non-significance, that is the homogeneous to the homogeneous. So all the 1080 subplots means data was used for analysis of variance, and the standard deviation is the 64.90. Here we used this formula for individual plot size. Individual plot means we have used for the standard deviation, how the if the plots was increasing, how it was the plot size was increasing, how the standard deviation was decreasing. We have plotted that one. That is based on the Sn is equal this formula. Sn is equal to n by means small n by n into Sn. That is Sn is the standard deviation, n is the sample size. And here if it is the plot size is 1 means that is we consider as the 1, and that is 1.5 meters 1 by 1 into total of standard deviation. Like this we have calculated. For two plots we have considered as the one single plot means that is 1 by square root of this 1 by 2 into, like this we have used for the each subplot for increasing the large increasing 1 by 1 and addition of sequential addition of this plot size, and the standard deviation and standard errors we have calculated while using this larger means increasing the larger error field, larger size of this one, the standard error and the standard deviation was decreasing. Here, based on this the data, we have standard deviation and plot length. We have to means we have made for a graphical representation. Here it is the elbow curve. Here the point where the upper of this means the variation between the standard deviation is more, and whereas it is coming down, it is the standard deviation errors is very less and minimal. So at the point where the reduction was started means there is the 9 meter length of this plot size. So this consider as the optimum plot size for this study. And here the different genotypes, only 3 genotypes were showed the very high variation, high contamination levels. That is the ICGM, ICGM 18-27758 and ICGM 18-2893 along with the JL24. Here we conclude that standard error decreases up to a plot length of 9 meters, and 9 meters row length is identified as the ideal plot size, showing minimum standard deviation. It is the preliminary study we used for this where the plot size plot size will impact the variation in the aflatoxin contamination or not. And it needs the further, it is the only one season data. It needs further confirmation also. Thank you.
All right. This is a good, very interesting topic about the optimum size. Okay. Any question now from the audience?
Yeah, I think with respect to the plot size in your current model, you had a plot, you had a long plot strip, a strip of a 70 meter, and then within that you divide it into subplots, and then you are analyzing the data. You are using the RCBD for that. I think I would suggest you to use either strip plot or a split plot design for that.
Split plot only, sir.
Because it's I think you are analyzing by RCBD.
RCBD.
It's better to use either split plot or strip plot.
Because the variation between the replication to replication is very high, so we are not enabled to get any data. Only we are presenting this range and aflatoxin of main one previously. Now only sometimes we are giving the standard deviation. General, if it is normality testing, it is giving, it is a non-significant, means it is not giving the proper results. So we are not unable to analyze the any other analysis models. That one only you ask or some other thing. Okay, thanks.
So I have a question about your ANOVA table. Can you go there, slides? The ANOVA table. So I have, yeah, here. So I just wonder what's the degree of the freedom? My question is your ANOVA table here. What is your degree of the freedom over there? So I have come confused about this table. So the variable you wanted to test is the plot size, by the what is the degree of the freedom for product size.
Product.
So you are saying trait is the aflatoxin concentration, but that is not your main purpose for test that variable. Variable you're going to test is the plot size.
Plot size, you're asking.
Yeah.
Plot size means each plot size we have first calculated, and then we have added sequentially. Sequentially, we have taken the standard deviation. Means adding 1 plus means sequential adding means 1 plot plus 1 plus like that we have added. Means 1 plus 1 means first is a single plot means 1.5 meters, and then we have added 2 plots means 3 meters, and 3 plots means 9 meters. Like that we have added, and we have done the analysis means calculation based on that one standard error, including that one.
All right.
Along with that total standard means 1080 plots standard deviation using that one.
All right. Okay. Thank you. So let us move on for next speaker.
So we are inviting our fourth speaker, Priya Shah from Osmania University, India, and her title is Revealing Peanut Blanchability, Genomic Insights for Identifying Markers and Candidate Genes to Improve Quality and Boost Industry Impact. Now, Priya, the floor is yours.
So here, myself, Priya Shah. I'm a PhD student from Osmania University, and I'm working at ICRISAT under the guidance of Dr. Manish Kumar Pandey. So here, I'm going to present a glimpse of my topic, that's peanut blanchability, genomic insights for identifying markers and candidate genes to improve the quality and boost the industrial impact. So, as we all know, peanut holds a great economic importance because of its nutritional significance. As we all know, it has some 52% of oil content, protein content, 32%, and several other important nutrients, calcium, vitamins. When it comes to its anti-nutritional factors such as trypsin inhibitors and phytic acid, it affects the peanut. So this can be inactivated by boiling or roasting. Apart from this, several peanut products which are important like salted peanut, raw and roasted peanuts, and these products, groundnut products, are important as per the industrial significance. Now, in the last 5 years, there has been a increase in the production of peanut. As you can see, China is the biggest producer of peanut, followed by India. Here, as this slide, you can see the China holds a production of 18 million per ton, and the yield is 5 ton per hectare. In the area, 5 million per million hectare. Now let me introduce my trait, that's blanchability. In the processing of peanut, the most important thing is the removal of seed coat. Here where the trait holds the important. Blanching, which refers to the removal of seed coat. The genotype holds difference in this ability to release its testa. This ability is refers to as blanchability. The blanchability has been governed by several factors, which include genotype, seed quality, harvest date, level of maturity, length of time and temperature of post-harvest storage. These factors govern blanchability. Peanut blanchability holds economic importance because of the production of several products which include butter, candy, salted peanuts, cookie, peanut bar, roasted peanuts. In spite of these blanchability is a tricky trade because some industries, they require some poor blanchability lines because for products like beer nuts and some confectionery, seed-based confectionery products. So here the requirement is based as per the processed product. Now in my study, I have utilized the germplasm which consists of 1704 fold collection followed by 300 reference set. And from that 300 reference set, we have made a panel of 184 accessions. These 184 accessions represent the diverse germplasm from different countries of the world. These reference germplasm represents the 1 to 8% of entire germplasm and holds a great genetic diversity. So yeah, this mini course hat comprises of 184 collected from different parts of the globe. Now, as these peanut holds a large genome size, so it necessitates the development of array which possess the uniform coverage. Yesterday, my colleague, Mr. Vinay Sharma, has discussed, like we have designed several arrays which include Axiom LHS array, then WGRS. So this is how this 58 Axiom LHS array has been developed for this 184 mini-core collection, and which has led to the identification of some polymorphic SNPs that we have utilized in our GOA studies. So this will be the workflow of my study. We have carried out some peanut mini-core trials, followed by phenotyping, then generation of data, phenotyping, and the final analysis, and ultimately leading to some diagnostic markers and candidate genes. So yeah, this 184 mini-core expressions were grown at ICRISAT in 2 different seasons, rainy and post-rainy, with the help of our gene bank. So let me come to the protocol for calculating this blanchability. Firstly, the sample is being weighed. Here, the sample size can vary from 200 gram to 50 gram. I'll be discussing about the sample size later, because to get 200 gram of sample size, it becomes difficult to get into 2 replication. That's why we have, like previous studies by Wright et al., they have also suggested like we can go for 50 gram sample size. So whether the results from 200 gram or 50 gram is opt or not, we have discussed these things also in my study. These, after this sample size is being weighed, we will be doing the process of heating, which is carried out at particular temperature for 30 minutes, followed by blanching with the help of a machine called blancher. Then further processing and weighing of these blanched seeds, which will be calculated in 4 different ways, that will be blanched, completely blanched, split blanch, unblanched, and split unblanched. Then we will be calculating the blanching percentage with the help of a formula. In the blanch weight, we will be including both complete blanch and split blanch. This protocol has been published by Janila et al. In 2012. So now let me come to the statistical analysis of my phenotypic data, which suggests that in one way ANOVA, we have found the genotypes have been showing the significant variations when we come to the two-way ANOVA, where we have taken the sample size as one factor and genotype and environment as one factor. So there was a significant difference in the environment, while for sample size, it doesn't hold the significant difference, which ultimately suggests the 50 gram or 200 gram sample size, we can use either of the one. Now, let me talk about the variability of peanut across the different botanical groups. As we have observed, like there are 4 agronomic types in groundnut: Valencia bunch, Virginia runner, and Spanish bunch. But in my study, I have observed the Spanish bunch has been the agronomic type which has showed the maximum blanchability. And while the Virginia runner was the one which was showing the minimum blanchability. So all the genotypes which were not all, like maximum genotypes which were showing high blanchability belongs to Spanish bunch. So here are some good and poor blanchability lines which I have observed from my study. So here, as you can see, all the good blanchability lines belongs from Valencia bunch and Spanish bunch, while the poor blanchability lines belongs from Virginia runner. Now let me come to the GWAS analysis. The GWAS analysis from 58K data has led to the identification of several important genes and markers. These markers were further validated with the help of Intertech platform. In these, like on the basis of the allelic distribution, we have identified some 9 to 10 polymorphic SNPs. Among those, we have, after sending it for validation, we have got some 4 markers which were clearly distinguishing the high and low blanchable genotypes. These marker belongs from chromosome A06, A09, T04, B07. This A06 has also been previously used and detected from the study by Korani et al. This study has also led to the identification of some important gene like fatty acid desaturase, then isocytrate dehydrogenase, and ubiquitin protein ligase, which has found to be a association with blanchability. Similarly, with WGRS data, I have found some 10 polymorphic SNP. Out of that, 2 were validated on chromosome A01 and A05. Now, the identified putative candidate genes, how it has role in blanchability. Firstly, the fatty acid desaturase. As we all are aware of, fatty acid desaturase has role in conversion of oleic to linoleic acid. When the oleic to linoleic acid it has been converted, it ultimately decreases the strength of the seed coat. When the strength of the seed coat is decreased, it affects the adhesion property of seed coat. And the adhesion property of seed coat is responsible for blanchability. So we can say, like, if the FAD's expression increases, which will ultimately will lead to higher blanchability. When it comes to ubiquitin protein ligase, this gene has role in the proteolysis of some important genes like pectin, lignin, which has role in cell wall modification and maintaining the seed coat integrity. So ultimately, when the expression of this gene increases, it might lead to the lowering the high, if the ubiquitin production increases, it ultimately leads to the lowering down of the pectin production and weaken the cell wall, weaken this seed coat, and ultimately the blanchability will increase. Similarly, in the case of isocitrate dehydrogenase, it is involved in the production of, again, seed coat related proteins like pectin, lignin, and which will increase the blanchability. So this is the take-home message from my slide, where I will be concluding all the maximum of blanchability lines belongs to Spanish bunch. And as per the statistical analysis, I can come to a conclusion that 50-gram sample size can be opt for phenotyping of blanchability. Further, the GWAS analysis has led to the identification of some important genomic regions like from B07, which holds some 35% PV. Similarly, with WGRS, we have also got a region A07, 17 with 54% validity, but we were not able to validate it as there was the calls were missing. So we are going to design some allele specific marker for this region, and we will be confirming the validation. Similarly, as I concluded, these 3 important genes which I have found in my study, fatty acid desaturase, associated dehydrogenase, ubiquitin protein ligase, this has found to be in association with seed coat. So yeah, again, final conclusion of these particular 6 markers which have been successfully validated by CAS genotyping assay, and the lines which are showing good blanchability are these few lines which we can consider for good blanchability lines. Here, I would also like to know, I have recently published the first ever review article on blanchability. It has been published 2 days back. So kindly have a look on this and read about this article. You will get more idea about the blanchability. So I would like to hold a sincere gratitude and thankful to various people, including Dr. Kuldeep Singh. With the help of GeneBank, we were able to conduct our trials. Then Dr. Janila, she has been like with that blanching calculation and all. Dr. Manish Kumar Pandey, my supervisor, for providing me this opportunity Dr. Sean Mays, Dr. Prashant Singham. Thank you, everyone, and the genomics team for continuous support and encouragement. Thank you, everyone.
Thank you, Priya, for the nice presentation. Any questions from the audience? Yes.
I have just one point for the discussion sake, not specifically for the Priya. Because we are trying to remove the seed coat for the blanchability, but the seed coat has a lot of resveratrol content, which is a nutritional very valuable one. So instead, can we, and then like in our germplasm collections, we have a white seed color. So can we not have instead of tan seed color, can we not have a white seed color and retain our seed coat also so that it's more nutritious? Thank you.
Sir, I would like to conclude on this point. Because for some industrial product, we need to roast peanut. And in the roasting of peanut, sometimes there is a secretion of tannins, which leads to off flavor, which is not good for industry. So that is the reason they prefer good blanchability genotypes based on the industrial product requirement.
Nice talk, Priya.
Thank you so much, sir.
So, how about this trait across the environments? How this trait behaves across the environments? Have you done any studies?
Yes, sir. As per my observation, what I have found is in rainy season, the seed is generally shrinked. So ultimately, when the seed is shrinked, it affects blanchability. So with the rainy season, also for the sample size, we have to struggle a lot to get a sample size of 200 gram. Because when we go for a collection of this sample size, we follow the grading method in which we use the material which has been with the grade first, like which is the more round seeds and more proper seeds. Because if it is not there, then when we will process the seed to blancher, automatically if the seeds are very small or very shrink, it will be passed off from the blancher. It won't get blanched. So yeah, I feel there is effect environment.
I have one question. Stability somehow correlates with the size of the seed and the size of the pod, maybe?
Yes, sir, it would. Because when we were going for some seeds which were bold, round, the blanchability was more. Because I think as per the machine has been designed, this this bold seeds, they were able to generate that friction and it was easily getting removed. But when it was coming to some long seeds, those were easily getting passed out from the blancher. This was my observation. But yeah, I need to confirm with the experiments, or I need to confirm with other studies like correlation analysis, something. I'll do that, sir.
Thanks.
Priya, here, first, I congratulations. This excellent talk.
Thank you so much, sir.
My question is about the branching time. So have you taken consideration of the different seed size, also the different maturity seed?
Sir, I have not done that, but yeah, we do have some previous data for that. So yeah, I can correlate with it. Actually, I have done some correlation analysis, but the results were not that amped, so I didn't kept it here. I need to work on that and make it more significant. Thank you so much, sir. Thank you for the suggestion.
Okay.
Uh,
Thank you.
So I want to add one point. As sir told about the association of resveratrol with the blanchability, yeah, there is a constant in seed coat. There is the resveratrol is present, but the amount and concentration it is very less. It is around 65 μg per kg, but in seed it is around 650 or around 1790. So the concentration is very high. If we so in this case, like we can ignore the concentration or we can remove the seed coat because that concentration is not that much. So yeah, it can be removed.
Okay, so for next presentation, because the speaker is not here, so we will not have that one. But I will offer you some time here if you have any question about the previous speaker, so you can ask it now.
Since we have some time, I just wanted to take advantage of that. Just a thought to Priya or anybody who can chime in. So is blanchability something, is it a routine, trait that you guys watch out for during making selection, at a post-harvest level, or is there a breeding actually dedicated for high or low blanchability?
Sorry, I didn't get your question. Can you just...
So is blanchability a trait that you just watch out for while making selections or during the post-harvest study as a part of regular, let's say, quality assessment parameter, or is there actual breeding program dedicated for blanchability itself as a trait?
Okay, like, yeah, blanchability is not considered to be that important rate, but we can highlight those lines which have other nutritional importance or other quality traits along with blanchability. So like that, we are going to target some genotypes which also hold some other important economic significance or important traits.
So that's always a parameter that you guys look out for?
Yeah.
Okay. Thank you.
Can I ask one question if I'm audible?
Can I go?
Yeah, I think there's a one online question,
Yeah,
right?
please, yeah.
Let it go online.
Okay, thank you, Priya. This is Tullu, if I'm audible.
You can go ahead. Your
Okay.
question.
My question is, groundnut is different in characters, like some have high oil content and some have not. We can take some as direct consumption and others for oil crush. So while this groundnut is different in its characters and in oil content, in seed size, can we conclude as these cultivars have good blanchability and others have low, while we are testing the same and different cultivars within the same protocol, or we need to have different protocol for those used for oil or used for direct consumption?
Actually, I didn't get the question. Can somebody summarize it for me? I didn't get the question actually.
So can I? Yeah. Tullu is asking whether the protocol remains same or will it be different, because we have two different product profiles, one targeting oil content, high oil, and other low oil and confectionery. So would the because of the oil content, so would the protocol remain same or different is the question he's asking.
I don't think so. The protocol will vary because we are following, like we have followed the same protocol, what has been designed. Yeah, with sample size we have differed, but the protocol would remain same.
Yeah. Hope Tullu, you could hear it.
Yeah, go ahead for your question.
So yeah.
Rina.
So and I have a comment, but before that, just responding to the question raised by Samir, whether blanchability is a trait in the groundnut breeding program at ECRISAT. Currently, no, as a defined must-have trait, because we work on two product profiles. One for is home and oil, and other is for food and confectionery. But then we have defined blanchability as an nice to have trait, because it's good to have, particularly for our food and confectionery uses. So having said that, for doing it, we require about 100 or 200 grams of sample, and it's a destructive. because you're
Yeah.
roasting, the seed is lost. It's a destructive method of doing it. So we cannot deploy it within the breeding pipeline. Where do you get 200 grams of kernel? So consequently, it goes to the latent testing. So at least for this product profiles, which is on for food and confectionery uses as of now. But then if you have a diagnostic marker, and from the study, it looks like that it can be pinned down onto one major gene. So
Yeah.
that will be really helping us to deploy it very early in the breeding pipeline. So that's to respond to you.
Adding to you, ma'am, like in previous studies somewhere long back in 1982, it was considered to be an oligogenic trait. But after that study, they have like concluded it is not an oligogenic. It can be governed by a major gene. I think you might be aware, and you can have in my review as well. I have pointed that study.
Yes. So yeah, thank you. So that's where we are. So we need to move in that direction. That's for that. And going back to the Mangala's presentation and aflatoxin, I think this one question which her work and her team's work is trying to answer on determining the plot size to assess the pre-harvest aflatoxin contamination is an important work globally. So since long we don't have that protocol wherein we have an optimal plot size so that replication to replication variation is not there. So I think I just wanted to make that comment that the outcome of that study would be really very, very important for the global community which is looking at that pre-harvest aflatoxin contamination. Thank you, Mangala and the team.
So is any question from audience? If not, before go to the conclusion, I will make a comment. So I think that all the speaker did a wonderful job. And also, I also interested to see the audience. We have a lot of the question here. That means this is a very interesting talk. So for that, I will conclusion here. So let's give another round of applause for all the speaker for this section. So we'll finish here. Thank you.
So much.
Ladies and gentlemen, we end the session here. Thanking all the speakers, chair and co-chair. Now we'll break for tea around 10:20. We all gather here and start our next session. Requesting all the participants to take your seats, please. We are proceeding with session 2. The session is on functional genomics, gene editing, and other frontier technologies. I now request Dr. Ramesh Bhatt and Dr. James Mololo to be the chair and co-chair of the session. He'll join shortly, sir. You can please take your seat. Over to you.
Welcome again. Start the next session. So this is on functional genomics, genome editing, and other frontier technology. And I would invite the first speaker.
Morning. I'm Naveen Pupala from New Mexico State University. I'm a peanut breeder working mainly on Valencia type peanuts. So today I'm going to talk about dissecting resistance mechanism to Agroathelias ralsii, which is basically Sclerotinia ralsii, in peanut through comparative RNA sequence profiling of resistant and susceptible genotypes. Here I want to acknowledge my participants or my collaborative unit. Pankaj Kumar Verma is a postdoc with Madhusudan Jenga. He works at Texas Tech University. And Arindam Das was a visiting scholar through ICR NHPA program. He came to visit us for 3 months, and that's how we got involved in this project to do this RNA sequencing. Manisha Oja is a research assistant helping with my breeding program. Kaushik Ghosh works with Gunwant Patil at Texas Tech and Venkateshwara Sripati at the Alabama A&M. Rebecca Bennett is with USDA plant pathologist who helps or collaborate on some of my disease screening at Oklahoma University and USDA program at Oklahoma. And Jenga is a new faculty at Texas Tech University. So peanut is globally a significant cash crop. It's widely cultivated in tropical and warm temperate regions. If you look at the production, it reached about 50,000 metric tons. And I just want to give you an overview of how peanuts are used in the US. Most of our peanuts are basically used for the peanut butter, contributes about 56%, followed by our snacks, which is about 19%, and then the candy 16%. The in-shell market is 5%, and the other, which we use as ingredients of peanuts in other food products, comes to 4%. And here you can see everything has gone up compared to 2019, except the ingredients, which is less, but that's overall picture. And I just want to mention most of the peanuts of runner type goes into our peanut butter. It's mainly for the kids, which they use it early morning. That's the quickest breakfast, which can be parents easy to give them half the bread slice with a peanut jelly and a half with the peanut butter. So just to give you an overview of how peanut production is grown in the US, we have 3 main regions. One is the Southeast, we call it, then the Carolinas, and the Southwest. So if you look at the Southeast, we have Georgia, Alabama, Mississippi, Louisiana, Arkansas, Missouri. These 4 states have started expanding, but if you look at it, Georgia, Alabama, Florida are the main, if you look at Southeast. And you can look at, of all the area of the peanuts, 54% of the peanuts come from Georgia, followed by 10% Alabama and 10% Florida. And then Texas is the second largest, you can say. But if you look at the contribution of it, the Southeast contributes 83%, Southwest 7%, which consists of Texas, Oklahoma, and New Mexico. And my station where I do most of the Valencias are grown are located here. It's a small town called as a Clovis. But most of the peanuts, it used to be the largest county once upon a time, maybe 30 years back. It's a Roosevelt County, which used to grow 30,000 acres. Because we have a Valencia market, if you look at now, our acreage has gone down, mainly because the dairy took over. Most of the dairies came from California and all because of the EPA regulations. They were moved out and a lot of dairy industry has come here. So Valencia peanut acreage has shifted from New Mexico to West Texas, these regions. But water is our biggest constraint. So water is getting depleted. We are on an aquifer. So now our Valencia acreage is moving more towards Oklahoma. So when I started in 99, it was mainly here. Then the dairy industry came from 2003 and completely wiped out the Valencias. So they moved to Texas. Now it's moving more into Oklahoma, mainly because of the water. And the other thing is the Carolinas contribute about 10%, which is North and South Carolina. So if you look at the market types, runners are grown here in the Georgia, Alabama, and to some extent in Oklahoma and Texas. Valencias in the New Mexico and West Texas. And then the Carolinas are mainly for Virginia type. Spanish to some extent grows in Texas and Oklahoma. So the reason why Valencias are grown here is mainly because we don't have that growing season. It's only about 120 to 130 maximum. And ours is the initial market. Ours biggest market is the organic peanuts. That's mainly the Whole Foods and other people from the towns. They like organics. That's the biggest market. And the markets for the organics is twice they are paid compared to the conventionals. So that's the main reason why peanuts. Just to give you, like I said, based on the market types, you can look at it. It's all 2-seeded. Valencias are mainly 4-seeded and reds in color. And yields are really not in no way in comparison compared to runners and Virginia types. They are 4 tons. Ours average, the best we can get it is about 3,100 tons. And because if we don't get the rains and all, since it's organic, we don't apply any fertilizer, pesticides, our average yields can go about 1,500 because of organic. But the demand is poor. I just want to stress a few things. Like I said, the US average is 4 tons. Georgia 06 released in 2006 is still the dominant variety. It's not only the market preference, even growers prefer the Georgia 06. And then in 2016, the AU 17 was released, which is more competitive for the Georgia 06. This variety belongs to the, nobody released other than Charleston, who is in our group. I don't know, most of you might have known. He's the one behind it. And one thing I want to stress here is we try to do some transformation work with Dr. Jenga's lab. And I was just mentioning to one of the person here from CIMIT that AU 17 was doing really good when we did the transformation work, that's doing better compared to all other market types. And even Florida DP 13, these were the 2 lines which were, when we tried to do the transformation work, was really performing better. So here is the, if you look at the 10 years average of the production in US, you can see from 2014, 4.4 tons. The peak we had is 4.6 tons in 22, and now last year was not a really good year, so just average per hectare. The challenges in peanut production is heat, drought, winds, and sometimes we get these cold fronts from the north, Canada and all, and that can make, even though we plant it in middle of May, but these cold fronts can be a biggest challenges for us. Once in 3 to 4 years, we have these issues of cold temperatures, and then we can have a really poor stand from this cold fronts which we get it. Disease, since it's an irrigated crop, we don't get that much rainfall. That's the reason why organic acreage is more in our area. And our soil bounds, mainly we see the southern stem blight. We don't see anything early or lately spot, but Rhizoctonias, cylindrocladium are more seen in the east coast. But what I'm trying to tell you is that if diseases are not kept in check, we can lose acreage to the tune of 50%. I just want to let you know that the stem rot, which is also called as a stem rot or the white mold, generally we see reduction from 10 to 25%. In severely impacted fields, it can reduce up to 80%. And we don't have a lot of resistant varieties, very few are available, and that limits our ability to look at it. So especially the Valencias, we wanted to see if there are any resistance. And so one of my graduate student did some work. I'll go over it. And here is just to show you the disease development. So there's an infection stage which occurs. Then in 2 to 4 days, you can see on the base of the stem, you can see the soft rot development on the lower stems. Then you can see this mycelium growth infection site, which is white in color. Then the mycelium starts and they start forming the sclerotia. And this sclerotia can stay in the soil up to 3 to 4 years. So this is a sick plot at Stephenville, which used to be maintained by Charles Simpson. Now, John Kaysen is the breeder who runs there, and we use this facility to mainly screen for all our stem rot disease resistance. And you can see here, this line is a Langley, which is a very susceptible line as a check. And then we send the material. The only drawback is we don't get back seed. So he just rates our materials, which ones are resistant to stem rot. And he has a scale he follows, and then he shares the data with all the breeders, tries to do the screening for stem rot. The reason why we started this study was one of my master student did some preliminary work on looking at the 105 accessions for Sclerotinia ralstii. And she used 3 lines, Georgia 03, Valencia C, and then this was published last year. And I'll just go through it. What she did it in the greenhouse, she tried to screen this 105 lines under 4 replications. And then she did the same thing under using the incubator. Under 2 replications, repeated it 2 times. So we got 4 replication data. The good thing about the incubator study is the temperature and the humidity can maintain consistently. And this is how just to look at how it looks at. We have 4 shelves with a humidifier here. So we try to do it. The only problem is we almost ruined the electronics of it by placing a humidifier inside it. And so that has become a big challenge for us. And we followed the ratings which was developed by Lee et al., where 0 is no infection or no disease symptoms. 1 is you saw visible outgrowth. 2 is symptoms with visible outgrowth. 3 is you can see the mycelium and partial wilting. And 4 is complete wilting. And the disease course was done 3 to 17 days. And here is how we measure the lesion length. And then disease score, as I said, 0 to 4 was rated. So we found from that core collection, you can see this is our resistant check we used, and you can see some lines in the PIs, which we found pretty interesting lines, which were lower than the disease score coming to 1.3 to 1.5, and these are the PI lines, and lesion lengths were also lower than the resistance. So what we did is we tried to build this kind of a chamber, which costed us just about $500. And this worked really good for our initial screening of any breeding materials. This is how we screen it. We use different methods, the slurry method and all. And then this is how we can, for someone who is interested, I can for it. And this really worked good for us. And this is just to show you the different way. And this is how we have a lighting inside it and a humidifier. And it works. The only thing is it is a little intense because in real life, you don't see this much intensity, but we can modify it, adjust it accordingly, and then get this done. So with this, I want to just introduce you. This work was mainly done at the Jenga's lab, just to look at the genes involved in the comparative transcriptome analysis. And coming to the material methods, this is what we grew it inside that growth chamber, in the incubator. These are 6-inch pots. We had 4 replications. We used 2 varieties, basically. And the Sclerotinia strain was obtained from one of the fields where we grow peanuts. And we cultured it on the dextrose. Here is 1 plate. The method which we developed is 1 petri dish in 100 ml makes a good slurry. We try to mix it in a grinder with 100 ml of water and make a slurry and apply 5 ml per pot, and it worked really good under 3 replications. And these are the O3 control, Georgia O3 control, infected plant, Valencia control, Valencia infected. And then these are the methods we followed: RNA extraction methods, raw read, read, and then count normalization. And then this is how we got the potential candidate genes. And just to go over the results, you can see the read counts indicated consistent sequencing depth across all samples, and the reliability and comparability. You can see the analysis across all the combinations or the treatments of Georgia O3 infected control and Valencia control. And if you look at the replication, all of them clustered together, which was good. That indicates that each sample clustered together. And then this is how we got the differentially expressed genes, upregulated genes, and the downregulated genes. And you can look at Georgia O3 infected versus control. The differentially expressed were 1,000, upregulated was 89, downregulated was 142. And if you look at Georgia O3 infected versus the Valencia infected, it was pretty high, close to 5,000, 3,000, and 1,500. And the middle one is Valencia infected versus control. And here is Georgia O3 had the least number of genes suppressed by fungal infection, and the fungal infection strongly affected the Valencia susceptible by altering the gene related to the normal plant metabolism. Here is, if you look at the upregulated genes, you can see it. There were 534 genes which were upregulated, followed by the 54 genes highlighted resistant response potentially linked to the pathway specific to the resistant cultivars. And the shared genes were 289 and 12. This one I'm talking about, that there was conserved responsive across cultivars, indicating co-defense or stress response mechanism in all conditions. And here is the gene ontology, upregulated, differentially expressed, highlighting key biological process with the gene expression changes, and the response to biotic stimulus, cell wall modification, glutathione metabolism, chitin metabolic process regulated in the resistant cultivar O3 after infection. And the gene ontology enriched the downregulated of the GEs, highlighting key biological process associated with the gene expression and response to biotic stimulus and sugar metabolic suppressed in Valencia C after infection. So here is the gene expression used by the Sienna model, and you can see the red, which is a resistant infected one. You can see the red one, higher expression levels was seen compared to the green where lower expression levels. Gene expressed in Georgia O3 infected are identified as the unique genes associated with Ralstii resistance. So these are the biotic stimulus response to oxidative stress, cellular homeostatic significantly enriched the Sienna4 gene module. And here is the genes which are involved in the Ralstii response. And these are the 15 genes we identified, and the functions are on the right. And these In conclusion, you can say uniquely upregulated 534-54, and these were resistant mechanisms involved in pathogen recognition, cell wall modification, and distinct defensive pathways. Strongly affected Valencia C with suppression of sugar metabolism and biotic stress response related to the genes. And Sienna model genes were linked to the biotic stimulus response, oxidative stress, and cellular homeostasis are critical for response. So the defensive proteins were 6SF5PD cytochrome, P450, and pectinorase inhibitors. Regulators were zinc finger proteins and actin depolymerase factors. And functional proteins were protein kinase families and peroxidase oxidoreductase. So these findings will contribute to the development of peanut cultivars to enhance the blood sugar oxide. I would like to acknowledge our funding agency, mainly the New Mexico Agriculture Experiment Station, the Peanut Research Board, the National Peanut Board. The Texas University was instrumental in funding the postdoc, and then Arindam Das was funded by ICR National Agriculture Higher Education Project. With that, we're happy to take a question. Thank you.
Hello, Dr. Pawala. Can I ask a question online?
Yeah, go ahead.
My name is Akshay. I'm from UGA. Very nice work on stem rot. So looking at your gene expression study, what do you think about the stem rot resistance mechanism? Is it a single gene, single dominant gene based resistance, or is it a multi-gene kind of resistance?
We think it's a multi-gene. If it was a single, somebody could have already, could have been like a fatty acid. You could have changed it, but I'm pretty confident it's a multi-gene. That's a good question. Thank you.
Thank you.
Hello, sir. This is Sunil here. Very good presentation. I have a question regarding this phenotyping. So whether this oxalic acid one is better or the one you use is the better one? What are the advantages for this screening? Since the phenotyping is a key for this.
For me as a breeder, this one is better than the oxalic acid. I feel like it takes the lab. We have the constraint with the people. So that's the main reason we try to go through this. And then it was.
Even the oxalic acid is in the growth chamber only we do.
Yeah, what I mean is to quantify the oxalic acid may take time, whereas this one is much easier what we are. And the other reason why we preferred this one over the oxalic acid was because that study was already done by Dr. Tim Brenneman. So we wanted to look at this method if we can find something common from what Bennett found already, if there are any common PI lines, which makes sense. So that was the main reason why we wanted to look into.
So in case of accuracy or correlation between both the methods, so which one is the better one so that community can adopt that?
Yes, we haven't really correlated because we didn't do the oxalic acid. I can't answer your question, but maybe somebody else who has done both the methods. But I have seen the literature where oxalic acid is used more dominantly. Yes, he presented on it, and then Sudinis and his group may be a good person. But the reason why we picked this one was, as a breeder, for me, screening and looking at the germplasm in a very quick process, this helps me. That's the only reason why we.
Okay, thank you, sir.
Okay, thank you.
Hello,
Yeah,
Dr.
please go ahead.
Kupala.
Yes, go ahead.
This is Bhuvan from Ahmedabad University, and very nice presentation. One thing I would like to know, we could see an in-house built growth chamber in your work. Would you be able to share a bit insight on how you constructed it, in the lab or in your corridors?
Sure.
Little bit
I mean,
disappointing.
if you want to email me, I can give you the details of it. If you are
Absolutely.
looking, yeah, if you're, because we are constrained on the time, that's why I want to it. But I can tell you it's working really good. And
Okay.
it cost only $500. So
Oh,
on
okay,
our end.
absolutely.
Yeah.
It would be
Yeah.
really good. I can email
Yeah.
you definitely.
Sure. Yeah.
Sure.
I'll be glad
Okay.
to share that information.
Sure.
Thank you.
Okay. Thank you.
Yeah.
Thank you.
Thank you.
Okay, we go to the next speaker, Dr. Sejut, who is going to give us a talk on biochemistry, some biochemistry in relation to peanut nodules. Dr. Sejut, welcome.
Am I visible? Sorry, audible. So, very good morning to everybody. First, I would like to thank the organizer and congratulate them for such a nice meeting and giving me the opportunity to present. So, I am a biochemist, a biotechnologist, a little bit odd man out here, but I would like to make sense that how our work on symbiosis is going to help the breeder and can have an impact on sustainable agriculture. So we work on root nodule symbiosis. So I will try to go in more depth. So bacteria, and they have the enzymes, which can actually the only source of sustainable nitrogen source. So before the Haber-Bosch process, we all are relying on this sustainable nitrogen, right? And legume, they evolve this mechanism to have house the bacteria, give them proper food so that the bacteria can give them a lot of nitrogen. So they are also a protein-rich material. So root nodule actually evolved around 110 million years ago. And the diversification and genome duplication, which stabilizes this trait, actually coincide with the dinosaur extinction. So there was a lot of environmental change which forced them to stabilize this phenotype. So like one is model legume, Medica truncata, just because of its resources, we are doing basic research. We have the cropland, and we legume is that Medicago and chickpea, they actually diversified around 20-25 million year ago when nodulation already established well. So there is not much difference in their molecular mechanism. But one thing you can see, though from outside, the nodule structure are very similar, but from inside, so this is a single cell and these are the bacteria. So you can see the shape of the bacteria is pretty different. So the mechanism that guide, they are pretty, still even between Medicago and chickpea, there are differences, not maybe a lot. So this bacteria, they got genome duplicated due to the plant factor. So there are plant protein that goes to the bacterial cell. It promotes endoreduplication. So now the bacteria, instead of one nitrogenase, that is the enzyme Medicago. In case of chickpea, it is around 4 to 5. And in case of peanut, it's actually more. You can see the symbiosum shape are pretty different, but the mechanism are completely different. So I will talk about this mechanism today in the talk. So this is the work mainly with Vikas Raul, who is my first PhD student, and this too also supported him quite a bit. So we started this work long back in 2013, 2014, before the peanut genome was there. So we had a collaboration with Peggy's group from Georgia. We started with the genome sequence variety, TIFF runner. So these are the different stage of the nodule which we take for RNA sequence. So I'll not talk about this work a lot because it's already been published. But what we realized during this course, we actually annotated more than 3,500 gene, which was not annotated. And many of them, they are actually unique to peanut. So just based on our knowledge from Medicago or Lotus, we can't Just simply by orthologous analysis, we can't identify them. So now I will just run a short video that what happened when the bacteria comes. The plant actually endocytoses the bacteria. There is a plant membrane, and then protein, they goes through this endocytotic, through this vesicular trafficking pathway, and which deliver the, which actually promote endoreduplication and expansion of the membrane. A lot of lipid delivery is happening. This is happening for all the legume, and in case of peanut, it is happening in a bit different way. So I already told you, so this is a transmission electron microscopy. This is the symbiosome structure. This is a single bacteria of Medicago, and this is the symbiosome structure of peanut. So it was known from the community work that in Medicago, there are more than 750 nodule-specific cysteine peptide. They were actually going, they're short peptides, around 20 to 40 amino acids. They're going targeted to the symbiosome, and they are doing this endoreduplication. Now the question is, what is happening in peanut and why they're getting this spherical shape? There was some, like I will say, preliminary work. It suggested that a spherical shape will have more surface area for nutrient exchange, and it is actually more efficient symbiosis. So we did transcriptome, and this is the different stage, like 6, 10. So DPI is post-inoculation when we put the bacteria with the plant. So these are the different stage. And when we did the orthology analysis, what we are showing here, that many of the gene which was known in Medicago, including my Priya presented leg hemoglobin, the canonical gene, is absent from peanut. So the NCR peptide, which was there at 750, they are completely absent. Instead of there are defensing like peptide, 55 of them were there. And apart from that, we got a group of protein. This is not a peptide. This is around 150 amino acid protein called CAP. This full name is cysteine-rich antigen 5 and pathogenesis 1. But the take-home message is, in plant, it is known as a pathogenesis-related protein. So this is the orthologous tier 1 protein. So, but we were able to identify, instead of canonical PR1 protein, they do have a signal peptide. And this signal peptide is very similar to the NCR signal peptide. So it was known from Medicago literature that this signal peptide get processed in the ear membrane, and this processing actually target them towards symbiosis. We thought, okay, this probably proteins are going inside the symbiosome. So we isolated the symbiosome using Parkle gradient and did proteomic analysis in packets. And what we able to find out, this non-canonical NCRs, and along with that, this CAP proteins are going inside the. So just to draw your attention, there are 54 nodule-specific CAP. I will talk about them. So we were by proteomics, we were able to identify 22 of them were going. So where these CAP proteins are located, they are located mainly at the end of chromosome 10 and chromosome 20. So they can actually be used as a marker eventually for breeding program. And there are some minor gene are present in chromosome 3, 13, and 8. In this chromosome, they are mainly located. So now, this, as I mentioned, this is peanut specific protein. So they are not present in their orthologs are not present in Medicago or Lotus. So the closest peanut ortholog is Ascinomini evania, whose from got sequence, and also have a very atypical nodulation feature. So we checked with Ascinomini evania. So these red genes, which were expressing in nodule, in case of peanut, they do have expressed ortholog in Ascinomini, and their expression is ubiquitous. And then we can see that this is the whole chunk which is uniquely present in peanut. So we did a large-scale phylogenetic analysis. We took PR1 protein from plants, from yeast, from human even, because they also are very important for human any developmental aspect. But what I'm showing here, these are the genes which is only present in peanut. They do express in peanut and peanut nodule. And this, the yellow ones, they express everywhere in peanut. And these red ones, this is also the same red gene. They were in the synteny with Ascinomini. They also expressed in peanut nodule exclusive. We call them nodule specific CAP. So we did promoter class analysis for many of them. And as I was talking to somebody, so as you can understand that peanut genome is so complex, so we eventually identified the promoters and their elements. So they express in nodule, but in different fashion. We tested this promoter in Medicago. Why we tested it? Because whether there is a similar molecular mechanism, nodule specific is governing it. In Medicago also, where this bacterial differentiation occur, most of them they do express there, and some of them they do express vasculature also. So to bring into the context of biology, we did RNA because we can't do editing for such a big group. There are 64, and among them 54 is expressing only in nodule. So we constructed different RNA construct to downregulate all of them, downregulate specifically like the nodule specific one. And what I'm showing, these are the deformed structure. These are the nodules. So they're forming on the root, but they're not biologically nitrogen fixing, most of them. So we quantified them. The pink one is the one which actually can fix nitrogen. So you can see in the control, and in case of at least 2 of our construct, almost gone. And here there is a lot of like deformities in all of this situation. So now I already mentioned that the symbiosome, these are the bacteria, they are the unit of nitrogen fixation. So inside a single cell, so this is a cartoon representation. This is how the bacteria looks like, but this is not the nitrogen fixing form. When they goes from here to here, they actually become the nitrogen fixing form. So we raise antibody against our this nodule caps, and we try to see their localization. So what we are seeing, they're actually localizing in almost all developmental stages on the bacterioid membrane. So we also used GFP tag protein. So here you can see that GFP tag protein is also showing its expression exclusively on the symbiosome membrane, because there are sometimes artifacts to use anti-GFP antibody also to validate it. So you can see they're on symbiosome membrane. Sometimes GFP give autofluorescence. So now the question is what this protein were doing. But we know from yeast literature that this PR1 protein, PR1-like protein, they can actually transport lipid. They can act as a lipid transporter. So we did a lipid overlay assay. So we purified 4 of this protein. And here what we are seeing, it is binding to PI4-5 bisphosphate, PI4 phosphate. But apart from that, so these are more like signaling molecule. But draw our attention most is this cardiolipin. So cardiolipin is a bacterial specific lipid. It is present in our body in mitochondrial membrane, and it is named cardiolipin because it is a major lipid in our heart muscle, because mitochondria is helping our heart to pump. So this actually draw our attention because they are on the bacterioid membrane, and they're interacting with cardiolipin. So we would like to validate. So it was known that cardiolipin in mitochondria, they actually help in bending of the membrane because of their unique the structure. So it is required for this bending process. So we look for the lipid biosynthesis pathway, which we actually see there is many lipid biosynthesis pathway, whether it's in plastid or in ear, they're actually getting upregulated. And this cap protein, we speculate that they are probably binding to some of this lipid here. And then this DNF1 was known to be localized in ear membrane. So they get processed and transferred to the symbiosome membrane. So when we express them in tobacco, they're no more getting delivered because this DNF1 is not. So we did co-localization. So if we do cardiolipin staining in this bacterioid, they are present in the 2 poles. And when we use anti-cap antibody, you can see perfect. So I forgot to mention it here that there is another phosphatidyl PG, phosphatidylglycerol. It is also very much present on the bacterioid membrane. So we would like to do the negative control. So we use the PG to see whether there is perfect co-localization, though there are many PG-rich bodies on symbiosome membrane, but we don't see a perfect co-localization, maybe a partial co-localization. So we came into this model that there are PG-rich bodies, which was known from literature, and cardiolipin is there on the bacterioid membrane. So here, due to the interaction with this nodule-specific cap or cystin-rich protein, they are actually helping in this bend process. So it is also known that cardiolipin if you destabilize cardiolipin in some pathogenic bacteria, they get endoreduplication. But we don't have any proof whether they are actually promoting endoreduplication or not. So with this, I would like to thank you for your attention. And I would like to just draw your attention, this nodule. They're beautiful structure. They're hidden inside the soil. So we don't pay enough attention. Just some bumps in the root, it's not nodule. So we should, but I'm also trying to have some AI algorithm to differentiate between functional nodule and non-functional nodule, but we haven't reached that. And this work was mainly funded by DBT, and this was possible with help of Peggy because initially she really helped me. And this is my presentation.
Thank you, Dr. Sejut. Let us invite 2-3 questions. Is you have saved us some time. 3 questions from the floor.
Great presentation, ma'am. I have a question about the quantification of the nitrogen fixation ability. For instance, we want to quantify the ability of genotype which can fix the nitrogen so that in the germplasm we will identify nitrogen efficient genotypes. So what is the key trait we can consider for that one?
So first of all, I would like to say that nitrogen fixation is not worldwide. Many groups are now in a position to move forward to integrate it to the breeding program, but worldwide it was not possible to integrate it into the breeding. So what people, and I also believe that what we can use nitrogen dilution method. What is nitrogen dilution? If it is symbiosis, it will take a particular isotope. And if it is a non-symbiosis, then it will take from the fertilizer nitrogen a different isotope. So there are nitrogen isotope dilution machine. We can discriminate between this ratio. So I think the best thing is on seed, maybe 3 seeds for, and that will be easiest because to dig down the root and to count them, we can do it on a mini core population. But to build that mini core population, probably that is what can happen.
Yeah, in ICRISAT, we did a screening of mini core for the nodules. So in that we counted different sizes, so medium size, large size, small size kind of. But then we don't know which are functional and which are not
Actually, I was
functional. So
asking
that is,
Dr. Kuldeep Singh, but
yeah.
when it is 6,000 population, the way we do, if I ask of Miss, or she's going to do
Yeah.
that, right? Very tedious job.
Yeah, to quantify the functional nodule is
Yeah.
a key part.
Yeah, that's why if we are trying that if we can develop some AI algorithm that color, but then you have to have a very good photo, which can be done on flat-bed scanner.
We have Dr. Sunita Chaudhary also she's working in that area. So we can be.
Yeah, I had a discussion.
Yeah. Thank you, ma'am.
Thank you. Thank you.
Any I have a question. It's some GFP expression.
Yeah, the question is like for GFP localization analysis, whether I use a single promoter or multiple promoter. So we did it with ubiquitous promoter, ubiquitin, but at least for 3 or 4 protein, we did it with the endogenous promoter also. So we tried among 54, almost 20 promoter we. So individual. Yeah. So because he did a very good job. Yeah.
My curiosity is towards variability of the different strains of rhizobium and its efficiency in utilizing the photosynthesis of the legume, especially and in arachis here in specific. I recall my reading where they quoted that about 20 to 30% of the photosynthate goes into the machinery of the root nodules. So do you see any scope where using different strains of the rhizobium, which are photosynthate use efficient, lesser utilizing them for themselves and giving more towards say our grain, at the same time. So do you see the in scope identifying those efficient strains?
Yeah, actually, this brings like 3-4 complicated things. So the first thing is, yes, it is possible. There is an enzyme called hydrogenase that that release that utilize ATP, but that don't contribute to the nitrogen fixation. So if it is a hydrogenase less or hydrogenase mutant bacteria, it can give. But NASH that bacteria that is itself is a the second thing is there is a mechanism in plant. Actually, what is happening in peanut, but what is from Medicago, we know there is something called AON or autoregulation number. So whenever there is a photosynthesis limitation, it stops. But on the other hand, whenever there is a nitrogen, then it actually promotes like photosynthesis. So it's like, whether chicken is first or egg is first, that situation. But there is a science paper published in 2022. What they showed, if the plant is nodulated, they did for many. Soybean peanut is also there, then it is early flowering. So if we are actually growing it in the symbiotic condition, due to high photosynthesis, it is giving early flower and early seed. Not they didn't do yield, they shows the timing. So it's very complete. And it's connected.
Okay. I invite the next speaker, Dr. Thompson. China.
So from last 2 days, I'm watching, there are no questions coming from students. I request them to please ask some questions. Okay, for today, we will take maximum questions from students.
Is it ready? Yeah. Yeah, Dr. Thompson. Oh.
Peanut is a widely in general.
Dongshin, you are available online? There and also. If she's there, she can present then.
Hello. Hello.
Yes, Dr. Bochau.
I think another way is that I can play it from my computer here.
Okay.
So,
Morning,
okay.
ladies. Morning,
All right.
ladies and gentlemen. Is a widely cultivated morning, ladies and gentlemen. Gratitude kernel of which I will provide me with the opportunity to present our work here. As we all know, name is acid position is a key factor of the.
So I will play here. So just stop that. Thank you.
Morning, ladies and gentlemen. Firstly, I would like to express my gratitude to the committee for providing me with the opportunity to present our work here. My name is Dongxin Huai from Oil Crops Research Institute of Chinese Academy of Agricultural Sciences, and I will introduce our work on the improvement of peanut fatty acid components through genetic and genomic approaches. Peanut is a widely cultivated oilseed crop, the kernel of which contains 40 to 60% of fat. As we all know, fatty acid composition is a key factor of the nutritional quality of peanuts. As shown in this figure, there are 8 fatty acids detected in peanut kernels, including palmitic, stearic, oleic, linoleic, arctic, oleic, bihinic, and lignoceric acids. Among them, palmitic, stearic, arctic, bihinic, and lignoceric are saturated fatty acids, collectively comprising 20% of the total. Although the recent progress in developing high oleic variety has contributed to lowering the saturated fatty acid content, the total saturated fatty acid content of peanut oil remains higher compared to other vegetable oils, such as rapeseed oil, sunflower oil, and soybean oil. In general, over-intake of saturated fatty acids could raise the amount of low-density lipoprotein cholesterol in blood, which may increase the risk of cardiovascular diseases. Hence, it is highly necessary to decrease the saturated fatty acid content to improve the nutritional quality of peanut. In saturated fatty acids of peanut, arctic, bihinic, and lignoceric belong to very long-chain fatty acids, which possesses more than 18 carbon atoms in the carbon chain. Therefore, we assumed that the total content of saturated fatty acid could be reduced by decreasing the very long-chain fatty acid content. We investigated the variation of very long-chain fatty acids in peanut. We found that the variation of very long-chain fatty acids in wild species is greater than that in cultivated peanuts. Additionally, 3 germplasm accessions characterized by low very long-chain fatty acid content were identified, with kernel very long-chain fatty acid levels ranging from 3.8% to 4.3%. Among them, Luoaowan was selected for crossing with GOA 16, and a real population was subsequently constructed. As shown in these figures, the contents of arctic, oleic, bihinic, and lignoceric in Luoaowan was significantly lower than those in GOA 16. Furthermore, the total very long-chain fatty acid content of Luoaowan was also significantly lower than that of GOA 16. The very long-chain fatty acid content ranged from 4.1% to 8.1% in the real population. Then the real population and the 2 parents were resequenced, and a high-density genetic map contained 3,337 bin markers were generated. Genome-wide QTL mapping for very long-chain fatty acid contents were performed on the novel genetic linkage map in combination with phenotypic data. 3 major QTLs, namely QA1, QB4.1, and QB5, were found to be consistently associated with contents of very long-chain fatty acids, which explain 5.7% to 14.5% of the phenotypic variation. The 3 major QTLs simultaneously controlled both total and individual levels of very long-chain fatty acid, suggesting potential pleiotropic effects or tight linkage within these loci. Based on the variations between two parents, 3 indel variations closest to the peak of the 3 QTLs were selected to design allele-specific markers. After validation in the rails, it was found that only the combined presence of the 3 favorable alleles could distinguish lines with their launching fatty acid content below 6%. In the candidate region of QA1, there was a gene AHKCS1 encoding beta-cateacyl-CoA synthesis, which has been reported to be involved in very long-chain fatty acid biosynthesis. The expression analysis revealed that AHKCS1 was significantly less expressed in LUA1 developing seeds compared to GHUA16. Similar results were also observed in reals exhibiting extremely high and low very long-chain fatty acid contents. Therefore, AHKCS1 was identified as the candidate gene for QA1. A total of 30 KCS genes were identified in peanut genome. The homologous genes of AHKCS1 in the B subgenome was located on the chromosome B10 and named as AHKCS28. Expression profile analysis revealed that the expression levels of AHKCS1 and AHKCS28 were highly expressed in developing seeds, and their expression levels were the highest in the developing seed among all AHKCS genes. Subcellular localization analysis indicating that AHKCS1 and AHKCS28 were located on the ER, where fatty acid elongase complex functions. The AHKCS1 and AHKCS28 genes were expressed in both yeast and Arabidopsis, resulting in an increased very long-chain fatty acid content in both organisms. These results indicated that both AHKCS1 and AHKCS28 possesses fatty acid elongase activities, and they were inclined to use saturated fatty acids as the substrate. It has been demonstrated that AHKCS1 and AHKCS28 are the putative regulators of very long-chain fatty acid contents in peanut kernels, but no sequence variation was observed within or surrounding them. Therefore, AHKCS1 and AHKCS28 were genetically disrupted using the CRISPR-Cas9 system. The agro-mediated peanut transformation system was employed, and the transgenic positive plants were screened using DSRAD2 as a visual marker. PCR identification was performed, and it was confirmed that both AHKCS1 and AHKCS28 were knocked out in 61 plants among 66 T0 plants. The editing efficiency achieved a high rate of 92%. The very long-chain fatty acid content in seeds of AHKCS1 and AHKCS28 double mutants were detected by GC. The peaks of behanic acid and lignoceric acid in the edited seeds were absent, and the overall very long-chain fatty acid content was significantly decreased from 6.8% to 0.4%. Hence, AHKCS1 and AHKCS28 genes are the key genes for controlling the seed very long-chain fatty acid content in peanut. Meanwhile, novel germplasm lines with decreased seed very long-chain fatty acid content were developed. In summary, 3 peanut germplasm lines with low very long-chain fatty acid content were identified. 3 major QTLs for very long-chain fatty acid content were detected. The AHKCS1 is located in the QA1, and its downregulated expression level contributes to the phenotype characterized by reduced very long-chain fatty acid content. The AHKCS1 and its homologous gene AHKCS28 are the key genes controlling the seed very long-chain fatty acid content. The novel peanut germplasm with extremely low wear launching fatty acid have been created through the knockout of both AHKCS1 and AHKCS28 genes. The experiments in this work were carried out by Xue Xiaomeng and Wu Jie. I would like to express my gratitude to my group leaders, to Dr. Liao Boshou, Dr. Lei Yong, and Dr. Jiang Huifang for their guidance and support. Additionally, I also extend my appreciation to our collaborators, to Dr. Rajiv Vashni, Dr. Manish Pandey, and Dr. Edgar Cajon for their invaluable contributions to this work. Thank you for your attention.
Can you take
Yes.
a couple of questions to her? Not. Okay. Is she available there?
Dong Xing, are you available? Maybe she is not here. I mean, this is in China, it's a non-step.
Hello.
Yeah.
Dr. Busher, very nice work. So I would like to know what is the impact of this AHKCS1 on extending the chain of unsaturated fatty acids, such
Can you
as
say that?
linoleic acid.
Mm-hmm. Can you say that again?
So I saw your presentation that about the extension of the very long fatty acids
Mhm.
of AHKCS1. So what
Mhm.
is the impact of overexpression of AHKCS1 on extending the chain of unsaturated fatty acids?
Yeah.
Not
online.
much. Not much. For the you see for the how to say it's there is no much influence on the unsaturated fatty acids. Like the oleic acid is not decreased somehow.
And if you allow me, I can ask you one more question. So you said that you have 90+% of gene editing. So what kind of construct did you use? What is
I'm
the
so
elements of your construct for gRNA promoter and Cas9 promoter, and what kind of Cas9 was used?
Okay, this is I'm not very familiar with their transgenic research. Dr. Dong Xinhui may have may can answer your question. If, I will tell her when I see her again, and we will can let you know the answer.
Okay,
Because
thank
I'm
you.
not contacting that chess for a transformation.
Thank you.
Yeah.
Yeah.
So thank
But
you.
the
Thank you for
person,
the question. Hello.
Yeah.
Yeah. Can we go ahead now?
Yes.
Okay. Okay. So now I'll invite Dr. Yogendra from Nick Research for the next talk. Yeah, Dr. Yogendra.
Yeah.
Yeah, you can proceed now.
Good morning, everyone. Can you see my slides?
Not it. We can see you, but not the slides.
I think I'm able to see Bonshow's screen only.
Yeah, now we can
Yeah, we can see your slide
screen.
now.
Yeah.
Screen.
Can you see my screen now?
Yeah. Go ahead.
Hello everyone, I'm Yogendra Kaledalli. I'm working as a scientist at ICRISAT. Today I'm going to talk on targeting aflatoxin resistant in groundnut, gene discovery and CRISPR-Cas9 based validation. It is estimated that by 2050, world population will increase to 10 billion people. To feed this increased population, we need a large-scale step change in the crop production. However, biotic and abiotic stresses, climate change is likely to have an adverse effect on the global yield of the major crops. In case of the groundnut, aflatoxin produced by aspergillus, which is a major constraint. Aflatoxins are the secondary fungal metabolite produced by fungi Aspergillus flavus and Aspergillus parasiticus. There are more than 20 aflatoxins are reported. Among them, AFB1, B2 produced by Aspergillus flavus, G1, G2 produced by Aspergillus parasiticus are very important. Aflatoxin toxicity, it ranges from acute effects. Sometimes it causes death to the chronic effect. Regulations of aflatoxin varies from country to country. In Europe, the permittable level of aflatoxin contamination is less than 4 ppb. However, in India, it's less than 15 ppb. Apart from groundnut, cotton, sunflower, wheat, corn, rice, they are the other key host for this aflatoxin causing fungus. So far, there is no commercial resistant variety available in groundnut for aflatoxin resistant. So we have to utilize advances in biotechnology to combat the aflatoxin problem. In our lab at ICRISAT, we have used a double defense approach to mitigate the aflatoxin problem. First approach we used is a overexpression of the defensin gene. Defensin is an antimicrobial peptide. When we overexpress defensin, it creates a pores on the fungal cell wall. That way, it prevents the fungal growth. Second approach we used is Higgs approach, host-induced gene silencing approach. Here, the fungal genes were overexpressed in groundnut. When the fungus infect, those genes were silenced. That way, it reduced the aflatoxin production. However, aflatoxin resistant is a complex and quantitative trait. So far, there is no molecular and biochemical resistant mechanism is understood. So to understand molecular and biochemical resistant involved in groundnut Aspergillus flavus interaction, we used a multi-omics approach. This is called as a forward genetic approach or a bottom-up approach. Here we know the phenotype. Our transgenics are giving a low aflatoxin contamination. From phenotype, we are tracing back to the gene with the help of the metabolomics and proteomic work. So for multi-omic approach, we used three different genotypes, three different set of genotypes. First one is transgenic groundnut expressing defensin gene. Here you can see the difference between the control plant and transgenic defensin. The fungal growth is more in the control plant. Same way, aflatoxin contamination is very less in transgenic. The second material we used is transgenic groundnut expressing 4 RNAi. Here the 4 fungal specific genes were overexpressed, which are NSDC, VIA, AFLR and AFLM. Here also aflatoxin contamination is less in transgenic compared to the wild type. The third material we used is a breeding groundnut genotypes. These are the contrasting genotypes for the aflatoxin contamination. 55437 is the resistant, TMV2 is the susceptible. Here, the aflatoxin contamination is less in transgenic in resistant compared to the susceptible genotype. So first approach we did is metabolomics. For metabolomics, we used a contrasting genotype, one resistant. Here, for this study, we used a 4 RNA transgenic lines. Same experiment we repeated for defensin and also for the breeding material. Susceptible, we used this ICGB 91114. We did an experiment at 2 time point, 48 hours and 72 hours post-infection, 2 treatment, pathogen and mark. Total, we got 4 groups: resistant pathogen, resistant mark, susceptible pathogen, and susceptible mark. All the samples used for the non-targeted metabolomics using liquid chromatography, high resolution mass spec. When we got the metabolite data, first thing we did is a multivariant analysis. We used a principal component analysis. Here, PC1 is separating the resistant and susceptible genotype. PC2 separate based on the treatment, pathogen and mark. This shows this experiment is reproducible. After multivariant analysis, we classified the metabolite into 2 categories. One is resistant related constitutive metabolite, resistant related induced metabolite. Constitutive metabolite, they are called as a RRC metabolite. Here, RRC metabolites obtained by comparing the RM by SM. Induced metabolites are produced after the pathogen induction. From our study, we have identified the metabolite mainly from phenyl propanide pathway, flavonoid, fatty acids, alkaloid, and terpenide. Among these, phenyl propanide, flavonoid, and fatty acids are the more significant number of metabolites, and also they produce high fold change metabolite. From phenyl propanide pathway, we observed high fold change metabolites. Some of the examples are ferulyl agmatin, kefilpitrazine, coniferol alcohol, sinapyl alcohol, coniferol aldehyde. From flavonoid, we observed more of petonidine, chalconarengine, epicatechin. From fatty acid, we observed more of omega hydroxy acid, which leads to production of suberin, and also more of the metabolite from sphingolipid and glycerophospholipid pathway. If we see specifically on the phenyl propanide pathway, it starts with L-phenylalanine. L-phenylalanine produced from the TCA cycle. First, phenylalanine converted into cinnamic acid. This cinnamic acid further converted into coumaric acid, caffeic acid, ferulic acid, and sinapic acid. This coumaric acid to the sinapic acid is called as hydroxycinnamic acid. These are the monomer compound. From 1970-1980 onwards, these are reported as an antifungal compound and antibacterial compound. But in our study, what we observed is this hydroxycinnamic acid further converted into HCA-CoA thioester. For example, coumaric acid converted into coumaryl-CoA. Same way, kefil-CoA, ferulyl-CoA, and sinapyl-CoA. This CoA, hydroxycinnamic CoA thioester further converted into their respective aldehyde compounds and alcohol. For example, coumaryl-CoA converted into coumaryl aldehyde, coumaryl alcohol. Same way, coniferol alcohol, coniferol aldehyde, and coniferol alcohol. It helps for the production of the lignin. Lignin is a polymer compound. In many plant pathogen interaction, it has been reported that lignin deposit on the cell wall help for secondary cell wall thickening. Parallelly, this hydroxycinnamic CoA thioester, they combine with amines. Amines comes from the amino acid from the TCA cycle. Here, ornithine converted into putrescine, arginine to agmatin, tyrosine to tyramine. These amines combined with the HCA-CoA thioester, they form the hydroxycinnamic acid amides. These are called as a conjugated metabolite, conjugation of amines and CoA thioester. For example, coumaryl CoA combined with agmatin, coumaryl agmatin, same way coumaryl tyramine. To prove this conjugated metabolite also deposit on the cell wall like lignin and help for the secondary cell wall thickening. To prove this, how these conjugated metabolites and polymer lignin deposit on the cell wall, we did a histochemical analysis. For lignin, we used a fluoroxucinol HCl staining. Here we used a 4 different sample: resistant pathogen infected, resistant mark, susceptible pathogen infected, and susceptible mark. Here you can see the dark pinkish fluorescence, which is more predominant in resistant pathogen sample compared to the resistant mark, susceptible pathogen, and susceptible mark. This shows that lignin is depositing more in resistant pathogen sample compared to other tissues. For hydroxycinnamic acid amides, we used a new reagent. Here the blue fluorescence, which is more predominant in resistant pathogen sample again, compared to resistant mark, susceptible pathogen, and susceptible mark. For fatty acid produced from acetyl-CoA to malonyl-CoA, it helps for the production of omega hydroxy acid. That leads to the production of aliphatic suberin. Suberin is also a polymer. It also helps for the secondary cell wall thickening. Parallelly, we observed the metabolite from sphingolipid and glycerophospholipid. These help for providing stability to the membrane and also acts as a signaling molecule. To prove this, we did a scanning electron microscope. Here you can see the scanning electron microscope images distinguish how susceptible pathogen and resistant pathogen samples differ in the surface of the cell wall and within the cell. Here, in case of surface of the seed coat, here the fungal spores, which is more in susceptible pathogen compared to the resistant pathogen sample. Inside the cell, you can see more fungal spores in susceptible pathogen and resistant pathogen. Further, we did a transmission electron microscope to show how glycerophospholipid, sphingolipid provide membrane stability and help for the secondary cell wall thickening. In case of the resistant pathogen, here you can see cell wall is thickened compared to the resistant mark, susceptible pathogen, and susceptible mark. With this, we hypothesize that secondary cell wall thickening or a cell wall reinforcement is the barrier to prevent the pathogen entry. After metabolomic analysis, we did a comparative proteomic analysis to know what kind of the resistant related or a susceptibility associated proteins are produced. For this, again, we used all three different sets. Here, I'm presenting only on the comparative proteomics from the defensin and comparative proteomics from the 4RNA. When the pathogen Aspergillus flavus it infect, plant produces receptor kinases, then it activate phytohormone transcription factor. Further, it activate the downstream secondary metabolic pathway. Here, in case of defensin, we observe more of phenylpropanide flavonoid and also fatty acid pathway. In case of the 4RNA comparative proteomics, here we observed more on calcium-related protein, heat shock protein, phytohormone transcription factor, and more on fatty acid pathway. Further, we integrated both metabolomics and proteomic data to know what kind of the hierarchy of the metabolite or hierarchy of the metabolite and associated genes are involved for responsible for either resistant and susceptible during groundnut aspergillus interaction. So this Sankey diagram clearly shows how the interaction happens. First one is the groups. Here the RRI, RRC from both metabolomics and proteomics. These are the pathway from alkaloid, flavonoid, fatty acid, glycerophospholipid, and phenylpropanide. Then this group shows how the differentially accumulated metabolite and differentially expressed proteins they form a group. With this, we were able to understand what is qualitative resistant and what is quantitative resistant involved in the groundnut aspergillus interaction. So with this pathway, we classify into a three group. One is signal perception, then the signal transduction, then the defense response. When the pathogen infect, here the Aspergillus flavus infect the peanut, it produces a pathogen associated molecular pattern. To prevent that, plant produces receptor molecule. They are called as a PRR. They are pattern recognition receptor. That resistant is called as a PTI or PAM triggered immunity. It's usually happens on the surface of the cell wall. It is the first level of defense. Most of the pathogen they can easily overcome PTI by production of the effector molecule. To prevent that effector molecule, plant produces the R gene. They code for NBSLR. So this resistant is called as a hypersensitive response or effector triggered immunity or called as a qualitative resistant or a monogenic resistant. Sometimes some specialized pathogen like Aspergillus flavus, they keep on infect the plant by production of more of virulent spores. That time plant activate the durable resistant or a quantitative resistant by production of phytohormone, transcription factor. Further, it will activate secondary metabolic pathway genes from phenylpropanide, flavonoid, fatty acids, and other. That way, it helps for the secondary cell wall thickening. With this integration, we identified resistant related protein, resistant related genes, and also we identified the susceptibility associated gene. Now, this susceptibility associated genes act as a potential target for CRISPR-based gene editing. Before using this validation of the susceptibility gene, first we standardize the CRISPR-based gene editing in groundnut by silencing the reporter gene PDS. PDS is reporter gene. When we silence PDS, we can see the albino phenotype. Here you can see the albino phenotype in the edited plant compared to the control plant. Then the three susceptibility genes are annexin. Annexin is a calcium regulated phospholipid membrane binding protein. Silencing of annexin leads to the providing the resistant to powdery mildew in case of Arabidopsis. Second gene is syntaxin. It's a membrane integrated Q snare protein involved in exocytosis. Silencing of syntaxin also leads to the production of help for the resistant to powdery mildew and Pseudomonas in Arabidopsis, Phytophthora in case of the potato. MLO1 like protein, it's a kind of like a universal susceptibility protein. Loss of functions mutation in MLO leads to resistant to powdery mildew in many crops, barley, tomato, wheat, and rose. We check the expression of all these annexin, syntaxin, and MLO gene in our sample. These genes are expressing more in susceptible pathogen compared to the resistant pathogen. So before going for the editing, what we did is like we targetedly captured all these 3 genes in 178 minicore collection. For this 178 minicore collection, we have the aflatoxin data as well, aflatoxin contamination data as well. So for sequence capture, we used the promoter, exon, and introns region from all these 3 genes. We observed the allelic variation. This allelic variation we associated with the aflatoxin contamination. This is the overview of the sequence capture analysis. This help us what all the which region to target for the guide RNA region. So this slide shows the functional validation for mainly for the annexin gene. So annexin having 6 exons, we designed the guide RNA for exon 4 and exon 5. When we did a CRISPR-based editing, we observed different kinds of the edits, deletion, substitution, and others. If you see the 73 edited line, we observed knockout score of 26%. But now we need to do the phenotyping for this phenotyping for this line for the aflatoxin contamination. With this, I would like to thank Team ICRISAT and our collaborator from Department of Primary Industries, Australia, and Western Sydney University, Australia. Thank you.
Yeah, thank you. Thank you. Thank you so much. Can you take 1 or 2 questions?
Yeah.
Yeah. Yeah, there is a question. Yeah, Mike, please.
Hello, sir. So it was a beautiful presentation. Sir, I just want to know about the regeneration protocol that you have used for your CRISPR system.
So we observed 20% of transformation efficiency. Editing efficiency is 5%.
Sir, the problem usually with groundnut is when we have transfer, after that the tissue culture, the regeneration is difficult. So, sir, I wanted to know how you have tackled it. Like, what was the protocol that you have used?
So
And
we
the
are
genotype
using
which you have
the
used.
off-cotyledon. That protocol is working good. Only problem with that protocol is like it is taking 6 months.
Okay, that is a very long time.
Yeah, long time. Otherwise, the protocol is working really good.
Okay, sir. Sir, and did you screen for different genotypes, like which was working well in?
Yes, we screened for more than 6 genotype. Now we are using ICGV 03043, which is a high oil content line.
Okay, and now the protocol is well established for
Yes, it's
the
well established.
generation. Okay, thank you, sir.
Thank
you. Any other questions? Yeah, there is another.
Yeah. Hello, this is Bhuvan from Ahmedabad University.
Hello, ma'am.
One question I have that in your gene editing efficiency, are you able to make sure that because being a tetraploid crop, whether you have all the 4 copies being edited because there would be some kind of SNP variants in them?
Yeah.
So how are you making sure whether only 1 copy or 2 copies or all the 4 copies have been edited? Because in Sanger sequencing, it's quite possible to have multiple peaks having homozygous, hemizygous, or wild type alleles present at the same time.
Yeah, it's a allotetraploid. So we can see the 4 copies. When we design the guide RNA is like whatever the 20 base pair, it is present in both the copy A and B genome.
True.
Yeah, so it
But
is
there will
present
be
in both
some
the
kind
copies.
of SNP variants around its flanking region.
That region
So
we avoided.
you avoided that
Yes,
region.
yeah.
Okay.
So yeah, we'll go for the next question.
Hello. Dr. Yogendra Garu, very
Hello, ma'am.
nice, excellent presentation. This is Padmasri from HCU.
Yes, ma'am.
Yeah, I very much appreciate your proteomics and metabolomics work, what you have done.
Thank you,
And you
ma'am.
have made a picture where you aligned the changes in both the metabolomics, proteomics to the metabolic pathway. So you have shown one picture, very nice picture. And may I know what tools you have used to correlate that?
So integration, it's a Sankey diagram, ma'am. It's available online also. And also SR plots, we can use SR plots for draw that diagram, ma'am.
Okay. Okay.
Yeah.
Thank you.
I think there is one question. Fine. Yeah. Yeah. Somebody is waiting online. Yeah. Dr. Asefa.
Okay, thank you.
Yeah.
Thank you,
Go
sir,
ahead.
for your
Ask
nice
your question.
work and informative presentation. I already put in chat. My work is related to this, and while reading different papers related to this, I came across with two contrasting reports with regard to fatty acid content and aflatoxin content in groundnut. If I can get any explanation from you.
Fatty acid content?
Yeah, fatty acid content plus the correlationship between fatty acid content and aflatoxin content. Some report said as oleic acid content in the groundnut, aflatoxin content is reduced. Some report said there are direct correlation and opposite correlation. So
yeah, actually we didn't test anything on the content. We focused more on like from the fatty acid for any cell wall thickening. Usually suberin is a polymer involved for the secondary cell wall thickening. For suberin is composed of aliphatic omega hydroxy acid. They are called as aliphatic compounds, and hydroxynamic acid amide, they are called as aromatic compounds. These two combined, they help for the secondary cell wall thickening. That knowledge we use for here, but we didn't correlate anything on the content of the fatty acid for aflatoxin thing.
okay, thank you.
Yeah, thank you. Thank you. Here is another question.
Hi, Yogendra. This is Anindya from CIMIT. It's
Yes,
a
sir.
very nice presentation,
Thank
a
you.
very detailed one. You have mentioned about syntaxin, annexin, and amylo type protein.
Yeah.
What about KTI? Do you like do anything with KTI, or you found it not usable?
We are working that one as well, but we didn't start anything on the editing part for the KTI gene.
Okay, and if you suggest a couple of good germplasm which are working well, like for groundnut transformation in your hand.
Whatever the lines they provided from breeding team, it's all working good, sir. So whenever we are selecting the genotype, we have a chat with our breeder. Dr. Janila is there as well. So based on her suggestion, we used to test this regeneration. But whatever the lines we tested so far, all are working good.
Okay, so I asked Dr. Jenilia and she told me you are the best person to ask.
Okay,
So I will
so
write
we
you
use
an email.
JL24 at the moment. We are using ICGV 03043. It's a high oil content. With that, we are editing for the aflatoxin thing.
Okay, thank you.
Sure,
Thank you. Thank you, Dr. Yogendra, for the nice presentation.
Thank you, sir.
Yeah, thank you. We'll go for the next presentation. And I invite Dr. Naik from US Dharwad.
Good morning. I'm Dr. Spurti Nayak, Assistant Professor at the Department of Biotechnology, US Dharwad. Today, I am presenting on understanding the heat tolerance mechanism in groundnut. We already know how climate change is affecting, and we are breaking the record every year. In 2023, we told that is the warmest year. And 2024, again, we are witnessing the same thing, like the rising temperature. It is like the extreme weathers events we saw in 2024. And the highest was heat wave. And 10 warmest years in the historical record have occurred in the past 10 years. And global temperature is rising too much. And because of that, even the due to this climate change, even the cropping pattern is changing. And the way forward is to breed for resistant variety which can cope up with this high temperature. If you see the abiotic stresses that groundnut faces, the major ones are drought and heat, and to some extent the cold stress, salinity, todicity stress, and heavy metal stress groundnut faces, and the yield penalty also is up to 30%, which is projected in the 21st century due to heat waves. And we need to be really working on heat stress in order to combat these things. And heat stress leads to oxidative stress. It also leads to seed inhibition in seed germination, reduction in plant growth, and also improper development and alteration in the photosynthesis. And which all together affects the dry matter and also yield. Not only that, even the quality is affected. Therefore, in order to check what is the optimum temperature in different crops, like what are the different optimum temperatures, you can see that in groundnut, the optimum temperature is around 20. 35, which is above which is considered as the damaging temperature. And in most of the other crops, if you see also, like the similar pattern is holds good, and we fairly witness now that there is a temperature is increasing more than 35 degrees, and this optimum temperature, like the base temperature in groundnut, for example, to germinate at least 10 degrees Celsius soil temperature is required for germination, and optimum soil temperature is 25 to 30 degrees Celsius. Optimum air temperature is again the same, 25 to 30 degrees. And for germination, we need 25 to 30 degrees Celsius, and root growth and development 30 degrees, and flowering and pod formation 25 to 30 degrees. Among all the developmental stages, the heat is affected, like heat is the major factors for the flowering, and because of when the flowering is affected gradually, because of that, you are also having the penalty on yield. And coming to our station, like Dharwad was considered as a cool place earlier, but now we are witnessing more than 40 degrees Celsius, like maximum temperature is going over the years. And it is like a time we have to work on high temperature stress tolerance in groundnut. There are many approaches, like integrated omics approaches we can use to study plant responses. And there are many approaches, and in other crops, there are proteomics, metabolomics, and ionomics, so many other things you can do. In peanut, right now, there are studies on linkage-based QTL analysis for high temperature stress. GWAS analysis is also done, and also transcriptomics approaches are being carried out. Coming to linkage-based QTL analysis, this work is done in the collaboration with ICRISAT as the main partner, and we have provided the phenotypic data, and genotypic data was generated at ICRISAT. For this, we got the recombinant inbred lines from Dr. Janila in 2017, and it was 55437 was the tolerant variety, and JL24 was the sensitive one. And we had 250 recombinant inbred lines. And JL24 is a bunch type variety, and it was sensitive to high temperature stress, and many of the diseases also it is sensitive. And 55437, it is also known as Samnut 14, is African cultivar, which was developed by IR Samaru, Jaria, Nigeria, and it is tolerant to drought, heat, and also aflatoxin contamination. I think the last presentation also they used the same 55437 as a resistant variety. And genotyping was done by sequencing GBS platform, and multi-environmental phenotyping was done. at 3 locations, US Harvard, ICRISAT, and RARS Tirupati. And 8-season phenotypic data was used to identify the QTLs. These are the traits which we got the measurements on, and morphological traits like plant height, flower initiation, days to 50% flowering, physiological traits like canopy temperature and specific leaf area, specific leaf weight, and SCMR readings, yield-related traits, and thermal indices like GDD, PTI, heat use efficiency, and other things. 78 SNP markers, and a map covered 1961 centimogon distance, and 45 major main QTLs were identified for 21 heat tolerance related traits. The PV range from like the major ones, it range up to 49% for shelling percentage. And 3 major clusters, hotspots were identified. They were observed on chromosome number 3, 12, and 20, harboring more than half of the QTLs that were observed. Like out of 45, 30 QTLs were on these 3 chromosomes. This was the cluster. They had also identified candidate genes that are on the physical map, and the major ones are like DHHC type, zinc fingerprint family proteins, peptide transporters, and pentatrichopeptide repeat containing proteins, and etc. This was published in Frontiers in Plant Science in 2023. Coming to another study from Rastagi group, I think yesterday also he presented this one. Just in order to make sure that I am covering all the heat tolerance related studies, I kept this one slide where what they did is 72 peanut genotypes from US mini core collection was used as the association panel, and they subjected the plants in the controlled growing conditions like heat stress simulation was given. It up to 38 degrees Celsius, and nighttime temperature was at 28 degrees Celsius. And 5,000 odd SNPs were selected for association analysis, and they did study PPV, that is percent pollen viability under heat stress conditions. And they identified some markers on chromosome 20. And the haplotype analysis also revealed that 6 kb region at this chromosome 20 that contained 3 candidate genes, and they were like ribosomal protein, copper transporting ATPases, and they showed high expression in reproductive organs. And this study also told that it is co-localized with the hotspot regions on chromosome 20 that was already reported in Sharma et al. Paper, that was a QTL analysis paper. And this is recently published two months back in Plant Stress. Coming to what for heat tolerance, we did do a bulk segregant RNA sequencing analysis. For that, we selected the genotypes based on the yield parameter over 3 years on the post-rainy season. And whichever the genotypes having high yield, they were selected at the high temperature, like tolerant bulk, and whichever were having lower yield, they were selected as a sensitive bulk. And along with the 2 parents, we analyzed several physiological traits on this panel. These are the parameters we recorded, like chlorophyll related traits like chlorophyll stability index and chlorophyll content, AAB content, and carotenoid content, and SCMR values. They were recorded on 20 days after heat stress initiation. And the membrane related traits like membrane injury index and melanaldehyde content, and specific leaf area and leaf thickness were also identified. And also biochemical assays like catalases and peroxidases were assessed. And also RNA like RT-PCR we did on some HSPs. This was the experimental setup. like where we sown the seeds and we maintained a control condition with optimum temperatures in one of the grass houses, and the other set of genotypes were maintained at growth chamber with simulated heat, like we incremental increase in the temperature we simulated, like how it happens in actual scenario, like 28 degrees up till 45 degrees. It would go until like 12 o'clock, and from there it will reduce again to 28 degrees, and night temperature was maintained at 25 degrees. And then that was done only during the flowering stage. And then the pots were shifted to greenhouse, and they were maintained around like 38 to 42 degrees Celsius it was showing. And the other control, which was maintained at 28 degrees only. And then we harvested. For this, like chlorophyll stability index, we did, and we found that in the HT bulk, we had, like, say for example, in 55437 was showing highest chlorophyll stability index as compared to even the bulk genotypes. But HT bulk had relatively higher chlorophyll stability index in the control condition, because when it is in control, the stability is more. When it is under stress, the chlorophyll content will be reduced due to heat stress. And similarly, SPAD meter reading, that SCMR also, like in the SPAD meter reading also, like high tolerant temperature bulks were having higher and comparatively to the stress control were higher, and stress condition they were lower, but the differences among the HT and HC was very negligible. But unlike this, in the canopy temperature, we had like lower temperature observed for 55437 and also to high tolerant bulks as compared to sensitive ones. The other one is like membrane injury index. We estimated the membrane injury index using EC meter, and then we saw like membrane injury was the main character in all these physiological traits, that the sensitive ones were having higher membrane injury as compared to tolerant genotypes. And the leaf area meters studies were done, like we estimated the leaf area meter area and also calculated the specific leaf area. Here, the HT like 55437 was showing highest leaf area as compared to other tolerant varieties as well as the sensitive varieties. We do calculated the catalases and peroxidases the activity, and high temperature under stress condition, the tolerant genotypes were having the higher catalase activities as compared to the sensitive ones, and peroxidase, in terms of peroxidase, there was no significant differences. And coming to MDA content, which is also a major component assay, where we can see that the sensitive genotypes had highest melanoidin content as compared to like tolerant ones, because here after lipid peroxidation, this is the product after lipid peroxidation. In case of stress condition, the genotypes which can which produces more melanoidin, that indicates that their membrane is damaged. We also studied some of the reported heat shock protein gene expression in this same panel, and it was mainly from Chakrabarty et al. 2018 and Kokanti et al. 2019, and we used ADH as the housekeeping gene. And the RT-PCR results, they showed it was a very overwhelming result, like HSP17, which is a small heat shock protein, showed very high fold increase in the expression in both heat tolerant bulk and as well as compared to like other parents. And the fold change was almost like 1900 fold change. And it was also true with HSP70, but not up to this scale. This we have already, yeah. Once this is done, the BSR, for BSR sequencing, what we did is we isolated the RNA from pot tissue, like flower tissue as well as pot tissue. And then we pulled in equimolar concentration, then subjected for RNA sequencing. And the RNA sequencing results showed that in the bulk flower tissue, when we compare, like total DEGs found was 155 DEGs, and out of that, like 84 were upregulated and 71 were downregulated. When you see this one, and when we go for putative candidate analysis in that region, we got phosphatidyltransferase transferase, which is the major gene that was upregulated in the flower tissue, and cell wall protein is downregulated. And they were mostly in membrane-bound proteins. And when we see the chromosomal distribution, we also find major number of DEGs were found in chromosome 3, which is also hotspot observed in Sharma et al. 2023. And just to check whether it has some effect on fatty acid, we also profiled fatty acid in the panel, and we did not see much differences in the fatty acid profile, although there was a minimal content, like MUFA was more as compared to PUFA. These are the conclusions of my study. I finally thank all the collaborators and the US, the Harvard students, and my team for backing me for carrying out this work. Thank you.
Thank you for the nice question. Yeah, please go ahead.
Thank you very much for such a excellent presentation. I am Dr. O.P. Shengi, Senior Vice President from DCM Sriram. I have very limited or practically no understanding of genomics and biotechnology, but I understand the practical aspect of all efforts being done by the global scientists for different crops, and that's why I here. In one of your initial slides, you gave the optimal temperatures for different stages. That is air temperature, soil temperature, germination, etc. If you see the groundnut cultivation in Rajasthan, for example, there these temperatures are irrelevant. I believe that your slide is speaking more about the Dharwad temperature.
No, it is general. It is
and
not.
If it is in general, then the temperature are much more higher, air temperature, soil temperature, when the ground is cultivated during the April, May.
Yes,
and
sir.
that is all over Rajasthan.
Yes.
So coming back from that background, if you see the adaptability of current varieties cultivated at the larger geographies, these are G10, G20. And I believe that perhaps choosing one of those parents in your studies might have given you better results looking to the Indian conditions.
Okay, sir.
You selected one of the line from other country, which is tolerant to heat and drought. But I think we have much better germplasm available, which have higher tolerance in India itself.
Yeah. This was to just decipher the basic information regarding heat tolerance, but the information whatever we get can be validated on these genotypes which are already available, like whatever you told, those genotypes as well, and we can identify like if that is relevant. Coming to the soil temperature, whatever that is a general thing, like for germination to happen, like 25 to 30 degrees is the optimal one. And whenever you irrigate, I think soil temperature will reduce. Otherwise, I think beyond 30 degrees or 35 degrees, 35 degrees is a damaging temperature. And we also saw, like I have not presented here, like temperature induced induction studies we have done in seeds also. And whenever we are going up to 42 degrees, until 42 degrees, some genotypes, they do germinate. After that, they don't. Therefore, I think
we can discuss later.
sure.
That's a good suggestion.
Sure, sir.
Charles?
Okay, sir. Thank you.
Yeah,
Hello. Yeah. My question is, maybe this question not for you, also for the whole community here. So how are we going to define the heat tolerance? As I'm working on a lot of the time on the drought tolerance, we still a lot of debating. So like I say, you're doing the QTL, you are doing so-called heat associated trait.
Yes, yes.
But those traits may not related with really with breeder one. So that's my question. How are we going to define that?
Can I start as like a discussion? If like, for example, whenever we are working on drought and heat, like in the heat conditions, we are ensuring that the irrigation is given. Only different, like only condition what is differing is only temperature. But the irrigated condition we are maintaining, that will ensure the, but I personally feel that there is many mechanisms that are overlapping with the drought and heat. Yeah, I think.
You can discuss, it requires a lot of discussion. You can discuss, yeah? Maybe during lunchtime.
Okay, sure. Thank you. Yeah.
Other question? Yeah.
Do you think temperature induced heat tolerance, especially heat treatment to seed before planting, will help somehow in heat tolerance? Like we have seen in seedling, like heat induced induction helps in tolerance. So will it happen in case of seed?
I'm not sure, but they're like a treatment kind of thing, right? I think we should check it out. I'm not sure about it. Thank you.
Yeah, Doctor.
It's a comment to the question on the seedling induced one. In groundnut, it doesn't hold for adult plant resistance. Unlike in chickpea, it does hold very good. And then hence it can be deployed in the screening protocol as well. But that's not true. A study conducted by one of the research scholar has shown it. And just to touch upon Charles' thing, like what we look for adaptation for heat is, like again, it can happen at different phases. At flowering or pegging happens, the number of pods formed, it gets drastically affected. But then most of the heat we are seeing is during the pod filling stage. And what we have observed is most of the genotypes, they grow very luxuriantly. So photosynthesis is not hampered. So what is hampered is translocation of the photosynthetes from the source to sink. You have pods, but they are pops. So I think, and in one of the study which another student has conducted, showing some of the pathways related to sucrose synthase and translocation being affected in the susceptible genotypes compared to the tolerant ones. Thank you. You have.
Yeah. I have a question, ma'am.
Recently, it is popular to use ROS scavengers, oxidative burst scavengers, because most of the heat damage happened because of the ROS generation. So, can we see use of some scavengers like thiourea or something, and see if it can elevate JL24 from the stress?
Actually, there are some studies like where tree halos application they have done for a drought purpose. I think the same way they can do it here also.
Yeah, I also have another comment here. Right now, we are more focused on the under the heat tolerance, how the photosynthesis is going on. But actually, we really need to looking for in the nighttime temperature go up and how the respiration going on. Because the respiration will consume a lot of the energy, will reduce the yield. That's just my comment.
That is one way. I think we are not focusing on that aspect. I think we have to work on that. Yeah. Thank you. Thank you.
Thank you. Thank
Thank
you.
you.
Dr. Murthy.
Now we go to the second last presentation from Dr. Anindia from CIMIT, a talk on gene entity for trade development in Global South. Welcome.
Okay, so thank you, organizers, for inviting me. Special thank to Manish and Rajiv for accepting me here to give this talk. So please bear with me. In initial few slides, I will talk about genome editing, what we are doing in CIMIT, a brief overview, and then I will get into the groundnut project which we have. So this is from one of our collaborator, Potiva, where we see that their first product for breeding, their product for breeding usually take 9 years, whereas transgenic breeding, they have shown us that it takes 13 to 15 years because of regulatory issues and a long drawn regulatory process. Whereas their Waxicorn, that was edited and brought into the market within 4 years. So this is where the interest lies, I think. First of all, CRISPR-Cas9 is a very cheap process. And there is a caveat if you have license. And then it can save a lot of time, and time means money. So in various ways, this can play a significant role in crop development, crop trade development. And what are the traits can be easily targeted by genome editing? I would say the traits which are well characterized and qualitative traits. Still, we are struggling here. Poorly characterized diseases and quantitative traits, drought, lodging, to some extent yield, where multiple genes are involved. First of all, we need to know what are the specific genes and pathways involved. And second, we should have capacity in doing multiplexing. So we can do multiplex 4, 8, 12, 14, like that. But some of the drought pathways, there might be 50 genes or 60 genes. So we need to really improve ourselves in multiplexing. These are the products I have just shortlisted here. Non-browning mushroom, coffee beer, spicy tomato, waxy corn, gluten-free wheat, decaffeinated coffee, etc. Approved in various countries, genome-edited crops. And
this is the scenario presently related to genome editing. So those who are from other section, like breeders and other physiologists, etc., who may not know this, or the students. So these are the green countries where genome editing is accepted as non-transgenic, non-regulated. So you can do any research, you can do any field trial, you can develop product, and that product is considered as non-transgenic, traditionally bred product. Only two places there is red. I can tell you this: New Zealand. They are still regulating it, and they go with UN. They go with EU, European Union, and European Union gives. They were against it initially, but now things are changing. So they came under yellow, not red anymore. Many countries, especially Scandinavian countries, they want it very strongly, and they are proposing it, and new legislation came up, and it requires another validation. So I'm not going in detail of different product already in market by different private sector or countries. You can find it in genetic literacy project, and there are trackers. And in CIMIT, what we do, this is our facility, biotech, and I lead this one. And we are majorly doing gene editing. We have a couple of GM projects, but majorly doing gene editing. And these are the crop, maize and wheat. It is our bread and butter. And last few years, we are also doing few selective dryland crops. And where do we see genome editing? So in our breeding pipeline, where we can see that GM development would be something not working. Anyway, so GM crop development. Have one more? So GM crop development and molecular breeding and trait design in between genome editing basically can be placed for rapid functional validation of allele, create allele diversity, optimize allele in product line, and then one-step integration. So in broader design of the breeding, genome editing can play a significant central role, not only by giving traits, good traits, but also improving different aspects of breeding, improving in the sense, most importantly, increasing the speed, like technologies like high edit, etc. And this is what we maintain in CIMIT. So we have a market intelligence team, breeding design team, trait team, G trait targets, and then we design CIMIT G tech thing, and then we have license from Corteva. So we can go end to end with the product development. And now CIMIT management has mentioned if any technology we use, which we don't have freedom to operate and develop product and give to the farmers, don't do that. No need of basic science. So collaborate with universities and institute to do basic science. But if you cannot generate product, you don't do. So we very strongly believe in this, and we are doing licensing as many as possible, because CRISPR toolbox is varied and different kind of tools are available. We are also working with Broad Institute to get Cas12a. We are very close, so hopefully we'll get there. So these are the tools, Cas9, Cas-Clover, and Piggyback. These are the tools we have, and we have license end-to-end. And then we are working with Pairwise for Shark and Redraw. And also we are working with Broad Institute for Cas12a. So rest of the stuff we have end-to-end license. So now maize, this is our one success I thought I will briefly mention here, just one slide. That maize lethal necrosis is a devastating disease in Africa. And we have solved it with all these collaborators. Field trial, we have with Corteva partner here. And we could identify the QTL, we could identify the fine map the gene, we knock out the gene, and we could generate lines which are MLN resistance, and it went to the field. We are now developing hybrid, and we expect to give it to the farmers in Kenya by end of 2026. So this is our most important. And then wheat, we have established a high throughput agrobacterium mediated transformation. Josh is working here. He's a postdoc. Previously, I didn't mention Easter has joined from Bing Yang's lab in University of Missouri. She is working on maize. And then pearl millet, recently we got some good result. We have cracked pearl millet transformation. We have established high throughput pearl millet gene editing. We have knocked out 2 genes and we have submitted for publication. And we have one collaborator here, Salim from King Abdullah University House in Saudi. So he is helping us funding from Bill and Melinda Gates Foundation. Now, Sindhu came from ICRISAT. She is working with us in the PGNP project, VAX project. And Yiping Chi from University of Maryland collaborating with us along with Danny. So this is the only one GM project we have, which we are basically doing BT, but we have plan of some editing in PGNP as well. So now coming to the topic, aflatox gene editing. Now, I have inherited this project from my predecessor, Pooja, who was in ICRISAT and majorly done the gene discovery part in ICRISAT. And then she joined CIMIT and now she left. And in her place, I joined actually. So I have inherited a couple of projects from her. This was funded by Gates Foundation. Mohan is working. Mohan is a senior IRS in our lab, and he's doing extremely good. So Yogendra and others have done a lot of background discussions, so I'm not going in detail how aflatoxin important, and people here, they all know it. And mostly with Pooja's work in ICRISAT, they found 3 genes by multiple work. Some of this work Yogendra has mentioned as well. So I was asking Yogendra about this, the syntaxin and annexin. These are the 2 target genes we will knock out. But there is KTI as well, and Yogendra added one more. So there are these 3 genes we are targeting in our project. But and also we are with one of our collaborators in University of Texas A&M. We are working on the phenylpropanoid pathway as well. We have done thorough RNA-seq, and now we are doing RNA-seq in our lines. And we have identified 5 target genes which we have started knocking out because seed coat has certain kind of chemicals which gives resistance. We want to overexpress those in the seed coat by GM method as well as by knocking out competing pathways in seed coat so that those particular resistant chemicals are highly expressed. So this is the second. So first was 3 gene knockout, and second is seed coat mediated resistance. And so these are the 2 I have mentioned. So syntaxin, annexin, KTI, the first pathway on the top, and then through Texas A&M University, the disease phenylpropanoid, targeting phenylpropanoid pathway we have identified. Now we're a little behind. We have started, I joined 1.5 years back in CIMIT, and we have started working on it, but we have taken quite difficult recalcitrant lines for Africa, which are SEMNUT 24, 23, 27, 28. So these are really recalcitrant, and we are mandated to do on this only. So we cannot use the easier lines. So we are trying it, and we have identified the genes, amplified all 3, designed our gRNA, and we have established a transformation system for it, which is a bit slow, but we have used mature cotyledons, shoot apex, leaf petiole, cotyledon node, and immature embryo. All this we have tried, and 25 different media combinations we have tried. And whereas in mature cotyledon, zero-day explants gave us regeneration about 32%. But there are certain problems we have seen in shoot bud development, and regeneration is very slow, time-taking process. It takes quite a long time. And for leaf petiole, Corteva is doing this one. Corteva is partner here with us. They have used leaf petiole previously. They could get very high regeneration and quick regeneration in leaf petiole. But transformation is ongoing now, so I'll be able to give our data very soon. In CIMMYT, this embryonic shoot gave us the best result, and we used SEMNAT 25 and SEMNAT 28 worked well for embryonic shoot. And different shoot apex main branch we have used as the major explant. And we could get about 69% to 90% regeneration efficiency. So this gave us best result. Although regeneration time is still a question, little better, but with respect to rest of the explants. And positive lines we have generated. These are transgenic with test construct. Now we have started editing this one. And in plant agroinfiltration we have also done. This is also quite fast. And we are working on it for editing. So, and the regeneration take 1 month, which is really interesting. This is just simple tissue culture. This is not transformation. So we are planning to do high throughput transformation. Root media, rooting is, again, we found a little bit of a challenge and we did many things. The green ones, MS28 and M35, these are the 2 media we have established for rooting for all the SEMNAT. So these are the ones we have done so far. So in conclusion, I'm not going in detail. We have tried all of the explants so far reported. Out of that, 1 explant worked well, as I mentioned. In-planter method is the fastest for these SEMNAT varieties. I know there are many easy varieties available, JL24, etc. people use, but we cannot use those. So, and also cotyledon method showed delayed elongation. And we are continuing our work with SEMNAT because we are supposed to develop these and give it to the farmers of Africa, mostly Western Africa. So these are the popular ones. I think we are continuing. So this is our team. Apart from the postdocs I showed you before, Esther, Mohan, and Josh. Josh came from John Inez, Mohan came from ICGB, Esther came from University of Missouri, and from ICRISAT Sinduri Joint. We have 4 senior people, and these are our LRS staff for gene editing lab. And last many years, we are getting ETS certification. I thought I should mention it because none of the private company are interested to work with you if you don't have excellence to stewardship. Especially for research, it's okay. But for product development, there is no way you can release your product if you are not a ETS certified member. So this is apart from the bottleneck of having license of CRISPR-Cas9. Last slide. I'm organizing 1 genome editing workshop in Orlando next week. And many renowned scientists, 2 workshops actually in AGBT-AG. Many renowned scientists in genome editing arena are joining. So please, if you can manage, please join, or if you have already registered in AGBT, then please come to these 2 workshops. And these are our collaborators and funders. And that's it. Thank you. And before finishing, I have 1 word for our colleagues and friends here, that there are certain crops where we can jointly work. Not only ICRISAT, everybody, I'm telling it to everybody and definitely to ICRISAT. We can generate grants together. We can develop some new areas in those crops. So at least from the scientist side, I believe there will be no disagreement on this. So rest of the part we have to see, and I strongly vouch for this, because instead of doing same thing and reinventing the wheels, we can work together in many crops, and we can generate grant as per our competence, right? So thank you very much.
Thank you, Dr. Anindia, for those insights in entity transformation. So honestly, frontiers in science to move forward. And again, your call for joint efforts. I think we all fall in the same boat, and we need to join hands together. So let's have some quick questions. I can see you have quite so far three. We start with Injanila.
Thank you, Anindya. A very basic question. Aflatoxin contamination is a problem both happening on the field, on a live plant, and also more of a problem in the storage. So when we have these events, what do we expect?
So that's why we have added the second part. There are two schools of thoughts that aflatoxin is mostly effective post-harvest, and when there is no green plant alive anymore, right? You harvested your crop, and in storage, aflatoxin is very difficult to handle. So two school of thoughts. First thought, they think that initial infection happens when green plant is there. Definitely it happens. And the genes, resistance genes in green plants, in many ways will take care of that. If we can properly identify the genes, either make GM or do editing in a non-GM route. But there is a second school of thought that green plants are not there in actual aflatox devastation. So the seed coat gives some natural resistance, and there are certain resistant chemicals in the seed coat, which are part of the phenylpropanoid pathway. So overexpressing those in the seed coat would be the way to go. Now, we don't know which one is right. So we are doing both. So our target is the genes in the green plant. We are also trying to identify some new genes. I didn't go to that details. So once we make those lines, then how to channelize seed coat mediated chemicals in the green plants so that both can be expressed in one background, high expression. So GMOA, it's easier, but as you know, there are complexities of regulatory and etc. So just knocking down the competing pathways to improve certain kind of chemicals in the system, knocking down competing pathways by editing could be a way to go. So we are combining both. In our mind, we are combining both.
Yeah, I want to add one point. You said there is one chemical that is involved in phenylpropanoid pathway that is present in the seed coat, that is resveratrol. So yeah, ma'am, we by overexpressing that gene with the genome editing tools, we can avoid aflatoxin. So the chemical, phytochemical sir has mentioned is resveratrol. So we can overexpress this gene.
Yes. So we don't know. We don't know 100%. There are many genes in the phenylpropanoid pathway.
Still been synthase.
Yes. So we'll see very soon. Yeah.
Thank you.
And I see Yogendra also gave a very nice presentation on that area, so I didn't go in the detail of that.
Yeah. So let's hear from Prof. And now we go back to him.
Yeah.
And
Yeah, I have
we
question still on the aflatoxin. I know in the aflatoxin not only happen in the peanut, also on the corn, you are from the CIMMYT. So I assume you have a lot of work on the corn. Can you have some detail, have some insight about what's the progress?
No, it's a good idea. It's a good idea. Breeding team, nobody is there from corn. He's from dryland crop, but I don't know. Probably breeding team will be able to answer it. I am not the right person, but it doesn't come as one of the priority traits for corn. So genome editing, we haven't thought yet, but it's a good idea to check actually.
Thanks for the nice talk. I'm Samir. What do you think is the biggest bottleneck right now in terms of gene editing? I work mostly on the trait discovery side, where mostly we work on quantitative traits, and oftentimes we just give bunch of QTLs and just leave it there, do not follow until the single gene resolution step, is that the biggest challenge, that we don't have complete haplotype knowledge or knowledge on the gene? Or the even more challenge is to actually edit the gene once you know what the gene is and produce it at the mass level. So what do you think is the biggest bottleneck in gene editing?
Yeah, so you are asking in the biological part. I will tell that, but before that, I will like to mention one thing very strongly. If you don't have gene editing IP is the biggest bottleneck.
Yeah, I get that.
Yeah, so that's the biggest bottleneck. If you don't have gene editing IP to bring it up to the field end to end, there is no use of doing gene editing. It's just basic science. Universities are doing it, organizations like us, we should not do gene editing if we don't have license. But anyway, that is the biggest bottleneck globally. So Jennifer Doudna's team, they are developing a new molecule which they want to give for free to the public use. Cas9, Cas12A, both are highly IP protected in US. So you cannot bypass that. So that's one of the different kind of bottleneck. The thing you have mentioned is true. I mentioned already that multiplexing, we have certain limitations. 14 gene, 20 gene multiplexing we can do. But beyond that, if a complex trait is made up of many genes, then at a time, bringing those by edit those is still not possible in existing scenario. Many new papers are coming up, different kind of things are coming up. Even people are talking about linkage drag break, and it's happening in Arabidopsis in a great way. HEI10 and other kind of proteins people are using. And this linkage drag break thing can be translated in higher crops, it will be a path-breaking thing, I believe. So many new things are coming up, but it is true for any research, right? So even if identifying a new QTL, it takes time. It was from infancy to adulthood, it takes time. Similarly, knowing solid biological data is very important, biology of the crop, and then the trait, and then identifying exact genes which are involved in that trait development. Then only you can. So
India, I think is well answered. Let's have
we can
final
discuss
question
more,
for
yeah.
Kuldeep, then we get to the final presentation from Dr. Padmasari. So Kuldeep.
Yeah, thank you. Thank you, Ananta, for a very honest presentation. My concern will be what you said is the most important thing, that IP. Now, CIMMYT has, as you told, CIMMYT has an already understanding with the Corteva end-to-end. What does that mean? If you have a product, either in any of these 5-6 crops, you can really, CIMMYT can go to the national partners and ask it for commercialization. National partners can commercialize based on the IP, the license the CIMMYT has, or it has to be something different that national partners really have to look again from the IP holders on that?
No, you know that I think quite well that it's not easy dealing licenses with private companies, right? And we also had a fair share of that. And each and every case, you need their permission. So whatever you want to do, and there is something called humanitarian license, which we are doing with the Cortiva, and they are okay to give it to us. There are certain caveats. Those caveats are your nurse partner or whoever is your partner in the country, they should have a very strong stewardship and they should pass ETS. Then they are comfortable giving their technology to be given to anybody. They are also concerned about one more thing. Certain countries' borders are porous. Like suppose if you give this seed to Kenya and then seed goes to Rwanda or Burkina Faso by some reason, it happens in developing world and in underdeveloped world. So if a farmer sees a good crop, that side of the border, they can request the farmer from the other side can give that seed to the friend to like that. So company thinks they might get sued because of the porous border. This seed may end up in a country where it is not allowed. Then they will be sued. So they want indemnification. Now, as an institute, we cannot give indemnification. That whatever happens, responsibility ours. We cannot give that, right? So there are ways we are trying to come up. Now there are a couple of like AATF and other organizations in Africa. They can give indemnification. They have done it for GM and they are experienced in doing that. For our MLN, same thing will happen. SPTA, same thing has happened. So these kind of challenges you will come up when you take the license from industry, big industry. But that's why we are trying to take licenses from Broad Institute, which is an institute, things are most liberal, and also some of the Chinese universities who are claiming
India, let's check time.
new
IPs.
IPs.
Time, please. Yeah.
Thank you.
Yeah, thank you. So let's have the final presenter for this session, Dr. Panmasari. So you are standing in between us and the lunch, so please keep it short. Thank you.
To all of you, I'm Padmasri from University of Hyderabad. And first, I want to thank the organizers, Dr. Manish Pandey. It was very kind of him to invite me. We discussed this work long back, and he remembered me, and he called me to this conference. And I also thank Raj and other members, and congratulate you for organizing a wonderful conference. Then I know all of you are so hungry. I will try to complete quickly. Okay? I think most of you must be looking at if I say peanut proteins, you must be immediately thinking about the allergens, anti-nutritional compounds. But in today's presentation, I would like to show some important and nutritional aspects also, positive aspects of the peanut proteins. So today I am going to speak about the peanut-derived Bowman-Burke inhibitors as potential anticoagulant molecules. Thank you. So, I would like to explain about some plant protease inhibitors and their role as anticoagulant. So, that is the rationale of the study. So, most of you must be knowing about the blood coagulation pathway. Somewhere or the other, you must have read about it. Even I am in the learning process. So, this blood coagulation pathway has several factors which function in a cascade manner. So, one of the protein will be inactive, then it gets activated by another coagulation factor. So, whatever factors we are seeing here, these are all serine proteases, and the Bowman-Burke inhibitors where we are working, they are all serine protease inhibitors. So this is one of the criteria we have chosen to work on this. And any imbalance in this blood coagulation process, it leads to a serious diseases like deep vein thrombosis. It means that when there is a problem in the blood coagulation pathway, clots will be formed. These clots will be hanging in the blood vessels. And part of these clots might get dissociated and move through the blood vessels. And they may get clogged at some other tissues. For example, if it reaches the heart, it causes heart attack. And if it reaches the brain, it causes stroke. So these are some of the serious problems associated with the blood coagulation pathway. And now what we would like to see is how these protease inhibitors can be used as the anticoagulant molecules. These protease inhibitors are seed proteins we have taken, and they are of very low molecular mass, around 8 kilodaltons. Then they regulate, in general, they regulate proteases within the system, and they regulate the physiological process. And these protease inhibitors are exploited for various therapeutic applications. Like initially, they have worked for insecticidal properties. Later, they have identified several therapeutic properties also. People use them for antifungal activity, then antibacterial activity. They are also known to induce hepatosis, anti-carcinogenic effect, antiviral, anti-inflammatory, anticoagulant, so on. So we are focusing on this anticoagulant effect. So in this coagulation pathway, so far a few inhibitors like heparin, warfarin are used. So it is good to exploit the natural product. So in this anticoagulation process, proteolysis plays a key role in the blood coagulation pathway, and the inhibition of these proteases by protease inhibitors like Bowman-Burke inhibitors can delay the blood clotting process. So here we made a study like we have purified the native protease inhibitor, that is Bowman-Burke inhibitor, and we also cloned, expressed, and purified it. And we compared whether the expressed protein possessed similar properties like the native protein. So we have frame the following objectives like purification of protease inhibitors, that is native protein, and recombinant protein. So from here onwards, you please remember native protein is called as PNPBI because this is a Bowman-Burke inhibitor, and recombinant as the RPNPBI. So this is the related to the first objective. So we have purified the native protein as well as the recombinant protein, applying the chromatography techniques like trypsin affinity column and gel filtration column using the FPLC. Then we tested whether the purified proteins are inhibitors or not by assessing their inhibitory activity against trypsin and chymotrypsin. Why I am saying trypsin and chymotrypsin is these Bowman-Burke inhibitors are double-headed proteins. They have two sides. One active site is effective against trypsin and the other active site is effective against the chymotrypsin. Then we did effect of these peanut Bowman-Burke inhibitors, both recombinant and native, on blood coagulation pathway. A preliminary test is performed, that is PT, APTT pathway. PT is related to the intrinsic pathway and APTT is related to the extrinsic pathway. After confirming this, then we move to check the inhibitory effect of these Bowman-Burke inhibitors on other clotting factors, which I showed in the previous slide. At least 4 factors we have checked, including the plasma kallikrein. Then we check the interaction between these Bowman-Burke inhibitors as well as the coagulation factors using the surface plasmon resonance technique using Biacore instrument. So this is the protocol applied to purify the native protein. We have prepared the peanut crude protein extract, then subjected to hexane and acetone washes. Then we subjected it to ammonium sulfate fractionation and pass through affinity column, size exclusion column. The highlights, these are the active fractions, and this is the representation of the pure protein that we got. And here are the gels. This is the trypsin SDS page representing the different fractions. And you can see with increasing concentration, this Bowman-Burke inhibitor shows oligomeric pattern, which is the characteristic feature of the Bowman-Burke inhibitors. And when you subject this to DTT, and all these higher order structures are lost, and you will see the monomeric form. And here we use the soybean as the standard thing. And we assessed in-gel activity to show that they possess both trypsin and chymotrypsin inhibitory activity. Then we performed the native page, and in native page we have seen 5 different bands, means more protease inhibitors, Bowman-Burke inhibitors are present, and all of them possess trypsin and chymotrypsin inhibitory activity. And we also check more isoforms are present in 2D electrophoresis. And this is the spots we have showing different isoforms. And all of these isoforms are showing, most of them are showing both trypsin and chymotrypsin inhibitory activities. So these 2 sites can bind to both trypsin, or one side to trypsin and another side to chymotrypsin. So here this figure shows the expression and purification of the recombinant PNBBI, and here we can see the pure protein, recombinant protein, and this shows the trypsin and chymotrypsin inhibitory activity of the in-gel. And the recombinant protein also possesses molecular mass almost equal to the native protein, around 7.5 kilodalton. And we did phylogenetic analysis of the recombinant protein, and it shows closer to Medicago sativa, Medicago scutellata, and we modeled using the sequence of this Medicago scutellata, and this is the modeled form of the recombinant protein. And we can see here the characteristic feature of the Bowman-Burke inhibitor having 7 disulfide bonds. That is the reason for its good activity, where it is stable against various temperatures and various pH. Here you can see the stability of the both native protein as well as the recombinant protein. We assessed first PNBBI with comparison to soybean BBI, and we found it has significantly higher activity against trypsin and chymotrypsin when compared to the soybean. And when we compared PNBBI and recombinant PNBBI, native protein showed more activity. Perhaps there could be loss or more recovery of protein need to be required here. Then we have done interaction of this Bowman-Birk inhibitor with trypsin, chymotrypsin of both forms, and we could see the dissociation kinetics with this technique. Then we incubated these proteins at different temperatures, range of temperatures and range of pH. And if you see at 90 degrees also, there is not much loss in the activity. That is TIR, chymotrypsin, trypsin inhibitory activity or chymotrypsin inhibitory activity. It means they are very stable to temperature and pH. And if you look at the structures also, there is not much variation in their structures. And it is not shown here, but when we did the recovery also, we could regain from 90 to 25 if we go. So here we see the effect of this native protein and the recombinant protein on the APTT and PT pathway. These are extrinsic and intrinsic pathway activities. And here you can see we used different concentrations of the PNBBI and recombinant PNBBI, and we compared with the heparin. So in this test, APTT, we have seen that blood coagulation is prolonged. If it is in the control sample without inhibitor, 30 seconds if we take, there is a twofold increase. We could see prolongation in the blood clotting when we use these inhibitors. But the factors which play a role in PT pathway, they are not getting affected here. So the APTT SA, which is related to the intrinsic pathway, we assume that the factors that are present in this pathway got affected. So we went and checked for the individual activities. Next. So here we have seen the effect of these proteins on the plasma kallikrein, which plays a role in activating several of the downstream factors in the blood coagulation pathway. For example, factor 12, 11, 9, and 10. So we can see almost when we do a series of concentrations of these protease inhibitors, you can see here this line at 1.48 micromolar concentration, there is 50% inhibition in the plasma kallikrein activity. Then if you take the recombinant PNBBI at 5.31 micromolar, we observed 50% inhibition. Means when we compare native versus recombinant, native form is more active than the recombinant here against the plasma kallikrein. One possible reason here is this protein, as I have shown, it must be having several isoforms. All are important in inhibiting the kallikrein activity. And when we show the recombinant one, only one protein is present here. Still, it is capable of inhibiting the plasma kallikrein at little higher concentration. So here we have verified the effect of these native and recombinant proteins on the activity of factor 12A, 11A, 9A, and 10A. And we have performed the dissociation kinetics as well. Then here you see the summary of the previous slide, what is shown. And here you can see the red one. If you look at, compare the results of half maximal inhibitory concentration, which is required for inhibiting the plasma kallikrein. In case of PNBBA, it is 1.48 micromolar. And in case of recombinant, it is 5.31. It means PNBBA is more effective, as I told just now. And if you see the dissociation kinetics, it is little less when compared to the recombinant protein, but still it has very high low IC50 value. Then this recombinant protein is much effective against the factor 10A when compared with the PNBBI. And here you can see the equilibrium dissociation constant. which is very good, which is in the nanomolar range. So the overall summary of the study is the purified recombinant PNBBA exhibited biochemical, biophysical, and anticoagulant properties similar to the purified native BBI. Then the recombinant BBA has shown differential inhibitory activity against trypsin and chymotrypsin. It is very, very effective for trypsin. It is only 0.23 micromolar concentration, while with chymotrypsin, it requires 5.31 micromolar concentration. Then both PNBBA and recombinant BBA are effective against 12A, 11A, and 9A, both of them. But the order is 12A is more effective than 11A than 9A. And this recombinant PNBBA acts as a potent inhibitor, as I told you, and heparin and warfarin are active against, in the commercially they are being used to inhibit the factor 10A. So the overall conclusion is the development of BBA as an anticoagulant, which is abundantly present in peanuts, a natural food source, may play a significant role in antithrombotic activity. So here you can see the summary of the protease inhibition by native PNBBA and recombinant PNBBA. So both of them have two inhibitory sites. They can inhibit two proteases at a time. And this is the intrinsic pathway, and this is the extrinsic pathway. So here we tested against plasma kallikrein 12, 11, 9, 10. And all of these PNBBA and recombinant PNBBA inhibited both of them, all of these, but their effective concentrations might be different. So this factor 10A is inhibited by so far heparin and dabigatran, and several other synthetic molecules are being used to inhibit this factor 9A. So this study, we think that it might really help to develop a product, but more clinical trials are required through this for human utilization. Thank you, all of you.
Thank you, ma'am, for the wonderful presentation. We are going to entertain only one question. And after that question, I hand over to the chair.
I would like to acknowledge Dr. Mallikarjuna for this, and was the person and Karda Tetali, he also helped me cloning the recombinant protein as well as coagulation studies, and these three students are involved in performing the study.
If nobody question, I hand over to the chair. A quick one.
The active compound you found in this, which helped in your work, whether you have taken any role to overexpress this compound in peanut, if I have understood correctly.
No, we
so
did not do in peanut. We did only
Yeah.
equally.
Yeah, so that a large quantity of that can be extracted.
Yes, sir. Can try.
Yeah, thanks.
Thank you.
Yeah, thank you, madam, for wonderful presentation. Also,
Then I want to also add one small sentence. Actually, after doing this method for purification, we have developed another method. It is very short. For example, if this method takes 10 days for purification, the method we recently developed takes only 24 hours. And we patented it, and the patent was granted by DNIP.
yeah, very good.
This was granted in
Congratulations.
2023.
Yeah, that will be very useful. So we come to the end of this session for 7 excellent presentations. Thanks to all the speakers and thanks to the organizer for the lunch. Thanks to the co-chair.
Ladies and gentlemen, we conclude the session here. thanking the speakers, chair and co-chair for actively participating in this session. Before we proceed to lunch, we have a small felicitation program planned in a very power-packed sessions. Now I invite the chair AAGB 2025, Dr. Manish Pandey, to come and carry forward the ceremony. Thank you.
Thanks, Shruti. So just we will take not much time, some 5 to 10 minutes of your time, because we don't want you are staying away from your lunch. So actually, in our previous plan, we had to state basically few of the our own reader, but at the same time also like some international partnership we had with colleagues. Many of the researchers you can name if you want, like say Peggy, Dreamwriter from Australia, Baushulia from China. So we wanted to have together, we wanted to take them back to the site, and from ICRI side, we wanted to thank and acknowledge them because they spent whole of their life for groundnut research. It could not happen, unfortunately, but it does not stop us doing something. The person we have with us, and who is Dr. Nigam here with us, he is here. So he is well-known groundnut researcher. I'm not going to tell about his contributions. If you don't know in this meeting who is Dr. Nigam, then I doubt whether you have come to the right place or not. But at the same time, here, not only for his research, at this stage also, he is actively engaged. He came from a meeting. He might have got tired, but still he said, Manish, I will come. And he is here right from beginning to end. So we thank you, sir, for your support and being here. And as a token of love, respect, and gratitude, we would like to felicitate you at this AAGB 2025 platform. We request you please come on the dais for a few minutes. I request to join Dr. Kuldeep Singh, Dr. Janila, James. I'm not sure if Sam is there, Sunita, if I can see here. We have here, please come on the stage. Aile, please come. Charles Chen, we have here. Yes. We have Ryan Harrow here. He's here. Maybe Kenta is here. Yeah, please come. And Ryan, yeah, from US Peanut Board. Yes. So he's not here. Yes, he's here. Please come. And then we have Shawl. So just can you please join? And you know, we have the most young person here, Dr. Nigam, of course. Thanks everyone to join us. We'll request Dr. Nigam to have few of the words from him. And so, sir, please.
Well, friends, thank you very much for honoring me. This honor is not for me alone, but my whole team, right from starting from 1974. Whatever achievement has been there, it has been a cumulative achievement over a period of time. And it has been a long journey, rather hectic journey. And probably I hold the world record of growing maximum number of peanut crops, 80 crops in my career. Because I had the advantage of location. At ICRISAT, we can grow 2 crops in a year. So 80 crops I grew, learned something, learned like that. So it has been a very memorable and pleasant journey. And I am happy that I am handing the baton to young generation who are more smarter, who are better informed, who got new tools. I'm sure they would be able to crack a lot of problems that we were unable to do so in spite of our best intention and best effort. And I'll just say, of course, there's plenty of research going on in different aspects. But if we can handle 2 issues in groundnut, one is drought. And of course, to me, it's a controversial statement I am making. There is too much hype on aflatoxin. It's important, I am not denying, but you can't put all the eggs in one basket. So a balance is required. There are other issues which require equal attention. So anyway, I won't teach you what to do on future groundnut research, but you have to evolve your own research and agenda, but keep the balance in mind, the reality on the ground in mind. Thank you again for honoring me, and I'm definitely obliged, and I feel so humbled with this honor. Thank you very much.
Ladies and gentlemen, thank you one and all. We'll break for lunch now. And for the next session, we'll all gather here by 2. Thank you. Ladies and gentlemen, welcome back for the final session of this AAGB 2025, a session 10, themed enablers for translating genetics into groundnut cropping system. We have Dr. Samuel Jiroge, the chair, and Dr. KSS Naik, the co-chair of the session. Chair and co-chair, over to you.
Well, thank you very much. So I'll hand over to the, okay. So this is a final session as announced. And my name is Sam Jiroge from Malawi. So the first presenter is Dr. Mohan Varma, who's going to be presenting on transforming dryland crop breeding for Africa. So if Varma is ready, we can load the presentation and start. Thank you.
Okay. Good afternoon, all. I think the audience are getting thinner as we coming closer to the end of the session of the AAGB. Okay, anyway, thank you, Manish, for inviting us from the CIMIT to talk about the art scene. What is art scene, how the art scene is operating in Africa, and then overview of the art scene operations in the Africa. So that acronym, I'm not talking, I'm an applied plant geneticist, Mohan, working out in the DCP crops based out at Nairobi. But in my present talk, I'm not talking anything about the markers, genomics or anything, but I am talking about ADSIN, which is a initiative, collaborative initiative of a network operating in the Africa. What is ADSIN? ADSIN is a African Dryland Crop Improvement Network. It's basically a network. It's not a project, but it's a network that is operating in the Africa, bringing several partners and scientists, NARS partners together, and we are working together to improve the dryland agriculture in Africa. So, how the ADSIN is formed? We had got a constant feedback from the NAS partners of Africa that they wanted to involve in developing the product and release the product. So they asked to have the equitable shares of the projects and investments that's going on in the Africa. That needs that this feedback brought up to design, co-design, co-develop, and co-deliver the product in the Africa for the dryland crops. That's how, based upon the feedback, the ADSIN has been formed and developed by the African partners. So, CIMIT has taken a leadership in establishing the ADSIN, in bringing about 18 countries and 3 CGR institutions. The 3 CGR institutions are CIMIT, TIAT, and IITA. And here I would like to emphasize here that I am glad that ICRISAT is also a part of the ADSIN network. And then there is a much opportunity that CIMIT and ICRISAT can work in this ADSIN network going forward to improve the dryland agriculture, dryland crops in the Africa and sub-Saharan Africa. Yeah, this is a logo that was designed by the communication team, and then it was released out. And then CIMIT has taken the leadership in establishing the ADSIN. So when ADSIN has formed, so ADSIN has formally established in the August 2023. Before that, there was a consultation meetings that has held in the Senegal in February 2022. and also the network meeting members came together in the January 2023. So if you see on the left-hand side, this is a big gathering of all the partners in the network who has come up and then formalized in a meeting, and then in meeting discussions held up in the January 2023. After that, in August 2023, it has been officially declared the formation of the AAGB. And then I can say that it has a diverse scientific collaborations. It has more than 200 scientists all across the 18 countries of Africa. So they're all bringing together the experts from the various disciplines of agriculture. It's not only the breeders, it has a multidisciplinary functions that come together as formed as an AAGB. So I don't go through the vision and mission of the AAGB, but I can I can tell that the vision is creating a dynamic environment in sub-Saharan Africa to leverage and accelerate the improvement of the crop varieties related to the dryland crops. And then mission, we work with the stakeholders in sub-Saharan Africa that aims in improving and accelerating the genetic improvements for the dryland crops for the smallholder farmers of Africa. Okay, so you can ask, what is AAGB about? I actually categorize it into 4 categories. What is AAGB about? It's a joint crop research improvement. That is that I can say an example of shared breeding pipelines. So shared breeding pipelines are the partners who come together to develop a particular product. They develop the populations, they advance the populations, and then advance the products related to that particular target product profiles. Okay, and then leveraging the strength. In Africa, one partner might be good at phenotyping, and then other partners are good at conducting the trials. The other partner may be good at the phenotyping, the pathological trials. So we leverage the strengths of each and every partner together, and then work for a common goal for developing the products. And then distributed decisions. So the distribution, this is not one institute or the one NARS partner going to decide, but a group of partners come together and take the decisions and the market segments, product profiles, and the breeding strategies. So once they all agree all together in one place, then these breeding strategies and then product profiles are going to be implemented into the AAGB. And last but not the least, developing of the infrastructure capabilities. So AAGB is greatly involved in deciding the roles and responsibilities of each institution and also addressing the developing the infrastructure and the human resources in the AAGB. So if you see the left-hand side, these are the countries which are involved in the AAGB. The red color represents the ESA regions, and then green countries represents then the countries which are in the WCA. So altogether, the network, the colored green and red portions indicates the countries that are working for the AAGB. Then what are the crops that are involved? There are all the dryland crops: sorghum, pearl millet, finger millet, groundnut, chickpea, and pigeon pea. And also there are a common bean from the CIAT and then cowpea from the IITA. So all these crops are the on which the AAGB is now framed up. Then AAGB has a structure and has a governance. So how the structures looks like and then how the governance looks like is, the structure is, it has a WCA network, crop improvement network, and then ESA dryland crop improvement network. So in the WCA, we have sorghum, pearl millet, groundnut, and cowpea. These work groups use the crop priorities. And then in addition to the work groups, they have technical teams who harmonizes the technical support for these work groups. And then and to oversee these 2 technical and work groups, there is a steering committee that overlooks and coordinates the network activities in the network. Similarly, it holds good for the ESA also, but the crops are sorghum, chickpea, pigeon pea, groundnut, and then common bean is in the ESA. The cowpea is in the WCA and the common bean is in the ESA. And it also does have the technical teams, work groups, which will be overseen by the steering committee in the ESA. So altogether, I was mentioning that there are 17 plus countries and then 200 more than 200 scientists who are involved and working in the African trail and crop improvement in the ARTSIN network. And then who are they? Who are the steering committee people? And then these are the people who is chaired by Charles Brett in the WCA, and then Harish Gandhi in, Charles Brett in the ESA, and then Harish Gandhi in the WCA. And they lead the steering committee teams in their respective places. And then these are the members. So if you see the members and their disciplines, they're all not only the breeders, they are multidisciplinary people who works all across the disciplines in the agriculture. And then what is sustainability of ARTSIN? Means how the ARTSIN is going to sustain. It is going to sustain by the investments that is coming from the Avisa Gates transition. And then earlier it was USAID Avisa, and then ABI. Now it is transformed to the bidding for tomorrow, and the CTH crops to end hunger. These are the projects which are investing and supporting the network. And then the CIMMYT dryland crops invest about 45% of its total available resources in ARTSIN, and then that directed towards the NARS partners. In addition to that, each NARS partner is expected to generate their own resources, and CG partners contribute to the network through their available resources. So in this way, the sustainability of the ARTSIN is taken place in the network. And then who the stakeholders and the partners. And here are the stakeholders and the partners, which includes all the NARS partners. It includes the CG initiatives and also the donors included. I think sooner or later, the ICRISAT also will be coming into this picture. And then I just wanted to say the example of ARTSIN, how it has operated last year in capacity development. ARTSIN has proposed for a calls of 1 million USD. The intention is to have the equal share of ES and WCA, mainly focusing upon the human capacity and the infrastructure capacity of 40 and 60% respectively. So in that one, after getting the proposals, the steering committee has taken the decision in distributing the awards in this manner, that infrastructure development, group training, visiting scientists, and studentships. And WCA got an extra additional benefits depending upon the requirements and the needs in that particular region. And then the other example that has done in ARTSIN is systematically defining the regional market segments across the dryland crops. So this is only for the dryland crops. The copy and the beans are not included, but just emphasizing the example from the dryland crops. There means through the ARTSIN initiative, it has been formally announced the 58 regional market segments, of which 28 are the priority segments, priority market segments, and then 28 regional breeding pipelines. And these pipelines will cater to an area of more than 65 million hectares of Africa. And then ARTSIN is perfectly aligned with the stage and gate system of CG, where the product design will come, in between the trade pipeline will come, where I belong to, and then product development, where the breeders belong, and then product delivery, where the product development managers will come. So ARTSIN is strictly adhering to the stage and gate process of CG. This holds good all across the CG, this pipeline. So it is very much aligned to the stage and gate, where the product design team, where the market intelligence, and then product design teams will come forward, and then they align each other and then develop the product profile. Based upon the product profile, the trade discovery team will work on those traits in discovering the markers and deploying into the breeding. And then breeders will develop the populations and deliver it to the product delivery team. And then thereby the products are going to come out for the launch. So in this way, the ARTSIN is actively engaged with the partners of more than, as I told, more than in 18 countries, and with the 200 scientists, and then delivering the products for the sub-Saharan African region. So Yeah, this is the overall slides that I have to explain about and give the overview on the AdSyn. In summary, I just wanted to tell that AdSyn demonstrates a robust and collaborative model for dryland agricultural, dryland crop improvement in Africa through shared breeding pipelines, and then targeted phenotyping, and then with the multidisciplinary approach. And then this is a novel approach that is going on well-established and well-working model currently for the dryland crop improvement in the Africa. So with this, I think I just wanted to end my presentation. If you have any questions, let us know. Myself and then Haile is also there. We're happy to address it.
Thank you very much. I can see we have a hand, please. Go ahead. Can we get a mic on the floor? A microphone is needed on the floor.
Yes.
Hi, Mohan.
Hi.
I must appreciate the network. It's um, we have market segments, ISO 58 regional market segment. We
Yep.
have ESA and WCA. Whenever we have a discussion of networking, the country-specific market segments are really emphasized. Are we like seeing to it apart from the 58 regional market segments? If we are seeing how difficult it is to deliver what we are committing from that market segments.
Yeah, yeah. As I mentioned, it's not the only one decision, one partner or one institute decision. It's a unanimous decision where the product design teams will unite, and then all the partners who are engaged in those countries will come for the product design meeting before formally declaring that as a target product profile. So it will be consensus and agreement among all the country partners before we set this is a regional market segment on target product profiles. So in that case, all the country partners will agree among each other, and then they define the target product profile or regional market segment. So in that case, we don't feel differences because they themselves involved in the defining a market segment. So so we didn't face any such critical problem.
Thank you, Mohan.
Any other question from the floor or even online? If there are any questions, please let us know. No? Maybe I can ask a question. So maybe you can give us a sense of maybe the challenges that you've had to overcome to get this network moving.
Okay, the challenges is bringing all the countries together and then aligning the each and every country's requirement into a one market segment or product profile is a challenge that we face. In some cases, in that cases we have given more emphasis to the major trades or major targets and rather than the minor one, where we kept it aside. So that was a main challenge where we bringing all the partners together and making one defined market.
Well, thank you very much, Dr. Varma. Maybe we can give him another round of applause.
Okay, thank you very much.
So the next speaker is Taukat Waja on behalf of the Anzutar.
Good afternoon. Myself, Shaukat Waza. I am presenting here on the behalf of Dr. Manzoor. I'll be discussing about ensuring genetic purity in groundnut seed certification through molecular marker based approach. Groundnut is a very important crop. It's a resource of plant protein and edible oil, in addition to antioxidants and vitamins. So it's also a climate resilient crop which can successfully grown under low water conditions, especially in tropical and semi-arid regions. Globally, as far as global production is concerned, it is 50.75 metric tons in case of groundnut, and which is equivalent to $93 billion as annual market. So as per the production is concerned, China is at number 1, which produces around 19 million tons of groundnut, followed by India, which produces 7.1 million tons. As per area is concerned, India is at number 1. So in India also, the states like Gujarat, Rajasthan, Madhya Pradesh, and Andhra Pradesh, these are the major states which produce the groundnut. Like any other crops, groundnut, in order to ensure the purity in groundnut, seed certification is important. As it has been established that 5% impurities in the groundnut may result in the reduction of our 10% yield losses. So seed certification, which ensures the purity in any other crops as well as in groundnut, is important. But in case of groundnut, we do have the problems we are not able to distinguish between the varieties on the basis of the morphological traits only. That's why seed certification is sometimes it leads to ambiguous results. So what is the option? So we can employ the molecular markers based approach, which can be utilized in certification so that we can have the purity analysis which is accurate and which is more reliable. These are the challenges in the traditional system of seed certification. What is that? There is a morphological overlap. Many groundnut varieties look very similar in terms of the color, in terms of the seed shape, in terms of so many morphological details, they are very difficult to distinguish. Moreover, there is a high G interaction in case of groundnut, which means external factors do play a major role in influencing the morphology and visual features of the groundnut. Besides these visual that factors are subjective, which means they are subjective to human errors because they are based on all morphological and visual observations. So adulteration detection, sometimes adulterated seeds, the seeds which are adulterated are very difficult to distinguish from the normal varieties. So it is quite difficult to detect the adulteration. And moreover, these morphological based disease certification is laborious and time consuming. It is a lot of investment of time and labor. So what is the option? Again, we can say the molecular markers have the potential to address this challenge. Besides, besides having the potential of these molecular markers to be used in purity analysis, we have the concerns that mixing the different varieties may lead to the seed that may result in industrial problems as well. As groundnut being an industrial crop, we know the mixing, the different types of seed, like mixing high oleic and low oleic varieties, may reduce the oleic acid and linoleic acid ratio, which will impact oil stability, quality, and storage longevity. So these groundnut products and groundnut oils, they are supposed to have good health benefits, especially they are thought to be very good for the hot problems. But when the seeds are contaminated with the other varieties, high oleic with low oleic, we are compromised with these health benefits. So there is the industrial processing inconsistency is also there. While we are not able to have the
consistent industrial processing. If the varieties are mixed, when we are having different varietal mixtures. So in market economic impact is also there. Impaired seed lots resulting in downgraded oil quality resulted in a reduction in the prices. So these are the different types of markers, like simple sequence repeats. This is based on the number of tandem repeats where we can utilize this for the purity analysis. Simple sequence repeats is be based on that. In addition to that, we have the SNPs. SNPs are based on single nucleotide polymorphism, which can be utilized again for that purity analysis. We have the sequence tagged sites where which targets specific sites for their presence or absence while evaluating the purity. Again, we have the CUSP assays. CUSP assays are based on the fluorescence where real-time PCR can be utilized for this purity analysis. Here are the case studies. There are so many studies which has been carried out right from 2004 to till now, which advocates the use of these molecular markers for purity analysis. But although number of studies, in addition to these, are also educating the implementation of these molecular markers in purity analysis, we have not been able to achieve these markers for utilization in seed certification. So taking this thing into consideration, we have designed one experiment. The objectives of this experiment are given here. We want to standardize the field sampling protocol for marker-based purity analysis in groundnut. We want to standardize the type of markers to be used, we want to standardize the sample size, how to do the sampling, and what should be the sample size. We want to standardize here in this objective. Our second objective will be to optimize the number of molecular markers and the type of molecular markers that we are supposed to test for purity analysis. Taking both these objectives into consideration, we have developed a third objective, which means we need to develop a workflow for seed certification in groundnut using these molecular markers. Here, we have started with the constitution of seed lots. We have developed 4 seed lots. We have mixed a Girnar 5 variety with impurities of CG M1, Chhattisgarh Mungphali 1. So first lot is having 5% impurities, second lot is having 2% genetic impurities, third lot is having 1% genetic impurity, and fourth lot is having 0%, which means it is 100% pure, consisting of Girnar 5 only. We have constituted these 4 seed lots. So this is the field layout where we have sown these 4 seed lots in the field on an area of 0.25 hectare each. So this is the total field of around 1 hectare. So L1, L2, L3, these are 4 lots where we have sown them in the field as layout is shown here. So we have constituted out of these 4 seed lots, we have constituted 10 treatments as is given on left side of the slide. T1 consists of L1 only, which means it's 5% pure. T2 consists of L2, T3 consists of L3, T4 consists of L1 plus L2, T5 consists of L2 plus L3, which means it is 1.5% impure. T6 consists of L3 and L4, T7 consists of L1, L2 and L3, T8 consists of L2, L3 and L4. So purity levels are also given there. T9 consists of full field of 1 hectare where all the seed lots are there. So T10 is the control where there are 0% impurities. From all the treatments, we have taken 5 sets of the seeds, 5 sets of the leaf samples, 550, 100, 200, 300, 600 leaf samples. We can say plant samples. In terms of leaf samples, we have taken from each of these treatments. So this is the methodology, how we have followed this. We have sown them in the field. Each lot we have sown at the area of 0.25 hectare at ICRISAT. So variable number of samples are discussed in the lot sample, 550, 100, 200, 300, 600. We have taken from each of the treatments. Then we are now supposed to do the DNA extraction to standardized protocols. After that, we shall be using molecular markers. Here there are 2 varieties only, Girnar 5 and Chhattisgarh Mungphali 1. We are using varietal specific markers. 1 or 2 markers will be sufficient to evaluate the polymorphism and to do the genetic purity analysis. So data will be generated from the polymorphism itself by simply counting the bands. So this is how we can interpret our results after generation of the distinct polymorphism between the two varieties and counting the bands. We shall be standardizing the minimum sample size. Suppose if our sample size of 50 is sufficient enough, is quite good to represent that seed lot which we have constituted ourselves. If on the basis of genetic purity, our sample, suppose we are taking the seed lot from 5% impurities, which we have constituted ourselves. And on the basis of molecular markers, this sample of 50 is also showing the same results, that is 5% impurities. So this minimum sample size is sufficient, and it is a true representative of that lot or field or treatment. So after that, we need to optimize the marker number here. Just 3 markers are sufficient because only 2 varieties are there. And afterwards, above all, which is the main hindrance in using the molecular markers in seed certification, is the cost involved. So we need to evaluate for the cost effectiveness of these molecular markers. So here we are using 2 varieties only. The broader, while simulating this at the broader perspective in the pharmacy field, as what we need to do, we need to develop a reference base where we need to make the reference library where DNA fingerprinting. We have to revise DNA finger for all the released varieties as well as the popular land races which are there in the public domain. So DNA finger for all these public variety of these released varieties and popular land races to be developed as a reference base, so that farmers can grow there are farmers which can grow any number of unique variety and having any number of varieties as a mixture. So we will be able to trace this if there is a reference library with us. So for that, afterwards, we have to optimize the number of markers, how many markers to be used, and afterwards, cost effectiveness most important. So because we are planning this to upscale this so that it can be used for purity evaluation in the farmers field itself. So cost effectiveness very important in that way. So although our main intention is to do the purity analysis, to develop this seed certification protocol where DNA-based markers can be employed. But you see in our experiment, there are 2 varieties, Girnar 5 and Chhattisgarh Mungfali 1. And Girnar 5 is a high oleic acid variety, and Chhattisgarh Mungfali is a low oleic acid variety. After carrying out the purity testing and after harvesting the produce of these treatments of the seed lots and of the treatments, we shall be evaluating the effect of different contamination levels on seed quality. Seed quality levels, like what will be the effect on its oleic acid profile, what will be the effect on oxidative stability and overall oil quality in the ground, and what will be the effect of different contamination levels. So we are facing some key challenges in this. That is why the DNA fingerprinting protocols have not been utilized in seed certification. The key challenges are we have high initial setup costs for molecular labs are there. So for that, we can have subsidized molecular labs, especially for low-income countries. We do have this lack of standardized protocols for molecular-based certification across regulatory networks, because regulatory frameworks, because standardized protocols are have not yet been developed for the marker-based assays in seed certification. We have the limited technical expertise among seed certifiers. The initial validation for marker efficiency and varietal differentiation is very time consuming initially. We do have the regulatory bottlenecks. We have the high cost per sample for DNA extraction, amplification, and genotyping. And there is a resistance to change from stakeholders lying on traditional certification methods. Limited availability of reference marker database that has to be done. And we have the scalability issues in implementing high throughput molecular assays that need to be addressed. And above all, limited awareness about the among policymakers for the importance of these molecular markers in certification process. These are the challenges, but we do have some economic benefits as well by using these molecular markers in seed purity analysis. We can get highly stable yields and productivity by ensuring genetic purity and varietal integrity using molecular markers, because these molecular markers are able to differentiate able to give the purity analysis up to 100% accuracy. So stable yields we can achieve, unlike the morphological based assays. So these varieties where we have used the molecular markers for purity analysis, they can fetch premium prices in the market. So minimum financial losses can occur due to admixtures, whatever the financial losses we are incurring due to the seed admixtures, it can be minimized by employing the molecular markers. So we can get faster marker access to increase the trade potential through globally recognized molecular certification. There are enhanced investment opportunities through public-private partnership in establishing the molecular testing labs. In conclusion, we see there is 100% accuracy in varietal identification through molecular markers, if compared with 15 to 20% human error by visual methods, because morphological evaluation is based on visualization, where we evaluate the morphological data, and there is 15-20% human error involved. And by virtue of using molecular markers, we can ensure speed and trait-specific seed certification. There is a global impact to ensure food security and boosting the trade compliance. So we need to invest, we need to collaborate, and we need to adopt the molecular marker-based approach for seed certification. Thank you.
Thank you very much. Would like to again open it up for questions, both online and in-house. Please raise your hand and we'll give you the mic. Any questions? Yes, I can see two hands. So would you please pass the mic? Dr. Chen?
Yeah, it's a good talk. I wonder how many % of the farmer use commercial seed?
Sorry, how much % of farmers?
Yeah, they are to buy the commercial seed. They not use the seed to produce their own land.
Online.
Yeah.
You're asking, if I am getting you very clearly, you're asking how much farmers are able to buy the seed online.
No. I mean, how many % of farmer they are affordable to buy a commercial seed?
Commercial seed.
Okay. Here in India, it varies from the state to state, and especially in case of groundnut, if we are distributing the certified seed to farmers as well, that seed is also having the contamination. So the farmers that are cultivating the seeds saved from their own sources, that is also the contaminated. So because of these morphological problems that we are having, although farmers are very ready to adopt the seeds, to adopt the improved seeds, but the problem is that they are not able to get much more out of that. They are purchasing the seeds at very high cost, but they are not able to get that much of produce out of that. So they are very reluctant in adopting the seeds, because it is we are not able to distinguish the varieties on the basis of morphological markers, and whatever the seed we are providing the farmers, that may be contaminated to some extent.
Okay, good. There was one question at the back of the room.
I would like to know how many markers represent in your QC panel, number one. And
here in our case, we are using two varieties, Girnar 5 and Chhattisgarh Mongphali 1. We can directly use varietal specific markers to distinguish between the two varieties. So 2-3 markers is sufficient. But when we are, suppose we are going to the farmer's field and we are collecting the sample, at that point of time, we know farmer will tell us that this is our variety. Suppose this is any variety A of groundnut. For that, we need to have a reference database. reference database, where, as I have discussed it must contain all the released varieties, including popular land races of farmers, where the DNA fingerprint of all those varieties should be there. When we are collecting the variety from the farmer's field, we will be that using that particular varietal specific marker. Suppose we have screened all the varieties, and for all the varieties, there are 2, 3, 4 markers varying from variety to variety. These markers will be quite able to distinguish these varieties from all the other existing varieties. So it will depending upon variety to variety. Even some varieties, we may be able to distinguish some variety based on 1 marker only, and some may need 2, 3 markers. So it is based on variety to variety, and it will be after the development of the reference base, we can only tell how many markers are needed for which variety.
Thank you very much. You talk about 100% accuracy in
Yeah.
molecular markers, but I beg to differ with your thought. Say, I give example of say Girnar 4 and Girnar 5, they are sister line, everything looks similar, but how is like accuracy in those scenario where we have exactly similar sister lines, still you quote about
Yes,
like 100%.
that is what I am telling you. We have the exactly similar lines based on the base of morphological features. Since molecular marker based assay is directly dealing with the genotype of the plant. So if it is dealing with, and at the same time, we have the reference library. Which this reference library distinguishes Girnar 5 from all the other existing varieties, whichever are in the public domain. So by virtue of this particular markers, 2, 3, whatever the markers may be there, by employing these 2, 3 markers, Girnar 5 will be distinguished from all the other contaminations that are there in the seed lot. So by virtue of that, we can achieve the 100% purity.
Thank you very much. I think we'll have to stop here. Another round of applause for Dr. Waza. Thank you. At this point, we have an online presentation by Dr. Rupa Vadaram, who's going to make a presentation on artificial intelligence for monitoring pest and disease in groundnuts. So if you're ready, we can. Okay.
Can you see my presentation, please? Can you see my presentation, Sam?
Hi, Srikant. I think you can proceed, but maybe you need to unmute your mic.
Yeah, I have.
We can't get any audio.
You can't hear me?
We are able to hear you.
Yeah. Okay.
Okay,
so
good.
Thank you. Yeah.
Thank you.
The topic I was given is the AI for monitoring pests and diseases in groundnut. So my name is Srikant, and I've been working on an AI-backed application called Plantix for the last 10 years. And this is the most important thing that is happening is farmers at the center, and everybody, including knowledge intermediaries, government organizations, research institutions, distribution channels like the private sector for inputs, pesticides or seeds, agri marketing units and distribution centers. Everybody deals with the farmer who is in the center. The biggest problem is reaching out timely way. And I think that's where the motivations of each of these players is different, and getting them on one platform is the biggest challenge at the moment. So this is worldwide, at least in the emerging economies, in Africa and Southeast Asia. This is the biggest problem we see in reaching out the last mile delivery that we talk to the farmers. The challenges for artificial intelligence applications in agriculture, I have put some of these things. The very agroecological diverse conditions in which the crops are raised, the agronomical practices followed by different farmers, which bring in variability. Of course, the soil type and the nutrients present in the soil, which brings in a lot of variation in the growth and development and the yields. We can't limit about weather. It's really highly variable and unpredictable, and that causes a lot of variation. And in fact, if we are looking for a AI product development, then we need to capture all the variability that happens to train the data sets. And of course, the varieties that the breeders come out with, they themselves have a lot of variation in their performance and how they respond to these environments that they are in. You'll not be surprised. Actually, I put this honeybee drone in the middle because that's what Walmart patented way back in 2018 in March because of some damage that the honeybee industry had in 2007 and 2008 in the United States. So 1 billion of these drone bees can be produced in every week and given back to the farming community, or there's a company that can make money if such situation comes. So the world is going at a very faster rate, and AI is something that we have been hearing since the Gen AI in November 2022. But thanks to ICRISAT's openness to embrace evolving technologies, so we believe that some of the challenges in agriculture can be addressed, and one of them is the limitation of the agriculture extension service to reach out to all farmers, especially in diagnosing their pests and diseases in a real time, because that's the key, and that's the key challenge we face, because the feet on ground are relatively less. In India, for example, each extension officer will need to serve as high as 15 to 20,000 farmers in their jurisdiction in some of the districts of India. So this is really complicating the way the farmer gets access to right chemicals, agrochemicals. If there is a problem in their fields, the traders are the de facto extension workers, or you can call them as the plant doctors. But we see the vulnerability of the farmers being abused in the evolving and emerging markets like India. So what Plantix is doing, just image processing, that I can say if your eyes can see and spot the difference between the two images of tomato leaves, I think that's what we can enable the software to do the same. AI can be made possible to do this type of a differentiation and also classify the image and detect it with very high accuracy. So if an image from a smartphone is uploaded by a user, a farmer, it can be a researcher, anyone, then it gets analyzed in a couple of seconds. And this is deep neural networks. These are one of the models that we use at the back end. We don't need to go in detail into it, but that's what happens. The magic happens in terms of detection and the response within a couple of seconds, depending on the speed of the mobile data service that the user has. So in short, if you see on the right side, the video, aiming at the crop, taking an image, uploading it, and getting back the result and the details about it, how to manage it, how the recommendations of organic, biologicals, or chemicals, everything is given within seconds. And this is possible because of the collective work that I've shown in the first slide, the partnerships that we had in India with the local state agriculture universities and the Indian Council of Agriculture Research Institutes to do training of very large data sets, including groundnut. So right now, Plantix is now, it was a German startup, but now they are based in India, and it's an Indo-German startup based in Indore. So it is available in 18 languages. About 45 million users have already been on board at an average of 1 million plus users per month on an average. It can classify around 780 pest diseases and nutrient deficiencies in 60 popular crops. This is available in Google Play Store, and it's available in most languages of India. And also recently, in December 24, we launched it in Swahili in Nairobi, Kenya on 10th. The first question we will get is the accuracy. We can boast of 93% accuracy for around 450 classes for these different classes of pest diseases and deficiencies. So that's significant and very high. It's not a statistical number. It's about the performance of the application. And I'll give you more details on it in the coming slides. one of the reason that this app got used by farmers and they are still using it is because of the user interface, which is very simple and understandable. So this is very critical. There are so many hundreds of applications out there. Why is this app getting used by farmers? So one of the main reason is the user interface and the user experience. We will delete any application that we do not use for our personal use. So also a farmer will immediately delete if there is no use of that app for them personally, I mean for their growing crops. There are few features about this Plantix before I jump into specific information that I want to share on groundnut pest and diseases. It's not only doing image detection, it also gives management options, and there is an online community that sends 25,000 messages per day in those 18 languages and gets answered within 6 hours. And if a farmer has he knows when he or she knows when the groundnut is sown, the whole agronomy is automated so that they get push notifications during the entire crop cycle of groundnut. Then in India, we have something called partner Dukan, which is like we have geofenced. If a farmer uploads an image, we know exactly where they are located and the time they have sent the image. So based on that, the shopkeeper is also intimated about the opportunity of a sale. That's how Plantix business models have been created. It is free for the farmers and it is also freely shared its APIs to government machinery in India and also outside India. If you see this, no wonder India seems to be one of the biggest markets for the users. Almost 74% of the users are in India. This is the performance evaluation that we did on Plantix. This started during the pandemic time. Assuredly, I can tell you, this is a huge improvement during the pandemic time. We have each sample for us is a video of 8 to 9 seconds that you can see on the right hand side. So this is what is happening, and every sample video is being watched by an expert. When I say expert, it's either an entomologist, pathologist, or a physiologist or a soil scientist to determine whether it is accurately identifying a disease and whether it appears in the top few or it is not identifying at all. So what is the advantage of this type of artificial intelligence being used? Because each image has latitude and longitude and also the timestamp, we are able to aggregate and make heat maps of every pest and disease for each and every crop around the globe. This is all automated. This analytics is available, and there are few people doing research in their PhDs also. I come now to use cases we had, that is images are around 5,48,854 from around the globe. And India is almost saturated if you see the globe. So this data is from 1st of January 2023 up till December 31st, 2024. This is just 2-year data that I'm talking about when I'm talking about this global spread of pests and diseases that we had in terms of notifications from the farmers around the globe. Now, if I see the list of pests and diseases and nutrient deficiencies, then I can see the number of counts or the users who have reported. It is very high on nitrogen deficiency. It's almost 78,000 notifications came only on nitrogen deficiency. Then if you see the other, of course, there is a healthy. So in order to classify every crop, and even a healthy crop needs to be identified by Plantix. So that's why you see a classification called healthy also. But next to it is foot and collar rot. And then we see helicoverpa, butt necrosis, tobacco caterpillar, and helicoverpa. So, and leaf miners too. Here are the 5 top problems reported in India. And I have put in red the outlines of the 5 districts: Gujarat, Rajasthan, Andhra Pradesh, Maharashtra, and Uttar Pradesh, from where we received. These are the top 5 pests and diseases that we could see in terms of notifications that comes from farmers' fields. So if you see something in the literature review, I'm giving you actual data source from farmers. So this can be very different from what people have reported earlier. But this is the truth from ground truth from the farmers' fields. So foot and collar rot stands head and above the rest. I'll give you individually how the spread happened in the last 2 years. And if you see the right side screen, I just put a timestamp and I'm going month-wise, how many we got every month, right from January to December 2024. We can see Gujarat actually has the highest number of notifications that came. So it's around 13,800, followed by Rajasthan, 8,700. But if you say Andhra Pradesh and Uttar Pradesh, far below, at least one, not even one fourth of the size of what has been reported. This also indicates that the number of notifications can be correlated with the number of farmers also. No wonder Gujarat and Rajasthan also have very high numbers of growers. So that's one indicator we can see. And this ground truth can be also used for land use, land cover discrimination using remote sensing. Here again, if we see tobacco caterpillar, the incidences, Gujarat stays way ahead, while Rajasthan and Andhra Pradesh are almost less than half behind them, behind Gujarat, and then followed by Uttar Pradesh and Maharashtra. This is about tobacco caterpillar. This bud necrosis virus, I think this is people who are students doing work on virology, viruses in and their effects. You can see we have this data we can share with you if you're really interested, because this is the real-time data that we have captured and put on a spreadsheet. This again, if you see, not much of a difference between Gujarat and Rajasthan, where it has been reported. Coming to Helicoverpa, no wonder it's almost not significantly different from each state, but the numbers are around 4,000 to 5,000 in the 4 states that we highlighted, because they contribute to almost 75 to 80% in some of the diseases. So coming to leaf miner, Andhra Pradesh, most of the thing is happening there in Andhra Pradesh. So the report almost 6,000. This is the first time where this also gives you an indication of the prevalence. It's a correlation that we are talking in terms of what is more prevalent in a specific state. But these are the real-time information with the timestamp that we have. And now the most important thing on the left-hand side, because if somebody is based in a geolocation, for example, you are in Goa, I know there are very few farmers farming peanuts in Goa, but in a 20-kilometer radius, based on the farmer, we can actually tell and give a push notification to the farmer. Here on the left-hand side, it is in Telugu language. Sorry that I could not get an appropriate groundnut-related push notification, but it says here a specific pest or a disease has been reported widely near you, so be careful. So this is what AI-related monitoring can do to crops. It can alert the farmer on what is happening near to your fields and to be vigilant. That gives them extra time to be prepared to address and control their pests and diseases. Thank you.
Thank you very much, Srikanth. We'll open it up for questions. 1 or 2 questions, please. Srikanth, maybe I can ask you a question. So you said it gives you data on prevalence, so which is more or less like incidence. But does it give you an idea of how severe the problem is, which is also linked to actionable interventions?
Yeah, not in groundnut, but we did it on fall army worm because it was invasive and it really took down maize in India in 2018 May. So a real-time tracker can be very useful for the policymakers and those who are working in the departments to actually put their extension people to the right place and also see those agrochemicals are available in those locations. So that's the use to be, it's more preparedness by, it's not an early warning, but we know as the numbers grow that, yeah, there is a huge problem. Last week, it was say 100 people reported. This week, somehow 1,500 from the district. So that's an indicator. If people are taking images, we believe that if there is a significant difference and a very high volume suddenly, then that's like a trigger for alerting the administration.
Thank you.
Srikanth, are there any cases of forewarning you did based on your data?
Yes, we are working on a couple of pest and diseases in rice, especially. And this data, which is available, actually, I can provide it to some students who can work on using climate data and soil. And because you also know the ranges of humidity and the temperatures for any disease, actually, this is very useful. I don't get proper students in agriculture, especially pathology or entomology, who are willing to do modeling seriously, because this data is available, which was never there before for us. So people from Scotland, somebody else is working on it. They are students, of course. So I would really encourage, if somebody wants real quality data, this is it's like a ground truth in their model. So the spatial in time also is available, and you can get the seasonal weather, which is already there, any reflectance data from satellites. How can we not use this data and combine? This is the treasure, sir. I really love to work with PhDs who are willing to do some modeling work to early predict. I think that's the real key. Yeah.
Actually, I also would like to ask on the same grounds what sir was asking. We also work with Helicobacter bollworm, and we know some of these protease inhibitors are very effective in leading to the mortality of this Helicobacter. But the main question is always I face is, how do you detect them in the early stages? Once if it goes into the plant, it is difficult to control them. So you have shown the pictures, no? That in these states it is very high, all this. That is taken in the adult stage or which stage of the
Yeah,
infants?
a very good question, ma'am. Actually, without seeing the pest, actual Helicobacter larva, we tried to train the algorithm using the plant damage symptom itself. So Spodoptera has a different signature. Very minute. For us, we get very confused actually, unless we see the larva. How can you say this? This is the first question I encountered initially. Then as we kept getting hundreds and even thousands of training data images, we could see significant differences from these worms also. The damage symptoms. Yeah, that's helping us.
Thank you very much. Thank you. We end here. Thank you very much, Srikanth, for a very interesting presentation. Another round of applause.
Thanks.
At this point, we'll move to the next presentation by Dr. AAGB, who's going to talk on evapotranspiration and deficit by precipitation as an eco-climatic factors driving oil yield in groundnut under multi-location trial. Is this an online presentation?
No.
Over to you, Dr. Ajay.
Thank you, Ikrisat. Thank you, Manish. And then thank you, Chair and Co-Chair, for giving me this opportunity to present my views on this envirotyping. And then just I'd like to thank Srikanth, also my earlier presenter, who had just given the hint that how we can use the climatic variables in the disease modeling. And here, I would continue with that point, and then I will try to present my views how we can use these climatic variables in addressing several of the issues in agriculture. And then here I will be mainly concentrating on genotype environment interaction, but it can also be applied in our different crop production or production trials also.
So I think all of us, we know this, that phenotype is mainly governed by genotype, environment, and genotype-environment interactions, right? But we have we have advanced a very lot in terms of genotyping. We have several of the genomic tools, and then even phenotyping also, we have a good high throughput phenotyping platforms are available. But have we understood the environmental component for that? And then without understanding the environment and their genotype environment interaction, is it possible to better understand the genotype and then better identify the better genotype for the location? So this is in this regard, I'll be presenting my talk, and then for this, I'll be using the envirotyping concept, which was proposed by Zhu in 2015. Yeah, here, just to emphasize how the environment and the genotype environment interaction plays in a role. As you can see in one of our case studies, here we had almost 40 environments, and then in another case studies we had 4 environments. As the number of environment increases, its contribution also increases a lot. And then, as you can see in the first case, genotype contribution is hardly 1%. So with this just 1%, it is very difficult to pinpoint any genotype for a particular location. So that's how complex is the environment, and then that's how understanding the environment becomes very important. And then how we can use it on our breeding programs or in any of our trials. This is our in the NARP National Agriculture Research Projects. We had different agroecological regions of India have been divided into different regions, and totally we have 122 different agroecological regions. So with such a diverse environment, and then without understanding about this environment, becomes very difficult to identify any genotype for a particular location. So, all of us want a very superior genotype for any of our breeding programs. But so again, our environment interacts, GEI is playing a major role. So in this regard, we want to understand our environment. But how do we do it? So Zhu, in his paper, he presented that we can try to, in order to understand this environment, we can have different environmental factors. Like we can have this temperature, we can have the solar radiation, so wind speed, we can have even a soil data, or we can have a different biotic and abiotic factors also we can club it. Along with this, along with it, we can also use the different crop growth stages. And then together, these crop growth stages and different environmental factors, together they can be constituted as ecoclimatic factors. And these ecoclimatic factors, further, we can use it for our envirotyping. And then, so understanding about these ecoclimatic factors will help us in better understanding the GEI. So this is how this model works. So details about this models is available in this literature, so it can be accessible easily. So they propose that we have a large data set like a NASA website or a European Commission. They have a lot of weather data is available online. We can using our latitude and longitude, we can collect a weather data for any specific location, and then we can process this all this raw weather data for our analysis, and then based on this weather data, we can try to see how many what is the association between the different locations in terms of this weather parameters. In addition, we can also try to include the different crop growth stages also, and then along with this, we can integrate with the environmental parameters and develop in a matrix wherein, and then further group we can have a grouping of the locations. And going to the further moving ahead, we can also integrate our genomic resources and also our phenotypic data, and then further develop environmental kernels, and then by way we can we can have a better understanding of genotype environment interactions. So just to study one of our I have used one of our case studies where we studied 6 of our breeding materials across 6 locations throughout India, and then we try to study how this climatic factors is affecting the oil yield. So these are some of the weather parameters that we have used it, and then most of it is available were obtained from the NASA website, and then few of them were analyzed using this weather data processing. This is, I think, Boot et al. 1982. He proposed different crop growth stages, and we can use this information, and then we can see we can use this information and then combine with the climatic factors. Boot in 1982, he proposed 11 crop phenophases, but here for my study, we have just using a crop emergence, and then first tetrafoliate stage. When after emergence, when the first 4-leaf stage is available, and we see that stage we have used, and then flower initiation, and then beginning of the pod development, and then when there is a full crop, when one of the seed is fully mature, that is R6 stage, and then R8 stage, whereas crop is physiologically mature. So we have used 6 different crop stages. So after combining this crop growth stages and 19 ecoclimatic factors, this is how the matrix looks like. So totally we have 108 ecoclimatic variables. Now, for each and every location, these 108 variables these matrix will become our variables for further analysis. Adding to it, we have added our oil yield and pod yield data for that, and then to identify the factors which are associated with the for better oil yield. Again, so for all this analysis, there is a package called NVR type in R. So this is very helpful package where you can perform all this analysis. Again, there is one more package called MDA tools, wherein we can do the partial least square regression models. We can run those models in that, and then try to get the variable important by projection and selectivity ratio. So using all this, we can identify the EFs which are associated with the oil yield. So after with this analysis, we could group 2 of our environments. That is Anand, which is in Gujarat, and then Mainpuri, which is in Uttar Pradesh. So they formed one cluster, whereas Hirur, there is Shirgaon, Akola, Raichur. These are mainly located in southern part of India. They have grouped into one cluster. And then major differences, weather parameters which could differentiate these 2 groups were the humidity, radiation, and then temperatures. So even with respect to yield also, we can see that Anand and Mainpuri, they were comparatively, that group had a very better high yield, and whereas other group had a very low yield. So with 108, 109 ecoclimatic variables, now using the variable important in projection, we could pinpoint to 10 we could come down to a few important variables which are affecting the oil yield. Like we have an thermal amplitude at tetrafoliate stage, and then we have an again thermal amplitude at just a seedling emergence stage, and then again evapotranspiration, solar radiation, and then deficit by precipitation. That is a water stress. Again, number of days above 33 degree when during R6 stage. That is one more factor which can affect the ground oil yield. Here, with a beta coefficient, we can identify whether this factor is having a positive or negative effect. We can see with a VI deficit by precipitation at tetrafoliate stage, it is having a negative effect on oil yield. As we have more water stress, oil yield is going to be reduced. Otherwise, for example, I'll take R6 number of days above 33 degrees centigrade at R6 stage. If we have a more number of days during the pod development stage with 33 degree and above, as the number of days are more with the 33 degree and above, we can expect better oil yield. This is how, then we also perform the selectivity ratio again. We could shortlist a few important ecoclimatic factors which are which can affect the oil yield. And also we also perform the validation with our earlier with original earlier variables and also with the selected variables. In the original variables we had 109 eco factors, whereas now with after selecting with the selected ones we could come down to just 9 variables. So if we can work on these 9 variables, we think it will be very helpful to improve the productivity and an oil yield also. As you can see in the selected variables, we could reduce the we could improve our R square also from up to 0.8. And also we also plotted the R square values of each and every factors with the oil yield. So here evapotranspiration at a tetrafoliate stage has almost a 0.8% R square, whereas a deficit by precipitation, it has a negative effect of a 0.7. So there the and then number of days above 33 degree at a full pod development stage, that is R6 stage. Again, it has a R square of a 0.7. Likewise, vapor pressure deficit at celiac emergence, it has a 0.6. So, but all other now from 9 variables, now we could come down to 3 or 4 important variables with a very high R square of 0.7. So again, going back to my equation of the Breeder's equation, that is a phenotype is equal to genotype, environment, and GEI. So we hypothesize that with the envirotyping and utilizing these environmental variables better in our breeding programs using this envirotyping platform, we can better able to understand the GEI and then use it on our programs. Again, with this envirotyping and then partially square regression models, we could pinpoint a few important variables now. Just if we can increase the number through agronomic practices, if we can have more number of days, with if we can increase the temperature in the field by a few degrees, we can expect higher yield. So now, what is this? Further, how we can use it in our breeding in various other aspects? We hypothesize that first is we can study the high yielding and low yielding environments, and then we can see what is the difference between these environments, why these environments are high yielding. For example, this Mando region in Rajasthan, northwest part of India, they are very high productive regions. Whereas this Anandpur in a southern part of India is very low productive regions. So can we study these environments better, and then can we study their soil parameters and all other variables, and then better identify why these environments are high yielder, why these environments are low yielder, and can we convert these low yielders into high yielders? This is one application that we can also do. And then other application yesterday. Purohit, he was making a presentation on his RGA. Again, we have used this pinpointed with the Vridhachalam Center. Vridhachalam. This is one small county in Tamil Nadu region of India, where in all of the when we compare this location with all other parts of India, anywhere crop will mature between 15 to 20 days early in this location when compared to all others. Even the late maturing ones, say if the late maturing ones are for 125 to 130 days, in other parts, they will mature by 100 to 105 days in this location. So we have, naturally, we have an advantage of 15 to 20 days in this location. So can we study this environment better so that we can mimic in our artificially, we can mimic in our field conditions and then advance our materials faster. So this is some of the concepts that we have thought of using this environment. And also, we also made one study with yellow mosaic virus in Chile, where NPPGR regional station, they had yellow mosaic screened their germplasm with yellow mosaic virus in Chile. And also using this envirotyping, we could identify the exact factor which is able to, contributing towards higher disease resistance. So these are some of the applications of this neurotyping. I hope this will contribute towards a better understanding of the GEI. And these are some of my colleagues. And then I'd like to thank the co-chair, and then ICRISAT for giving me this opportunity. Thank you.
Thank you very much, Dr. Ajay. Questions? I see one. Do you please pass the microphone?
Hello, sir. Sir, it is a very informative lecture from you, and I totally understand that objectives of your study were to shortlist the factors which are responsible for high. Sir, my question is that here you have been studying 6 to 7 genotypes, including TAG24 and all. So with that, you have built this model. But sir, did you have any testing set? So that was used as a training set for your model. Did you then use this as a prediction model also to test some other genotypes?
Actually, already 2 papers are available in the online. I just gave the Costa Neta paper. It's already been, the model has been used in several other crops. Here, I was trying to understand for with respect to oil yield, which factor may be affecting the oil yield. How can we increase, can we understand the environment better for increasing the oil yield for the environment? So, sixth genotype that I have taken is just for the case study. But in general, this is better for understanding of the environment.
Yes. And that is why I wanted to know that with those conditions, have you checked the other genotypes also? Like,
Yeah, now we are working with other, say I showed you about the future works. Now we are trying to, I have got to take a data from for the high yielding and low yielding environments of India. Now we are going to test this model. So so how can we increase the improve the productivity of the low yielding ones by studying the high yielding regions? So that with a few management practices, can we improve the yield? So this is our concept.
thank you, sir.
Thank you.
So it's a very nice presentation, very in-field data for rapid generation advancement studies, temperature studies. I want to know that as we increase the number of environment, you say the genetic component diminishes to 1% or maybe less than 5%. But how about the soil? This environmental data also comprise of the soil.
Yeah.
So can we take that into account to see how much that attributes to environmental effect?
Yes. Yeah, I showed this matrix, right? Here I have considered in the column row wise, I have considered only environment, but you can also include your soil data like say your pH, soil content, EC, several soil parameters.
You mean to say we can add several
Any
aspects to and see its effect?
you can add any abiotic factors.
That's really nice.
Because weather is not just environment means weather is not the sole criteria. Any environmental factors we can try to add.
Yeah, I enjoy your talk. It's very complicated, but it means a lot. My question is, have you consideration of the higher lay tree? As we already know, higher lay, if you grow in the high temperature environment, you easy to maintain the higher lay. If you grow in a low temperature, most likely will difficult to get that high. So that's my question.
Yes, sir. If I have the data, I think we can try to do it, sir.
In your line, you're not including the higher lay.
Yeah, because with respect to high oleic, we don't have the we didn't concentrate on those lines, but we can do that work also.
Thank you.
Thank you.
Thank you very much. Great applause to him. A very good discussion is there. Thank you, Ajay.
Thank you, sir.
Thank you. The next speaker is Israr Mazid. So his topic is a systems approach to agriculture sustainability, building resilience through the landscape management and genetic advances.
A very good afternoon, everyone. My name is Israr Majid. I'm from ICRISAT, Hyderabad, working in ICRISAT Development Center. Although this conference is more primarily focused on advancements in genomics and biotechnology in case of groundnut, but my presentation will be more focused on an integrated approach where we consider different components of the landscape, including soil, crops, to be able to sustain the what demand we have got to the projected demand in 2050. So for Indian agriculture, we have several challenges in terms of soil degradation. We have overgrazing, deforestation. We have permanent seasonal fallows. We have poor agriculture productivity. We have climate change. And when we talk about soil degradation alone, about 120 million of hectares, 120 million hectares of land is affected by soil degradation. This not only reduces the crop yields, but also has an impact on the soil cation exchange capacity, as well as the cost of cultivation gets increased. If we look at Okay, if we look at soil as a function, soil cation exchange capacity as a function of soil degradation index, we could see a strong negative correlation between the soil degradation index and the nutrient holding capacity of the soil. Plus, if we look at the yields are getting decreased with the increase in soil degradation. We can see with the improved soil quality, we see the significant improvement in soil yields. We can see the reduction in seasonal fallows. And when we talk about climate change, there is no debate whether the climate change is happening or not happening now. It's happening now. It's changing our lives. It is changing. It is driving our daily lives. If we look at the difference in annual rainfalls between 1960 and 2020, that means the 30-year average rainfalls, we see a significant shift in the rainfall patterns. We can see a decrease in rainfall in the northern and central India, where we can see the increase in rainfall in some of the parts of Odisha, Andhra Pradesh, as well as Gujarat, Rajasthan. 200 millimeter of increase in rainfall in 50% area of the country, and the same magnitude of rainfall has decreased in 36% area of the country. If we see the changing face of irrigation sources in India, in 1960, the groundwater withdrawal was only 25 kilometer cube, which has increased to 300 kilometer cube. And the number of bore wells standard at 1 million, and that has increased to 33 million. A major reason is that the state and central governments providing subsidies to farmers to support their irrigation demands. As a result, there has been increase in the number of bore wells well recovery period was brought down to 10 hours from 120 hours. And crop productivity increased by 20 to 60%. And groundwater, as I said, groundwater levels, if we look at the treated versus controlled watershed, groundwater levels were increased, whereas outflow was reduced and the base flow was increased. So there is need for system level transformations following ecological principles for resource optimization. There is a need for water-based planning for designing NRM interventions. There is need to strengthen the ecosystem services. We should focus on minimizing the water carbon energy footprints. We should target carbon and nature credit. Thank you.
Thank you, Mazid. I invite questions. Anybody? If there are no questions, our chair wanted to have one.
No, thank you very much for your wonderful presentation. So I think at this point, we'll move on to the next presenter. So thank you very much. No, we'll move to the next presenter. Thank you. Another round of applause. Our next presentation is going to be by Dr. Jihendra Kumar Tiwari, who's going to make a presentation on groundnut cultivation in Buchenland, a path for farmers' prosperity. And this is an online presentation. So could we please load the presentation? Thank you for your patience. We have, this is the second last presentation of this session.
Hello, good afternoon, sir. Can you please load my presentation?
Good afternoon, Dr. Kumar. Can you hear us?
Yes.
Okay. So please go ahead with your presentation. You have about 15, 18 minutes.
Okay. It is visible?
Not yet. I think it's still loading. Are you controlling it from your side?
Okay. If you please, you can, otherwise. It is visible?
Not yet, no.
Yeah, your carries.
Dr. Tiwari, do you want us to run the slides? Here we have your presentation.
Okay. Sir, can you please play my slides from your side, sir?
Here.
Meanwhile, we figure out this glitch. Chair, I give permission for the audience to put up their questions. We can take some questions here.
Sure. Oh, okay. I think that has been fixed. Somebody has a question?
Anybody have a question? We can proceed.
Okay, thank you very much. So Dr. Kumar, your presentation is showing, so please go ahead and we'll control it from this side.
Okay. It is not visible to me.
It is visible from this side, so maybe you can
Okay,
just
okay, no problem, sir. So who will move it? I will try to finish in 5 to 10 minutes. Okay, sir.
Yes, we will move it from this side, so just say
Okay,
slide
okay,
one.
no problem, sir. I will start now.
Thank
So, respected chair and co-chair and organizing committee, good afternoon to all of myself, Jiten Kumar Tiwari from Rani Lakshmi Central Agriculture University. Today is my talk on groundnut cultivation in Bundelkhand, a path for farmers for prosperity. So, here I have data on area production productivity of 2022-23 and 23-24. I want to show something change in Madhya Pradesh and Uttar Pradesh. Here, the 10 major groundnut producing state of India. What in 2023, 22, 23, Madhya Pradesh comes area-wise comes in 4th rank, but Uttar Pradesh comes in 10th rank. But in 23, 24, Madhya Pradesh comes in 3rd rank, and similarly Uttar Pradesh comes in rank 9th. And this scenario also for 24 and 25, and recently we have data on groundnut production. The total area in India is about 5.396 million hectare, and the production is 11.31 million ton, which is previously around 10.18 million ton. So why I mentioned here Madhya Pradesh and Uttar Pradesh is because I will talk about it later. The Bundelkhand is the central part of India. Some part comes under Madhya Pradesh, and some part comes in Uttar Pradesh. So previously, it is a pulse ball of India, but it is a primarily agrarian, vulnerable to natural calamities, low level of industrialization and urbanization is occur. But it is a pulse ball of India, and sesamum is also a major crop. But due to erratic rainfall during the month of September, all the crops was collapsed. So farmers shifted to groundnut cultivation from 2015 to 16. So groundnut produce a assured income for the farmers. So previously in 2015-16, the per capita income was 50,000 to 60,000. But at present in 2022-23, it is rose up to 1.5 lakh to 1.6 lakh. So this is the change due to groundnut cultivation in this region. The brief overview of the region is it is comprises 7.1 million hectare area and 14 districts, each 7 from Madhya Pradesh and 7 from Uttar Pradesh, and it is a semi-arid plateau, agroclimatically diverse region, and it comes under the agrodiversity region 10 and receive annual rainfall of 800 to 900 millimeter. So major challenge in this region is water scarcity, soil degradation, frequent drought, erratic rainfall, low productivity, and declining genetic diversity due to some developmental project and climate change impact. So why groundnut in Bundelkhand? There are 3-4 points by which farmers choose to cultivate groundnut in Bundelkhand. First is this climate. It is a hardy crop and suitable for this region, and soil is also suitable for production of groundnut. And there is lots of fallow land in Bundelkhand. in 2015-16, about 0.55 million hectare area is there, which is fallow in Kharif season. At present, in 2022-23, it is 0.22 million hectares. So it provides an opportunity to expand upon our groundnut cultivation. And it is a historically rich region. So tourism as a revenue stream for groundnut production is also there. It is rich in proteins and other minerals. So it is also provide a solution for alleviating malnutrition in this problem in this region. So it a small brief scenario of groundnut production in Bundelkhand. Here the Madhya Pradesh productivity is 2.02 ton per hectare. But in case of Uttar Pradesh, Bundelkhand, it is only 1.16 ton per hectare. Average is 1.56 ton per hectare. The gap is about 0.52 in average 0.5 ton, and in Madhya Pradesh and Uttar Pradesh, it is about 1 ton per hectare. So there is need to improve for in groundnut production. So this is the SWOT analysis of groundnut production in Bundelkhand region. So I have discussed about the strength and opportunity. Some weakness here is very high seed rate, poor allocation, resource allocation, high cost of cultivation, and labor cost, very high seed rate, very less seed replacement and varietal replacement ratios. And these are the challenges across the groundnut value chain in Bundelkhand. The first is seeds. Farmers used only very old variety TG37A, which is not pure and mix of many varieties. So they will not get up to the mark yield, and there is need to introduction of high oleic varieties like Grinar 4 and 5. In case of sowing, they will do broadcasting of different broadcasting method for sowing, and late sowing also done, so they will not get up to the mark yield. And in case of, they will also not do good agronomic practices, which is very essential for getting the maximum yield. So in case of harvesting, they will do manual harvesting, which increase the labor cost and 5% yield loss in field condition. In case of post-harvest operation, after manual threshing, farmers are washing the groundnut to remove dirt instead of grading machines. This increased risk of aflatoxin. And there is a problem of storage, and they have only 10 to 20% processing in this region, and remaining 80% goes to Gujarat and Maharashtra. So there is no branding of Bundelkhand products, no tasting facility in the region. So these are the challenges across the groundnut value chain in Bundelkhand. So we have key groundnut promotion strategies for Bundelkhand. Our research priorities are development of short duration, high yielding, and abiotic stress tolerance varieties for the region. And major interventions are capacity building, training program, frontline demonstration, and in good agronomic practices, how we will do water management, soil management, and improving the infrastructure for post-harvest management. And the timely input supply is the key factor for getting the major, getting good yield and suitable price, and some government policies needed to intervene. So these are the keys groundnut promotion strategies for Bundelkhand. And in recent year, we have identified 7 varieties which is performing very good in this region, which is from ICRISAT and IIGR, Junagadh, Nityaharita, Grinar 4, 5, Kadri Lipakchi, J87, Raj Mungfali 4, and VRI 12. Previously, I have mentioned the major breeding objective for this region is development of short duration, high yielding cultivar. But farmers of this region is also want confectionery purpose varieties. because it has high demand in this year. With the help of AAGB, we have grown. Last year, we have grown confectionery purpose varietal trial stage 1. So we will doing work on that. So in conclusion, there are 3 to 4 points which I want to highlight. We need to improve seeds chain, seed multiplication ratios, SSR, VR, varietal replacement rate, seed replacement rate, good agronomic practices, and introduction of mechanization and demonstration. It will increase yield up to 30 to 40% in Bundelkhand area. Simultaneously, we are also doing work on the development of short duration, high yielding varieties. So this is the picture of left side. I've shown the picture of groundnut farmers producing their own seed, and right side we have tested entries of IIGR and ICRISAT, one of the entries we have shown. And I acknowledge Dr. Rakesh, Dr. Yogeshwar, Dr. Yogendra, Anita, our Director Research, S.K. Chaturvedi sir, for research coordination, and also ICRISAT and DGR for supplying seed. Thank you, Dr. Manish, sir, organizing secretary of this event, for providing the stage for presenting this. Thank you. Thank you very much.
Thank you very much. A hand of clap for him, please. At this point, because of time constraints, we'll open it up for just 2 questions. 2 questions, anybody from the floor or even online? If there are no questions, thank you very much again. We will move to our last presentation for this session, which is going to be given by Sunitra Chowdhury.
Yes.
And the
Thank you,
topic
sir.
of presentation is integration of sensor-based technology for modernizing groundnut breeding and enhancing genetic gain.
No. As presentation of conference. Without taking much time, I'll just start with my presentation. So, lot has been spoken about genomics, genotyping, and all the biotechnological tool. Probably this will add a little bit towards the phenotyping, where there is a lot of constraints and somehow we are lacking the proper SOPs for actual phenotyping. So emphasizing there is a big gap on those precise rapid evaluation for selection in bottleneck, because the phenotypic tools are not as robust as we have for genotyping. So phenotyping this gap can be closed if we take technological help, and sensors are the first which can for this kind of keep moving. So just for example, if I have to measure plant height, there is not much technology in core of the R&D sector of research. We have to take our measuring scale and go to the field. So technology has advanced a lot, but how much it is utilized in research is a question. It's not like we don't have technology, we have, but the proper utilization and integration is a breeding pipe. So I think a lot has been emphasized that how phenotyping is complex. There is a lot of factors, E and M, and interactions of it. So not going to that, but then there are digital system available, there are models available, and also utilization, this 100 of technology which is available, how we can fit so that it can be focused on delivering the value to the end user rather than having a fancy technology for any type. So coming to those, it's also complexity is there, but it's also a opportunity to digitalize our system. But digitalization should be based on science-based, and it should address the actual research question what we are looking for. So there is a lot of big industry for plant phenotyping. It's not like our industry lack of phenotyping tools, but question is like fit for the purposes, like whether it fit to our purpose. The kind of phenotyping I'm looking for selections, effective selection, whether those fit that, whether it is a large scale, but whether it's fast, also whether it fits my budget. So answer like technology is there and are working very much. What we learned that everybody cannot work on all the technology, and best idea is to collaborate. So far, I want to set a context that it's a complex type, complex, especially for abiotic study, and for the purpose, one of the key things we learn over time that technology address my question I'm looking for. Partnership is a good way to start. So in my later part of presentation, I'm to emphasize little, start with the understanding of environment. What are the skill gaps, and then what are the traits which address those gaps, and how so that it can help programs. So understanding production environment, I think a few of the colleagues has already emphasized. So there are some static inputs in this modeling exercise. So climate data, or having that soil data are the static application. But to make it more dynamic, also need some other. I don't know. Yeah, so there are static parameters and also there are dynamic parameters. Important that when we create a model, we should include this dynamic parameters so that your model is more interactive and address and predict the actual environment, production environment parameters. So classic environment typing is already placed, and some of the dynamic parameter coefficients, if they are included, those TPs are more addressed to the relevant environment. So usually to make it more dynamic, we try to study also the key parameters, dynamic in nature, like these are the supply and demand ratio of soil. So you see there is a different kind of pattern. So when the moisture deplete early, it is like it causes the flowering stress. Moisture is deplete little later, like the flowering stress, and when it very, very early, it causes pre-flowering stress, and it is depleting little after, that is a post-flowering. Also, there is a pattern where we see there is an intermittent kind of stress when stress is there for a shorter period, but it get released. So some 3 years back, we have optimized this environment for groundnut. You can see these are the different colors, which are the major producing districts of India. And the color address that the homogeneous units of the production environment. Means the red one environment is very different than the green one environment. So what does it mean? Like when one environment is different than other environment, there are many key parameters which are different here. And this breeding strategy could be different for each environment. Maybe one breeding strategy may not address all the environment requirements. Some are well-versed, some are like little bit more stress, some are having different kind of biotic challenges. So this homogeneous units of production environment are very, very key to understand the environment and their actual constraints. So once we have this environment homogeneous unit characterized, then we try to see more parameters, what kind of soil stress is there, and all the I was trying to identify. So the second graph, you can see that this is the environment where we talk always about the genetic enhancing the yield, but sometimes our environments are also very constraints. They may not allow, no matter how best genetics is put there, but the environment itself has a constraint that it cannot increase the yield as much as we have in well-versed environment. So this understanding how much drought is there and how much potential for the environment, we can strategize our breeding pipeline for that. So this is one of the example coming from our WCA that some of the locations, if I'm taking a multi-location trial, I should know which locations have this, which kind of stress is going right there. And accordingly, those kind of traits should be selected, which can address those kind of stresses. Coming to the genetics, so one of the physiologists' favorite structure which they follow is like yield is the top of all these small traits which interact, and then yield is a consequence of that. So focusing on this small traits is more beneficial than those on big trait, because if your building blocks are not selected, those allelic frequency for this building blocks is not increased in your selected material, then probably to achieve this one will be a challenge. So seeing that, we built some facilities which are around, which captures this, and some of the facility which I'm going to talk about is one is the LEASY scan, which captures the canopy dynamics and water dynamics, and then we have a lysimetric system which captures the water use, water uptake, and then field. So our upside down, which address first the number of lines, which is higher in number, goes to platform, high throughput platform, but the number of lines goes to field is lesser, and but the percentage of desirable alleles are higher than what goes to field. So this is just an aerial view of our phenotyping facility. It's a large scale phenotyping facility. And just moving to that, one of these LEASY scan is equipped with RGB and NERS bands. It has 3D point cloud technology, which captures the point, create the virtual image, and algorithms are set. It also has the load cells, which capture the transpiration dynamics of this. And because it has a NERS range, some of the traits which are related with foliar diseases, we are trying to connect with this. Just a view. So LEASY scan has not only it's a decade-old platform we are using for different purposes, just to show that it has captured a lot of genetic variability in different crops also, and some of this usefulness you can see with some of the cases. So why different patterns of water use is available? If we see, it's just because the canopy and canopy dynamics which bring differences in their water use uptake. And that's why we try to use early vigor trait, which is based on the leaf area and leaf area conductivity in groundnut. So we, all the F6 material we try to capture in our platform with, sorry. So all early F6 material we screen here, which is large in population for early vigor, and same material then it is those contrasting material which is available, the huge genetic variability available in this material. And also the elites are always kept, and some of the breeding selections from previous generations, you can see they are also towards the high. So we follow the first step in LEASY scan, and the selected material goes for managed stress environment in the field where different kind of stress is given. And then we, as some of our breeding colleagues have already presented, then drought screening is done. So the key message is here is like one-step selection may not work out. You have to take for 3 steps of selections where you can increase the traits which are at component traits of adaptation in your selected material. So we use platforms first, and then we try to increase the allelic frequency there. Those component traits are in place, and then we test them in managed stress environment. And the selected material then finally goes to multi-location trials. So all the MSc we are trying to integrate with UAVs so that the phenotyping is easier, and also if phenotyping can uniform across. So this is some of the examples and some of the algorithms which are set for the groundnuts. You can see some of the traits with very high correlation we can predict with the UAVs, and simple sensors are working for us so far. So second is like, what is drought tolerant? So some of our key here is like, one is like plant with enough water to fill the grains. So if soil moisture is enough to fill the grain, they are tolerant. And second, no more water is left after grains. So they should not be very highly conservative, that it did not fetch the yield, as we are interested in yield, so that we have to also balance the traits which are not conservative and not leveraging type. So tap more water. For that, early vigor is selected. We are using it. And then for actual tolerance mechanism, we are testing them in different platforms, field and lysimetric system, so that adaptation is actually enhanced there. So one more for trait dissection, we are using lysimetric facility, which is again a large scale facility where individual profile is given to each plant to grow, and also calculated water is given, and also we calculate how much they are transpiring, and per unit of transpired water, how much biomass and grain has been accumulated. So it is a very classical old technology, but that very effective to capture the resource use along with the agronomy. So usually our principle is like, most of the time plant don't feel stress until the 60% of soil moisture is depleted. So stage 1 will not differentiate the material very quickly. Stage 2 which differentiate the material, and stage 3 the survival. So we don't want to select from stage 3. So most of the selection is based on stage 2, and if there are some better adapted which can perform in stage 1, will be good for us. So VPD is another where the environment is very demanding, and sometimes soil moisture is available, still plants are not able to transpire. So these two traits are very much linked with each other, and that's why we try to see. So you can see the water uptake and transpiration efficiency are no trade-offs. They are not related. You will find the one which utilize a lot of water, but they are not very efficient. And under stress, you see their water uptake is cut down, but then still they have a variability for tea. So these are the traits which can exploit under drought breeding. Also, the tea relation, you can see when environment is very favorable, tea doesn't have any role in adaptation. But when the environment is very demanding, tea has direct role in your yield parameter. So stages are also important, like how the water uptake and all those, and that's why we exploit these traits drought breeding programs. So with this kind of technology, we can cut down all this kind of hard work, and we can use this high throughput phenotyping facilities for our better selections. Coming to some of the sensors which we are also using for post-harvest and grain nutrition technology. One is NIRS for high oleic acid. You see that the time is a sensitive in all the breeding programs. So if any chemical-based method to assess the fatty acid is very, very time-taking, so whether we can use any of the sensors which can do a fast, rapid assessment of our nutritional parameters. So in that, we are using NIRS, near infrared spectroscopy, which has a 400 to 2400 nanometer of range. And we have exploited. ICRISAT is using from more than a decade, and it is very high throughput, like 8 samples if I go with wet lab, but with this sensors, 250 samples easily I can do per day. So in this way, all the high oleic selections has been done in ICRISAT with using this benchtop technology for that, and almost 7,000 sample has been screened because our prediction models were very good and close to that. Enhancing further, we are also using the mobile version of this NIRS technology, which can not only address the bleeding, but it can go to also the value chain system. So this has a still higher capacity, 300 samples can be done. And one of the pilot study where we buyback policy was applied in one of the districts, and they gave the Girnar and they asked the farmer to bring it back. And we applied this handheld sensors to test, and premium price were paid. So some of the farmers, without knowing, initially they mixed, and the oleic content was lower on that. So, but next year onwards, they learned that mixing is causing them penalty on their premium price. So next year onwards, they started bringing the pure oil. So there are other technology also we are using, like XRF for iron and zinc, and computer tomography. So I will just take 2 more minutes to explain. This is one of the new technology which we are applying for assessment of shelling percentage, which is computed tomography, and it is very effective so far. So it can scan, and algorithms, image analysis algorithms were set to predict those kernel weight and also the shell weight. Based on that, we are able to predict the shelling percentage and also kernel size. Later, breeding programs find it useful because it's non-destructive, and we return their seeds. So they started, they were looking for kernel grade, established the method for kernel grade also. So now if you see the material, breeding material, which is very less in quantity, we only take the pods, we scan them, and we return them. So effectiveness in terms of cost, if you see, it costs them $7.6 per sample. But if they go through all 3-4 trades with just scan and return of their seeds, so it is quite cheaper, $2.4 per sample. Similarly, Harvest Master also, because when groundnut is harvested, it is always harvested at different moisture level. And if we can correct it for along with weight, it gives the moisture level. And if you correct your yield for moisture, then you are making more precise selection and generating more better data for that. So we build the calibration model for Harvest Master, and it is also fetching the good. It is also integrated in breeding pipeline. So this is my final slide, where in different stages, different technology has been integrated. So in F6, LithiScan and the selected material is also going for MSc trials, where we are trying to integrate some more technology. NERS is already getting used from F5 for selection for high oleic and CT for later stages. Thank you.
Thank you very much. Like to open it up for 2 questions, if any. Any questions from the audience? If there are no questions, thank you very much. And another round of applause for all the presenters today. Would also like to thank the audience for your patience, for your active listening and participation. And that lasts our last presentation for this session. And I will pass the mic over to the co-chair for maybe your last remark.
Thank you. This was a diverse session, last session. We had different transformers. Good. And I thank all of them who were online and private. And I thank you all. Thank you.
Ladies and gentlemen, we conclude the session here. And thank you so much for very cooperative chair and co-chair with our last moment changes, and congratulating and thanking all the speakers who have done their wonderful job. Ladies and gentlemen, that is a good wrap. First 3 days was a sprint of greater wisdom, knowledge, research, innovation in peanut research history, converging with present tech nuances, laying a red carpet for future peanut improvement. This is a big takeaway message from this AAGB 2025. With this, a round of applause for all of us for being present here and contributing in a bigger way for this AAGB 2025. With this, now I invite the chair, AAGB 2025, Dr. Manish Pandey, for the final wrap and concluding remarks. Thank you, one and all.
Thanks, Shruti, and thanks to everyone, of course. And then we had wonderful 3 days. So we started with plenary, and till now the presentation from phenotyping, that is key for all aspects of genomics that we do. So we covered most of the topics we can related to it. So now we have at least used this platform to discuss the updates. But now it's time to thank the people who have helped in organizing this conference right from the beginning. So I will say, as we reach the conclusion of this 13th AAGB 2025 conference, I take immense pride and gratitude in addressing you all at this closing ceremony. Over the past few days, we have witnessed a confluence of brilliant minds, groundbreaking discoveries, research innovations, and inspiring discussions that we had not just during the presentations, but also panel discussions, and also during plenary sessions. We are pleased to share that we have confirmation of attending this conference by how many people? Any guesses from the audience total, in person and online? Any guess? Okay, I will tell. So we have confirmation for attending around 250 people, around 120 on the first day, I mean say in person. Then we have like 150 online, and there are group, basically like in one group there are 30 people. If I have to name, like oil crop research shoots, there is like good, and they're sitting in one conference, or I mean to say meeting room, watching Sandong Academy of Agriculture Sciences, and like you have in Fujian Agriculture University there, I mean to say, and Guangzhou. So all those that they use this online platform. So all the issues that was related to whether few of the partners could not come, either because of many reasons, but mostly if you say funds and the visa issues. So I think this technology has solved. So that has not stopped us sharing information. So that's a big achievement for all of us. We are happy that we could connect with all these people. Yeah, that's what I said. And we had around 10 sessions, including 3 inaugural plenaries, 36 lead presentation. And you might have noticed that we had flash talk presentations from 13 young generation minds to listen. And that is a good experience for them. Maybe they are the future generation that they will be taking up this crop further. So a big congratulations to those young guys, their energy they brought to this conference. That was very exciting. And then I'm telling you, I got very good feedback from many, including our DG and our DDGR and ADG that session was really, really very good. They seen first time, like say, in the conferences in India, especially. So that was a good, like say, feedback for us. And these exposures, it was like sometimes you see even like poster elevators, the time given was not enough, and they were struggling, like say, to see. But our poster evaluators, so hats off to them. If you see, Kenta, Ryan, wow, now, and then our ABCD, means Dr. Ajay BC, that presentation you have heard, so we call him ABCD. So all these people like say posters, and then flash talks. It's not easy to pick the best ones when you have those people, because everybody has some of the things good, very difficult. And if you might have remembered, our decision, the panel from it was quick. They were just calculating those, taking out means. So hats off to those evaluators. They have done really, thank you. It's that you gave your assessment. It was really very quick and it helped us in taking decision. We did not to print. So actually we printed all the certificates for all the potential candidates. So that we can the pick which is selected and rest we can destroy. Okay, now let me start thanking our management. If you see the okay, before that, this conference has provided, and you know all, invaluable platform for researchers, scientists. You saw the industry leaders as well, of course, young scholars to share their cutting edge work. They have been like good discussion all those around. And also like it is good like exploring innovative solution to enhance productivity. That was like many tough questions being asked. If you remember, more was about aflatoxin content. So still that is a big challenge and there are some hard discussions happening whether it is possible, not possible, and genetic solution is there. So that was like a tough talk, and then we should accept those challenges in our future research. Let me say that thank to our management, DG, DDG, if you see our research program director, Konmej, he could not come, but his support was there right from the beginning. We got from our DDG stand, he was there, and because of him, he said that I want to be there when flash talks are there. He just had his flight according to schedule. So that like really shows good. And he interacted with, if you might have noticed, you all left, but he was engaging with research scholar that I want to talk. That shows. And then Dr. Himanshu Pathak, DG, because even he was busy, he is a new joinee in our institute. He has so much engagement at the beginning, but he said, okay, I will come. Because it was Sunday, so you can say Sunday helped us because it is a free time, personal time. So we all gave our personal time to those days. That is why we assembled so many people. So thank you to them. And even like we had inaugural session, so I would like to say all these team guests that we had on the dais, whether it's Dr. Himanshu Pathak, Dr. Sanjeev Gupta, Dr. Sandeep Bera, Stanford Blade, Dr. Ravi Mathur. We had here like Lepisier, so right from beginning to now, so there's his support. Charles, he has been like a big supporter from previous, but it's thank you, he has been here. Our old friend, but he's not old, neither me. So our old friend Haile, and then Boshulia, of course, he is one of the like 40, 45 years of time he has given. So his support, he was available online. So thank you to Dr. Boshulia. Now we have Steve Brown, who is co-chair of this conference. He could not come here, but he was awake to give message. And also thanks to Bauju Gyo, you have seen that it was late for them, but he did not wanted to even share like recorded presentation. He said, I will be whatever time it is there, I will be there online, and I want to give live presentation. So that was like a good, I mean to say effort and engagement that you can see how the people are related, I mean to say, with groundnut. It's really good. And then we thank Dr. Bauju Go for all these things and Steve Brown. Those were online, they were available and they made their best to share their views, presentations, and information with us. Our sincere thanks to those plenary speakers, of course, again, Dr. Sanjeev Gupta, Dr. Sonmej, and Sandeep Bira, that gave like a basic of how it is happening, especially in India, on this aspect. I extend our heartfelt gratitude to even panelists. It was very engaging discussions, and our moderator. Not only that you saw moderator during panel discussion, but how the moderation by Sunil was done during even young presentation, it was done. So that is why we put up young ones to handle the young ones, so they can read their minds and handle accordingly. And we acknowledge the support of Peanut Research Foundation, Steve Brown, not in just co-chair, but they really support right from the 1.5 year. We got information basically that we have to organize this conference in Goa. Many of them wanted to come. We are feeling very sorry that they are not there because We wanted that they should come here, they see a different part of India, enjoy some personal life also. And then, of course, we can discussion, but not, we should not be discouraged. We will find another opportunity to have some other conferences at this time and we'll bring them here. So now coming back to, you see, we have 3 days engaging. Thanks to the National Peanut Board, USA, we have all three sitting here, right, from morning to third day of evening. So thank to all of you for coming over, providing support and being with us. It means a lot. So thank you to all of you and the US Peanut Board. Now, if we are really very humbled. You have seen that we facilitated Dr. Nigam. Really, he is always available. So that's like, we are very much, he is stalwart of groundnut breeding that we know. But we are thankful for his support all the time. So we'd like to thank him. We have marveled with energy and vibe he set in there. So that's always there. Whether during the conference, and you might have seen, if you know him personally, more than that, outside the conference. Now, we have here in this room one person. His name is Dr. Ramesh Bhatt. Once we discussed that we are doing this, and though he said, whatever task is assigned, just don't hesitate to me. So we gave him one of the most tough job to arrange the field visit for some people who were interested. And he has, identified, how many fields, like you have options. You can have a field visit in 3 hours. You can have field visit in 6 hours. You can have field visit in 9 hours. So he had all the 3 options ready based on the requirement time we have with the candidate. And he has facilitated that field visit. So thank you, Dr. Bhatt, for all your efforts. Now, if you see, we have not fundraising is very important. So not like we got very good support from Dr. Hari Sudni, Janila, Kunil is sitting here, Sri Swati, Rajiv Rai Chaudhary is here. And these all, they helped us basically contacting people and all those things. That's a great support. And without funding, we are helping us in bringing some sponsorship for the conferences. And again, what I'm saying, again, I'm coming to Ryan and Naveen and Ajay and Kenta. I'm really that, because you should have been rewarded much better the job you have done, because the 2, 3 hours, 4 hours continuously you engaged with those posters, and then that one, so I can assume really it's a great, say, support and help to us. Very, very much thankful we are for you. This. And congratulations to all the winners. You will relish and cherish, and we also filled with the energy of your engagement and presentation during posters. So we want to thank all the young researchers that you did a wonderful job. All of us are happy and proud of you that we have such a talent here in the conference. So thank you to all of you. A special thank to, MC, Shruti Piliyappa, right? Shruti, you heard. So now you have seen a big one. He has done wonderful job. So even one of the event partner came meeting me here. He said, which company she is in there, I want to hire her or in her company, event company. So I mean to say it was like very professional. And when I told that this much minimum salary you have to give so that I can talk to her, then he went away from here. Okay, coming back to our International Advisory Board and Organizing Committee. So we took help whether in consultation, how to make program, venue, we kept on discussing a technical program, like lot of engagement from Dr. Kuldeep Singh, you might have seen here, but it was like he was having heavily engagement in deciding where to keep sessions and all those things. So very much thankful to you. We will be sharing abstract book where our team has worked. Sunita will be here. Just he has finished presentation. So we have wonderful abstract book which we'll be sharing with all of you. We'll put the final program also inside so that that will be for official record and share with you. Now, to our sponsors, it's a big one. This is all because of our sponsors that we are sitting here for 3 days. We are discussing science, but actually they are behind the scene. They have provided the opportunity to be here and comfortably discuss about science that they can take forward to the industry, farmers, and others. We would like to thank Mars. Mars not in just conference, but it has been a great supporter, even like decades with ICRISAT. But even like when we asked for conference, so they have been the first one to confirm that whatever it is, Mars is always there, will support you. So without any hesitation. So thank you. Here we have with us Arun Shetty is standing there. So thanks to Mars we have here. Now also we are very much thankful to Saga, South African, you see, groundnut association that we have, Dr. Hector Lejaras. So they are not here, but we are thanking them a lot with the same spirit, and thanks to them for funding us. Then we had, right from the beginning, you might have seen Mr. Hemang Bakshi, and with his team, you see some bottles there, right, from Bombay Super Seeds. Yeah, so like, and panel discussions. It's not like just they funded us. I mentioned about Mars, and then like say Bombay Super Seeds. They're really like interested in science, not just like marketing or products. So that's really interesting to us. Thank you. Thanks again. We had green crop science seeds, Mr. Suresh Patel and Yash Kumar Patel. But I'm not sure whether they are there now. They left, but we are very thankful for them. They have been very supportive. We had DCM Sriram. They have very limited operation in groundnut, and we had here Dr. O.P. Sringi and Dhanashwar also. So we are very much thankful, sir, to you for all our support, and it was a great support. So many thanks to you. Then we had like Anand Kumar also from Takara. They are not here as stall, but their support is there. We had Pawan Kumar. I think he just left. He's not here as stall. So for their support, and Premas Life Sciences, who are also not there, but they are all like the sponsors because of whom we could take care of partial, say expenditure related to this conference venue and other related, to say related expenditures. So thanks to all our sponsors. Thank you. I also acknowledge and appreciate the support of different departments, because before coming here, we took help from different people from ICRISAT, whether it's a finance, whether it's a DG or DDG office to follow up, whether it's an IP office for visa related issue, or travel office for arranging tickets and all those things. So thanks to them for their support and help for related to this conference. I would like to extend my gratitude to the technical and logistic teams, volunteers, and administrative staff, especially the work that been done by excellent job with very much dedication. I'm very much appreciative. I think they did wonderful job beyond my expectation, I will say, because they are all part of my team. Whether it's a Vinay Sarma, if you're there, you can stand and raise your hand. Whether it's a Virendra, if he's there here, he can stand and raise hand. We had Nikhil. Are you here? Yes, this gentleman. We had a Reservatol content girl, Madhvi, somewhere here. Yes, she is here. We had Namita. You might have seen she is hiding somewhere there. Okay, so now she's there. Otherwise, she has been there. So for online every step. So that was really appreciated. And now we had Priya. You might have seen sometimes presentations, uploading it. When we were asking recorded video, he has to check with Sagar to see whether the sound is okay, video is okay, or the timeline is fine. Basically, the video we received. So that like basically it's good. So thanks. We had Haja, Suresh, Hema Bindu to support. So these were like, so can you stand and just raise your hands? So they did really wonderful job. Wherever you put, they had specific jobs, but in addition to that, they were assigned some tasks and they have done. And they are all research scholars. So I feel they might have learned even in future. I will say it will be useful. When you are organizing any event, now you will get more confidence in doing so. So I will say it is in both ways. We received help and you got to learn how to organize an event. So thank you thank you to all of. Now, so this is like 1 month, 2 months, but we had 3 hour musketeers. I am not musketeers. You know whom? We have been using their, I mean to say, their support and help from 1.5 years, the time we started working on AAGB. And those are Anjaya, who is our admin in our office. Anjaya, are you there? Yes. So all admin mails and something, if you see, if you just run here and there, I hope you reduced some of your weight also, maybe 500 grams during this. So he was running here then behind. So Anjaya is one. We have here Rajiv Roy Chaudhary. Can you just please stand up? Sunil Gangurde here. So each of these they handled right from the beginning. Suppose like say a technical program like Rajiv from there, abstract book there, and then Sunil on finances, basically how much it is coming. So maybe you're becoming good in math as well. So that is so because of this, I mean to say it was a lot of time we have taken during this one, and they're like in I will say they have completed their PhDs, both of their like postdocs. Sunil will be joining Mage Molecular Geneticist in CIMMIT in China next month. Now, a special thanks to Miss Rebecca. If you might have seen, she was sitting there. She was taking care of like transport, discussing with Meti, and there it's a great support. Now, since we agreed to organize this conference right from the beginning, we want, because it was happening in Goa, so we had a CRISAT office in Hyderabad. So we wanted a good event partner. And with METI, we had already organized many conferences before. So we had confidence in METI to that, okay, we can give task. And we did not pay any money, to METI in the start. So they have to do some investment, some advance, and all those things. So even without, I mean to say, moving forward, paying anything, they took care of like websites, many other expenditures. So because of the just confidence in us between and then working relationship among the conferences. So you have seen like we worked with METI and around 1.5 years, I mean to say till now, and maybe we'll continue few months for this websites and other things. It was impossible actually to organize without your support. I will say from the METI. We have here like, and that METI private limited, basically it is led by Tirupati, who could not come unfortunately because he has to go for travel to Sri Lanka. Tirupati has not come, but his team is here who have taken care of everything, whether it's like website, registration, kids, accommodation, or local logistics. We extend thanks to Venkat. Venkat, are you here? Can you stand up? We have Shruti, Vinod, Anish, and Shloka. I will request to all of you from the METI to please come on to the stage. We have it to show that our token of love and appreciation for you. Can you please come to stage? Take step. We also thank that all the support we got from Novotel Hotel where we are currently. So they have done wonderful job. It's the food or you saw the facility here, accommodation. So I think everything was fine. So thanks to them, Novotel Hotel staff. I hope you had good time in Goa. You might got some time to see around, I mean to say, streets and then some beaches. Hope you enjoyed some time there. We tried our best to provide best experience during the conference to all of you. The entire credit goes to my team and event partner Meety. In case you faced any problem, I take responsibility and apologize. We will do better next time. As we conclude AGB 2025, let us carry forward the spirit of collaboration and innovation. That's one of the most important aspects. That is the conference has fostered in us. The discussions and connections made here will undoubtedly shape the future of groundnut research and its impact on global agriculture. I encourage all of you to stay connected because that's most important. Most of the times we forget, we just be in comfort zone. I will say don't be in comfort zone. Keep the connections going, moving. If somebody is not contact replying, send the mail next time. Okay, until you get the response. Okay, that's way that we have to collaborate more and more. I encourage all of you to stay connected and continue pioneering research. That's more important because we are working for the farming and the farming community, especially the groundnut. So we have to be say not to shy away with doing, I mean to say getting connected or making effort, whatever is needed for your research. I mean to say impactful work that you are doing. And with that, I officially declare that the 13th International Conference on Advances in Arachis through Genomics and Biotechnology is closed. Thank you and safe travels to all of you. Thank you.
Requesting everybody for to gather here for a group photo, and then we can disperse having our tea and coffee. Please join us for the group photo.